that so many people had during the last couple of years. But that move was no excuse for disappearing from the H3Africa community without a word to any of my African colleagues. So I'm now glad to have a second chance to say farewell to this exceptional group of scientists and to say, very well done. I have actually not kept track with much science since moving, but I do pay attention to anything coming out of Africa because I consider H3Africa to be one of the two highlights of my career at NIH, the other being the Human Genome Project, of course. But in some ways, I'm prouder of my involvement in H3Africa than in the Genome Project because I feel I was even more involved in helping to develop and implement the concept of this wonderful effort. Of course, most of the credit belongs to you who actually created the H3Africa community and did the outstanding research that the project has accomplished. You were the ones who proved the skeptics, and there were many at the start, were wrong, and that such a complex collaborative effort could be assembled and could thrive. We said at the beginning that however much H3Africa accomplished during its lifetime, it would not be a success unless the research community it fostered became sustainable. Well, that has happened, and I think we can now truly say that H3Africa has been a major success, and you are all to be heartily congratulated. Thank you for the collegiality and friendship, and good luck to all of you going forward. You have earned the respect of all of us. Bye.
Hello, everyone. My name is Sonia Abdelak. I'm head of the Laboratory of Biomedical Genomics and Oncogenetics. Our lab is based in Institut Pasteur in Tunis, and I'm very happy and honored to share with you what are the significance and impact of H3Africa for me. Actually, I had the chance to see H3Africa project even before its creation, when it was a project idea proposed by Professor Charles Rotimi as the former president of the African Society of Human Genetics. And I'm really very impressed by the way the dream of harnessing genomics in Africa is becoming progressively true. I have to say also that I have the chance to see concrete results, in particular for members of my research team or close collaborators that benefited from the trainings that were conducted in the frame of H3Africa projects, in particular in the H3A BioNet. What does H3Africa means for me? It means generosity, and it also means solidarity. In order to be more precise and translate these values into examples of concrete actions, let me give you some examples. I saw several young researchers involved in this project. They acquired new competencies and they shared them with others. They acquired not only competencies in genomics and bioinformatics, but also developed their competencies to work in huge networks with international and multicultural teams. They learned also how to write publications with big teams. They learned how to be proactive and work within teams at the level of the African continent with no frontiers or boundaries. Several former PhD students had better chance to get recruited. For the moment, most of them, they succeeded in having positions outside the country. We need really to continue the efforts and go together for new ambitious projects. My aim today is to provide better working conditions in Africa for the young African researchers and future leaders who would be able to translate their knowledge into better health systems. we need to invest in the future. We really need to invest for the future, for the benefit of the African populations, and to reinforce the way they have paved for the precision medicine in Africa. I would just like to express my gratitude to all who made this happen, from whenever they are, at the strategic or managerial or operational levels. Thank you very much to all of you, and see you soon, I hope. Bye-bye.
I would like to seize this opportunity to congratulate the H3Africa Consortium for a wonderful run bringing genomics revolution to the African continent. My name is Fumi Olopade, and I'm a professor of medicine and human genetics at the University of Chicago. This project started from investigators at the meeting of the African Society of Human Genetics, which really started as an initiative from my good friend, Charles Rotimi. I remember the first meeting I attended, traveling from Nigeria to Douala to Yaoundé, running into travel challenges and having a bus breakdown on the road between Douala and Yaoundé. It was a harrowing experience, but it was worth it, because when I got to Yaoundé, I met very many interesting people who were focused on bringing the genetics revolution to Africa. Congratulations to all of you. And the completion of the H3Africa initiative is really a momentous period for us, especially coming after the COVID-19 pandemic, where we've all been shut down in our own countries. And yet, through the resourcefulness of scientists, we've been able, through sheer determination, collaboration, and participation by scientists all over the globe, we've been able to bring vaccines to populations that would never have seen vaccines at warp speed. While we know that there's inequity in the distribution of vaccines, we really must go back to what Francis Collins, an inspirational leader, had in mind, thinking about the Genome Project, its implications for society, and then realizing that H3Africa had to be done because the Human Genome Project is not completed. It was just the foundation. And then the foundation really meant that we needed to do more research. And that research really is fundamental to understanding the root causes of health disparities. And I know that when we started, I really wanted us to study sickle cell disease. And I must really congratulate Professor Ambrose Wonkman, Julie Makani, Professor Ofori Akua, and Professor Unodu, who really came together. I met them at the meeting in Yaoundé, and they were determined to bring genomics revolution to the study of sickle cell disease. And now we have the opportunity to have countries have access to genetic testing, to genetic counseling, and soon to gene therapy for sickle cell disease, because Africans came together, Africans in the diasporas, Black people all over the world said, we're going to do genomics research, and we're going to do genomics research to improve the health of populations of African ancestry. And so while we understand that there's so many societal issues while we understand that we have to think about the ethical, legal, and social implications of genomics research, we also know that by doing this, we're going to have more people engaged in computational biology. We're going to have resources on the continent, and there's going to be technological advancement. There's going to be real movement, real innovation in the field. So I really want to thank the NIH and all of those who had the foresight to focus on training the next generation of scholars. And now, when I started my career under Janet Rowley, when I went to the first Gordon conference and I met Francis Collins and Mary Claire King, they transformed my career from studying somatic genetics to actually really studying germline genetics. And now in our lab, we're integrating germline genetics to dissect the complex evolutionary trajectory of breast cancer among people of African ancestry. And we can't do that work fast enough because people are dying because they don't have access to life-saving medications, life-saving cancer medicines, life-saving precision medicine, precision oncology. And so with the evolution of data science initiative in Africa, with all of you coming together and moving the needle, with companies like 54 Genes and all of the other innovators that are in Africa, thinking about how to innovate ourselves out of poverty, innovate ourselves out of poor health, innovate ourselves out of not participating in global research. I'm very optimistic. I am actually happy that this moment has happened, that countries will now take up what H3Africa has left behind, that it will no longer be necessary for the United States or Wellcome Start or other countries to fund research and innovation in Africa, that Africans will do this work, that Africans will do this research, and that the research will not only benefit humanity, but it will benefit society in Africa, and it will benefit the health of all populations of African descent. And as you know, all of us came out of Africa. So thank you for doing work that has significant implications for all of medicine and all of us on the planet. Congratulations.
And is coming in from is a Zoom presentation. It is Karina Schlebusch, who is Dr. Karina Schlebusch, who's an associate professor at the University of Uppsala, whose primary research is focused on understanding evolutionary genomics of humans. The title of her presentation is African History, Inferences from Modern and Ancient DNA.
Hello, everybody. I apologize for not being with you today in person. However, after this talk, I will be available online for questions. Thank you very much to the organizers for inviting me for the talk today. Today, I'm going to talk about African history, inferences from modern and ancient DNA. We, as a species, human beings, wonder about our history. We wonder about our ancestors, where our ancestors lived, how they interacted with each other, and basically how they became us. And this is an area of investigation, a discipline of investigation for many different subject areas. For instance, archaeology, paleoanthropology, linguistics. These are all disciplines that have investigated this question for decades, if not centuries. Genetics is another way of how we can investigate this question of human history. But genetics is a relatively recent addition to the field. However, genetics already have answered quite a lot of questions that were posed by some of these other fields regarding how humans evolved and also migrated to populate the planet. We know now that humans evolved in Africa, and genetics has shown that the highest genetic diversities are seen in African populations. Thereafter, humans exited Africa relatively recently, around 60,000 years ago. Fully modern humans exited Africa and populated the rest of the world. During these migrations across the different continents, they met with archaic humans that exited Africa earlier, for instance, Neanderthal and Denisovans. And thanks to genetics, we know that modern humans actually admixed with these archaic groups, and all non-African humans carry a little bit of Neanderthal admixture, and people from Southeast Asia carry Denisovan admixture. We see this migration of people across the globe very clearly in genetics patterns. And this pattern is encapsulated by what we call the serial founder model. In this model, populations in Africa has the highest genetic diversities, and then diversities of populations decrease as we go with distance from Africa. So using genetics together with all of the other disciplines like linguistics, anthropology, and also archaeology, we have started to form a very good picture of how humans interacted in the past and how they migrated and formed the populations that live across the globe today. For instance, in places like Europe, we also have a lot of inferences from ancient DNA that helped resolve questions, for instance, like how farming influenced the populations, the current day populations in Europe, and how different farming migrations influenced population structure in Europe. Up until recently, as you see here, not so much was known regarding the genetic history of Africa, but we are learning more and more. And from various genetic studies, modern DNA and also ancient DNA studies, we've learned more about the history of African populations. And what we see for Africa is that we have two clear, distinct phases. We have the deep human history in Africa. And this deep human history is studied by studying hunter-gatherer groups across the African continent, and also ancient DNA that give us access to these deeper histories of African populations. Hunter-gatherer groups are ideal to study this deeper history of Africa because hunter-gatherer groups have lived for very long times in places or geographic regions that they are found today as well. And this is in stark contrast with more recent migrations of populations connected to farming. Farming usually causes expansions, very fast expansions in populations, and then these expansions are followed by migrations. And there are large-scale migrations across the African continent due to the expansion of farming populations. The most well-known farming expansion, perhaps the Bantu expansion, where populations adopted farming and developed farming practices around 5,000 to 3,000 years ago and started to expand across the African continent in such a way that all of the majority populations in sub-equatorial African countries today are Bantu speakers. However, these migration patterns was also complex, and ancient DNA can also help us to resolve these complex patterns related to farming migrations. I'm going to give a few examples of studies that have looked into population structure of Africa, both the deep population structure and the more recent farming migrations, using both modern day genetic variation and also ancient DNA studies in combination with each other. So regarding population structure for modern day variation, I'm going to use this study as an example that I was involved in almost 10 years ago now. This is a study where we included genetic data from many more hunter-gatherer groups across the African continent to especially look at deep population structure of the African continent. There are also other seminal studies during the same time period that also provided valuable insights, but our study agree more or less with these studies regarding inferences of population structure in Africa. What we saw from this study is that if you represent African populations in the form of a tree, this is more or less the tree that we see and how different populations are related to each other in Africa. So we see a tree with the first split in the tree between the South African San groups, hunter-gatherer groups in Southern Africa. Then we see a split in the tree that have the Central African rainforest hunter-gatherers, and then a split between East and West Africans in the tree. And then as a subset of genetic variation in Northeast Africa, we have the genetic variation of all non-African populations. And you can see the split time here in the tree is around 100,000 years ago. And this was using the old mutation rate that we used previously. The new mutation rate that we use is half of the old mutation rate, basically, and this will double the date of the first split in the tree to around 200,000 years ago. However, I will come back to this specific date of the first split in the human tree. We also represented the genetic variation that we saw in the data set in 2 dimensions using principal component analysis. And what we saw is that the principal component analysis, so genetic variation in 2 dimensions, reflect the geography of the African continent. And we see that PC1 separated Khoisan hunter-gatherers from Southern Africa from other African populations. Then we see the genetic variation of West Africa in this corner and the genetic variation of East Africans in this corner. So more or less the north to south and the east to west variation axis is represented also in genetic variation. And then recent large-scale migrations, for instance, Bantu speakers who have West African ancestry, all are clustered in this corner with West African variation. However, you see that today there are Bantu speakers living in Southern Africa and East Africa as well. However, their genetic variation still map to this West African genetic variation. We also look at diversity patterns in African populations, and by using these summary statistics. So these various genetic summary statistics is just different ways of capturing diversity patterns in populations. So here we have 3 different summary statistics: haplotype, heterozygosity, haplotype richness, and linkage disequilibrium. And firstly, comparing African populations, the colored lines here with non-African genetic variation, the gray lines in these plots, you can clearly see that African populations have much higher genetic variation and much higher genetic diversity compared to non-African populations. And this clearly indicates the out of Africa bottleneck, where when populations exited Africa, a lot of genetic variation was lost. But the African population still retain a lot of the genetic diversity. And this was then also referring back to the serial founder model predicted by the model, where the most genetic diversity is in Africa and genetic diversity decrease with a distance from Africa. And then when you look at this pattern in the summary statistics, you can see various studies have looked at this. Heterozygosity decrease with a distance from Africa, linkage disequilibrium increase with a distance from Africa. All of this indicating that as you move away from Africa, genetic diversity decreases, as clearly shown by the genetic data in these different studies. So in this 2012 study, we then wondered whether these summary statistics can perhaps pinpoint the origin of modern humans in Africa. If there was a single point where modern humans originated and migrated to the rest of Africa, maybe we can use the genetic summary statistics in a similar way as the out of Africa migration to pinpoint where people originated in Africa. Modern humans originated in Africa and perhaps populated the rest of Africa. However, when we looked at these different summary statistics in our data set, 4 different ones: haplotype heterozygosity, haplotype richness, runs of homozygosity, and R-square, which is the L linkage disequilibrium measure, we see that these different summary statistics points to different places in Africa as having the highest diversity, therefore indicating a possible origins of modern humans. So it was definitely not a unanimous vote, if you can say it like that. Some summary statistics indicated Southern Africa, some of the summary statistics indicated West Africa, and some indicated East Africa. So basically all of the places where we had representative populations were possible places where humans could have originated in Africa if you just look at the highest genetic diversity. So definitely not a simple answer, not a clear pattern as we've seen for the out of Africa migration. So already this study on modern day groups have indicated that perhaps the demographic of the history of Africa is a bit more complex than the out of Africa migrations. We also investigated this further, deeper population structure using ancient DNA. So ancient DNA studies in Africa is relatively recent because ancient DNA in Africa doesn't preserve very well. The conditions in Africa is not so conducive for DNA to preserve for long times. The hot climates and in some places humid climates caused the DNA to break down. So ancient DNA technology was more successful earlier in places like Europe and the higher latitudes. But as technology developed, and both in the lab and computational technology, ancient DNA studies for Africa also became possible. And the first ancient DNA for Africa study was published in 2015. And since then, quite a few studies have been published, and we have 11 different ancient DNA studies published to date for Africa. Today, I'm going to discuss this specific study, which I and my group was involved in. In this study, you can see that we have this heading of Southern African ancient genomes estimate modern human divergence to 350 to 260,000 years ago. So this date is a bit earlier than the date I showed previously for our 2012 study. That using the new mutations rate, we estimated that earliest divergence to 200,000 years ago, while in this study we estimate earlier date. And the reason for that is explained in the next few slides. Here you can see a sampling tent. Ancient DNA studies needs to be done in very clean conditions because modern day contamination easily can swamp reactions for ancient DNA, so that it's very difficult to extract ancient DNA signals and discern that from signals of modern day DNA contaminations. And therefore, you need to work in very clean conditions when sampling ancient DNA, and you need specialized labs and equipment to do ancient DNA studies in. In this specific study, we looked at 7 ancient DNA individuals or 7 human remains in the eastern part of Southern Africa. And these remains are indicated by these symbols with the black lines surrounding them. And then current day comparative data sets have the lighter lines that surround the symbols. And looking at the ancient DNA samples and the carbon dates of these samples, you can see that 3 of the samples were older. They were around 2,000 years old, and 4 of the samples were younger, more recent samples, around 500 years old. When we analyzed these samples then together with the modern day comparative data, or first, when we sequenced these samples, we got quite well good coverage for some of the samples, up to 13 times coverage for certain samples. So almost high coverage genomes that we were able to sequence for some of the ancient DNA samples. Some didn't work as well. There only 36% of the genome is covered. However, these samples then gave us a very nice sample set to work with, and especially the high coverage individuals were very valuable for more specialized inferences. When we visualize the genetic variation of these ancient DNA individuals together with modern day comparative data sets, we saw that there is this divide between the individuals dated to more recent times and the individuals dated to older times. And we see that these green individuals, the more recent individuals, when we look at with which modern day groups they are grouping together with groups with a West African genetic background, for instance, the Yoruba here in dark green, but also Bantu speakers from South Africa here in light green. And because Bantu speakers migrated relatively recently from West Africa into Southern Africa, they still have this West African component. And these more recent individuals group with Bantu speakers, and the individuals from older time periods, around 2,000 years old, group with current day Khoisan groups, mostly living today in Botswana and Namibia. So this attests to this population replacement that happened in this part of Southern Africa. At earlier time periods, the individuals group with Khoisan, and more recent time periods, the individuals group with Bantu-speaking groups from the area. But to look a bit more into these Bantu-speaking individuals to see if we can see more differentiation when we group it into groups from the region, different Bantu-speaking groups. We did a recent study together with the group from Michelle Ramsey from the Sydney Brenner Institute of Molecular Biosciences. This study was actually to look at the population structure of South African Bantu speakers. And it was a huge group of different Bantu speakers across South Africa, where we look whether there is genetic substructure in Bantu speakers of Southern Africa. And what we saw when we look, for instance, at the species plots or the UMAP plots and compare it with the geography of where the different Bantu speaker groups live in South Africa. So these are the linguistic majority areas of different Bantu speaker groups. When we compare that with the genetic structure in 2 dimensions, we see there's quite a good correlation between the genetic substructure and also the geography of different Bantu speaker groups. But we also in this study look at where different ancient DNA individuals from the previous study group with regards to these different Bantu speaker groups. And we saw that different ancient DNA individuals group with different Bantu speaker groups and the ones that are a bit older, around 500 years old group, preferably with Tsonga individuals on the UMAP plot, and the ones that are a bit younger, around 400 years old, group with the Nguni groups. So again, we see some complexity regarding ancient DNA if you compare it to modern DNA, and there seems to be an indication that different groups arrived at different times in Southern Africa. And if you look at older ancient DNA, it groups with different groups compared to younger ancient DNA. But returning again to then this plot where we have the Bantu speakers with the West African variation and the Khoisan individuals that group with the modern day Khoisan individuals together with the ancient individuals that probably were ancestors of Khoisan, we see that all modern day Khoisan individuals are shifted towards these other PCs, and that the Khoisan or the Stone Age hunter-gatherers, ancient DNA individuals form the extreme of this specific PC. And this indicated the possibility that all modern day Khoisan individuals perhaps had admixture with other groups. And this is something that we wanted to test. And we did formal tests, admixture tests, to see whether it is actually true, whether there was admixture between Khoisan individuals and other surrounding neighboring groups. And we use the ancient DNA samples as reference points. We use a modern day sound group, the Ukwansi, that has the least admixed group to see if we can detect admixture. And indeed, we saw that all Khoisan groups have admixture with another group. And the Ukwansi, that's the least admixed, have 12% admixture. And the Nama, that is the Khoi group with the most admixture, have around 30% admixture. And that the admixture came from a group of East African individuals that migrated into Southern Africa around 2,000 years ago and introduced herding practices into Southern Africa. And this group had both East African and non-African genetic variation, and they mixed into all of the Khoisan groups, and the least admixed Khoisan group, the Ukwansi, received 12% admixture from this specific East African migration. However, the admixture of this East African herding groups into modern day Khoisan influenced the first split or the dating that we did previously for the first split in the tree of populations. And if you just look at the time in calendar years, if you date these different split times, you can see that the Khoisan split year based on modern day Khoisan in Southern Africa, in this case the Ukwansi, is shallower compared to if you use the ancient DNA that didn't receive any admixture. So the admixture in all modern day Khoisan groups influenced that previous split time that I showed based on our study on modern day groups. So we re-estimated then the split time between all populations, and we saw that this was then around 300,000 years ago. So we redraw this phylogeny then of different African groups, and we see here that the first split in the tree is between Khoisan groups and other African groups. So between this red branch and this turquoise branch, then we have the Central African rainforest hunter-gatherers, the East and West African split, and then the non-African split in the tree. But this time period of the first split in the tree is a very interesting time period. Also in the fossil record, if we look how humans look like living in Africa in this time period, these are the fossils of different regions in Africa in the time period between 700,000 years ago and present. We have North African, East African, and Southern African fossils. And we see that in all of these regions, the fossils look not yet fully modern human, but also not archaic anymore. So these were all human populations that were transitioning from archaic forms into fully modern forms. And this happened not just in one place, but it seems to have happened all across the African continent. Humans evolving from more archaic forms, anatomically more archaic forms into modern forms. So more and more people in the paleoanthropology community start to believe in pan-African origins of modern humans. So not just one place in Africa where modern humans originated, but perhaps several places, because the fossil evidence indicate this. So if we look at a genetic tree then like this, a population topology, how different population groups are related to each other on the African continent, it seems that it sort of coalesces in maybe one population. But the question is then whether genetics suggest a single place where humans originated in Africa. If you remember from that previous example that I showed of our 2012 study, where we analyzed genetic diversity, didn't look like that it points to one place where there's the highest genetic diversity. So if we look at a tree like this, then what does it really say? What does it really tell us? But to understand the population topology like this, it is important to look at the genetic data that underlies this population topology. For instance, if you do a phylogenetic tree of mitochondrial DNA, so mitochondrial DNA is a single locus, and you can build a phylogenetic tree of all mitochondrial DNA of basically everybody on Earth today, and then all of these mitochondrial DNA sequences will converge into a most recent common ancestor, and people have done this. And we know that the mitochondrial DNA most recent common ancestor lived around 200,000 years ago. And if we look at the diversity patterns, she lived either in Southern or Eastern Africa. The same can be done for the Y chromosome. And if we look at the Y chromosome, we can also build a tree, a Y chromosome, and infer where our Y chromosome Adam have lived. So one person that gave rise to all of the Y chromosome sequences that we see in the world today. And we can do that. And there is actually one Y chromosomal ancestor that lived around 400,000 years ago somewhere in West Africa. So we have a mitochondrial Eve and a Y chromosome Adam, but they lived in different places in Africa, and they also lived in different time periods. But they didn't live alone on the continent. There were many other people living with them. And these other people were the ancestors of our other chromosomes. So the mitochondria and Y chromosome is quite small compared to all of our other chromosomes. However, it's easy to analyze them because they don't recombine. They are inherited from one generation to the next without any recombination. So it's easy to build these trees for these single locus genetic variation. However, our other chromosomes recombine with each other and exchanges genetic material all the time. So one way, there's different ways of analyzing autosomal genetic variation, but one way is, for instance, to chop up the chromosomes in small parts and then build phylogenetic trees of all of these small parts on the chromosomes, like you do for the mitochondrial and Y chromosome, and then combine all of these small trees into a population topology like this. So if you look at a population topology or a population tree like this, it means there's many thousands of small trees that underlies this population topology. And like the mitochondrial and Y chromosome that had ancestors at different time periods and in different places in Africa, all of these thousands and thousands of population topologies that underlie the population tree in the end can have ancestors at different places and different time periods in Africa. So when we look at a population topology like this, it's not likely not a single origin, but a model like this can also underlie this population topology, a model that we call in genetics an isolation by distance model, where populations have lived across the African continent for very deep time periods, and then these populations had stepwise gene flow with each other. And populations that live the furthest from each other on the African continent will have the least amount of gene flow. But when you build a tree from these, a model like this, it will also result in a population topology like this, where populations that live the furthest from each other on the continent are the populations on the extremes of the topology. So this model then would correlate with the paleoanthropologist Pan-African origin, where populations in different parts of the continent evolved from archaic forms into modern forms, and then have gene flow with each other. And it's a possible model that can describe how humans evolved across the African continent. But to test these models, one needs to simulate genetic data and compare these simulated models with each other by fitting it to our data, our empirical data that we generate from modern day populations and from ancient DNA to see which model fit our data the best. And these are basically two extreme models. There can also be models in between these. Maybe one of these populations could have expanded and merged into these other populations, which is an in-between model. And also the gene flow events in between these populations can be more pulse-like and not continuous. These are all variations on these models that needs to be tested to see how human origins happened in Africa. So this is a question then that are being discussed in different fields, and also in the paleoanthropology field and also in the genetic field. This, for instance, is a review article written by both geneticists and paleoanthropologists. Did our species evolve in subdivided populations across Africa, and why does it matter? This is also a very nice review article written last year, again, by both paleoanthropologists and geneticists, addressing this question of the models of human origins in Africa. Here we have the long-standing Pan-African connectivity model, which is the same as the isolation by distance model that I discussed earlier. Then here is the single origin model, where one population expanded and basically replaced all of the other populations living in the continent. Here is a sort of in-between model where you have the long-standing connectivity or the isolation by distance, but then one population expands and merges with the other populations. However, what this review concluded was that we can reject, with our current evidence, we can most probably reject the single origin model, where modern humans just arose in one place and then replace all of the other humans in the different places in Africa. So just to summarize a few thoughts, Africa probably doesn't have a single origin of humans on the continent. It has a complex demography. It's most probably not this simple tree-like structure or single origin, but it may look like this complex river delta network that's different human populations split and merge throughout time and place. And what we need now is a rigorous testing of different models. We need to simulate different models like this And these models must be representative of the histories that we expect. And then we need to test these genetic summary statistics that is sensitive to detect differences between the models. And into this, we also need to integrate geographic and climatic modeling. And then also we need to incorporate ancient DNA studies into modern day genetic variation studies to get access to different time points in our human history. And then also farming expansions were complex events. And for instance, for the example that I showed for the Bantu expansion, ADNA can help us to resolve this complexity by accessing certain time points and seeing which modern day populations are connected to which time points when ancient DNA, when populations represented by different ancient DNA samples moved into the different areas. Then lastly, I just want to thank my group at Uppsala University and also the other groups in our program that investigates different questions regarding human history. And I also want to thank our collaborators from South Africa, Marlies Lombard, with whom we are collaborating with the ancient DNA studies, Imla Sudial for many of our modern day genetic DNA studies, and also Michelle Ramsey's team at the Sydney Brenner Institute of Molecular Biosciences, and also on all of the funding that we received to be able to do our research. Thank you very much.
Okay, great. Thank you. So I believe we have.
Hello, everybody.
Oh, great. Hi, Karina. Can you hear us?
Yes, I can hear.
Okay, thank you, Karina. So that was a fantastic presentation and a great summary of a lot of interesting data and complex scenarios. So I think we have time now for a number of questions from the audience here, and I believe we will have a microphone traveling. So do we have questions for Karina, please? Yes, one there in the very back. I can make the run if there isn't. Okay.
Hi, okay. Hi, Karina. Thank you so much for the presentation. I am Kevin Esso from the University of Cape Town, and I've been following your work with great interest with some of your other collaborators like Pontus. And because I'm so very much interested in ancient genomes. So I wonder whether there is ancient genomes for African populations that's freely available or publicly available, and how one can access them. And also, are these ancient genomes for African populations, do we have enough sample size to like maybe investigate adaptive signatures and maybe incorporate them with modern day genomes to maybe compare adaptive signatures for some genes that I'm like interested in, like the beta globin gene cluster? And also, I would just like to hear your thought on a concept that was recently published, I think Lipsin in 2020, about ghost archaic and ghost modern in Africa. I would like to hear your thought about that. What's the progress in trying to delineate what those ghost ancestries are? And also, just one more question. Yeah, I really am passionate about this subject, so I was really following very closely. And you talked about perhaps a Pan-African origin of African populations. So I would just like to, and the tree kind of like converged prior to 300,000 years ago. So is there like progress in maybe trying to understand the demographic or evolutionary history prior to 300,000 years ago, before we have this divergence of these populations? And lastly, our simulation methods that we have now, are they robust enough to maybe to simulate complex evolutionary models in the context of selection models like balanced polymorphism model, and maybe soft and hard sweep? So are these methods robust enough to capture the complex demographic history of African populations? Thank you.
Yep. Thank you. Yes, there's quite a lot of questions, so I hope I remember all of them. I didn't write them down, so please just remind me. So I think the first question was regarding the availability of ancient DNA, and all of the ancient DNA data is available. So in the publications, it's usually uploaded to the EGA database, and it's freely available then just to download from there. But there's also a very nice resource that is curated by David Reich's lab in America that also curates all of the ancient DNA studies that have been published to date. And it's also a very accessible format, and you can browse the database and download samples you're interested in. So either through EGA by looking at the references in the publication, or by using the Reich database, all of ancient DNA data sets are available. If you can just remind me the second question, I forgot now.
Do we have enough sample size for African?
Exactly. Yes, very, very good question. So for Europe, where there's a large ancient DNA sample sizes already, many people are doing selection studies. For instance, Ian Matheson have done very nice selection studies of how frequency distributions change during time. And for instance, for the lactase persistence gene, he actually showed that it relatively recently increased in frequency, while from modern day studies, we thought it was a very deep, like 7,000 years ago, maybe it started to increase, but with ancient DNA, they actually showed it's only around 2,000 years ago that we see a very strong increase in frequency of lactase persistence alleles in Europe. And there was many other studies with regards to disease-associated alleles that show frequency changes through time using ancient DNA data sets for Europe. Unfortunately, at the moment, the sample sizes are still a bit small for Africa, but it's increasing quickly. More and more studies are becoming available. And I think quite soon it will be possible for us to start with those kind of studies to look at frequency trajectories through time and how it changes. And obviously that will be very valuable to see both the selection strength and also the pressures that drive the increase of allele frequencies or decrease. I hope that answers the second question. And then the third question, if you can just remind me.
Yes, your thoughts on ghost archaic and modern archaic Africa.
Yes. Unfortunately, I actually have a few slides on that, but I didn't have time to include it in the presentation. But that's a very interesting question, this question whether there's ghost archaic admixture in Africa as well. Because we saw for non-African that we were able to intermix with Neanderthals and Denisovans, even though they separated from us around maybe 700,000 years ago. So the question is where we see fossils of archaic humans persisting in Africa together with our forms that are busy evolving into modern humans, whether the groups mix with those more archaic looking forms. And we have examples for that. We have the very archaic Homo naledi in Southern Africa that live alongside humans that were becoming modern humans. They probably were evolutionarily very separated from us and maybe too archaic or too diverged from us to mix with us. But then we also have the broken heel skull that is possibly a Homo erectus that has recently been dated to around 200,000 years ago. That would also have lived alongside modern humans. So there we also have an example of an archaic form living alongside almost modern forms. And there would definitely have been a chance of admixture between these groups. Unfortunately, for these archaic forms living in Africa alongside our evolving lineage, we don't have DNA from. For Neanderthal and Denisova, we had DNA, so it's easy to just show that they mixed with non-Africans when they left Africa. Before the Neanderthal and Denisova genome, there were studies that said we did mix with Neanderthal and Denisova, and studies that said we didn't mix. It was a true to and fro, just based on modern day genetic variation. People couldn't really decide whether we mixed with Neanderthal and Denisova or not. There was indications, but it was very difficult to prove. It's only after the Neanderthal and Denisova genomes became available that we definitely could say that we mixed with them. And I think it's the same situation now in Africa. There's some indications that we mix with these archaic forms, but it's very difficult to say for certain because we don't have sequences from these archaic forms. So obviously that would be absolutely great if we are able to get DNA sequences from these archaic forms. Some of them are bone, they're not fossilized, so there might be a possibility, but so far people were not successful in generating ancient DNA sequences from these archaic forms. And I think there was another question.
I think it was about balancing selection. But I think
yes,
in regard to selection.
Yes. So again, the models sort of the simulation software is there, and they are good simulation software, but they're very computational intensive. So usually you have to simulate relatively simple scenarios. So very complex scenarios, especially complex African demography together with selection would be very complicated, possible to simulate, but complicated and computationally very intensive.
So Karina, I have 4 questions. So I'm just kidding. I'm just joking. So I do have one question though. So in your last slide, you mentioned you showed almost 6 models, and that we can probably discount 1 model of a single origin. So what's next? What do we need to be able to discriminate those other models, or is that even the question? Do we need more data? Do we need better models? Do we need a better way to analyze the data that we have? More archaic genomes?
I think all of that, actually, all of those above would help. When I did the presentation, there was a paper that recently came out on bioRxiv that was not yet available. It's now out. It's work done by Brenna Hen together with a group in Canada. So they simulate various models, demographic models, and they tested differences between them. And what they propose, the most likely model is what they call a weak stem, which is basically like that river network that I showed, that there are populations merging and splitting through time. So what they think is that weak stem model explains our origin the best. But again, archaic admixture could also be a possibility just because of what we see in the fossil record. And I recently talked to one of the authors, and he said that they really want to incorporate it. But it's very difficult if you don't have these archaic genomes to test this hypothesis of archaic admixture and to distinguish it from these weak stem models where you have populations splitting and merging in time. Because actually archaic human is just earlier diverging models. So deep structure and archaic admixture, it's very difficult to distinguish. They're actually the same, they're just gradients of the same thing. But that would be an interesting paper also to look out for. I think the first author is Ragsdale. Erin Ragsdale is the first author, and it's on bioRxiv now at the moment.
I think we have a question here. Well, also, you just while the microphone's moving, I just thought I'd sneak in another question. So you mentioned mutation rates, and I think you're probably using a mutation rate of around 1 × 10⁸ now, which I think is going right. But there is discussion about how mutation rate may change or evolve over time. Thoughts on how that might impact our inference?
Yes, obviously it is going to impact our inferences. We use the best mutation rate that we think is the best at the moment, and it's an average mutation rate. And of course, we have different mutation rates for different base pairs and different parts of the genome. So that will have influence. There's a researcher doing research on mutation rates and also heterogeneity of mutation rates between the different primate lineages called Priya Muriani. And she's publishing a lot on that. So interesting work. Apparently it does vary quite a lot between different time periods and different populations or lineages in the tree, different primate lineages. But we have to use the best we can at the moment. Obviously that will be refined in the future. Maybe the holy grail would be to have like a site-specific mutation rate and a mutation rate that change through time, maybe based on what we think generation times were, because if generation times are, if people have children at older ages, also the mutation rate is higher. So it's connected also, generation time and mutation rate. So it is a difficult question, but I think we just do the best that we can at the moment.
That's right. Thanks. I think we have time for one more question, and then that'll be it. So go ahead, sir.
Oh, thank you. My name is Bigba from Ghana. Throughout this consortium, we've mentioned African genome quite a number of times. I wanted to ask this question to Professor Rotimi yesterday. I couldn't get the opportunity, so I'm bringing it to you today. So the problem has been population descriptors as to how, I mean, if we want to narrow it, we bring it from the regional level to even a basic level. Population descriptors is always a problem to define who is part of Africa and who is not part of Africa. Considering the fact that we are now living in a cosmopolitan environment, we have integrated over time. So I don't know, in your work, I mean, which were some of the population descriptors you considered in saying that this is an African, and which genome that you are working on? Yeah, I'm interested in this. It's also something that I'm following. Thank you.
Yeah. Yes, very good question. I mean, it is difficult to draw boundaries around things. We know there's been a lot of back migration into Africa also after the out of Africa. So there's not really a border that we set like 60,000 years ago. Some humans left Africa and they are non-Africans, and all other people are Africans, because some of these non-Africans have been back migrating into Africa and mixing into African populations. But we have some population divergences between the different populations, as we see when we look at their genetic variation. And we can see that there were some barriers between gene flow in populations in the past. So today we still have that population structure in Africa, where we can see that certain populations come from certain areas in Africa. And as you say, we are homogenizing more and more these days, which is a good thing, but it will make work on population history more difficult in the future if people start to mix more and more. So I think it's crucial that we describe genetic variation as we see today, that we still have this power to use differences between populations and population structure to help us to infer our population histories on the African continent.
Thank you, Karina, and thank you for joining us this morning. That was a great talk and inspirational.
Thank you.
Okay, so we are going to keep moving through the agenda, and today we are going to be listening to presentations around genetics and epigenetics of intergenerational transmission and impact on vulnerability and disease. Our first presentation is from Natasha Cohn, and I believe that's a video. And all questions will be left to the end of the session.
Good morning. My name is Natasha Kun. I am a senior lecturer in the Department of Psychiatry and Mental Health at the University of Cape Town in South Africa. Today, I will be providing a brief overview of transcriptomic studies in the Drakenstein Child Health Study. This presentation will incorporate two ongoing H3Africa projects, namely transgenerational effects of maternal stresses, investigating the role of infant gene expression, and secondly, the nasopharyngeal microbiome and respiratory disease in African children. Across these projects, we have measured gene expression in umbilical cord blood samples and have more recently also generated RNA sequencing data in the context of the Drakenstein Child Health Study. Broadly, the aims of this work have been threefold. First, to identify genes or pathways which may mediate transgenerational effects of maternal stresses through child gene expression profiles, operationally RNA sequencing in umbilical cord blood samples in the Drakenstein Child Health Study. Secondly, to elucidate the contribution of microRNAs via genome-wide microRNA expression profiles in the umbilical cord blood samples. And thirdly, to identify pathways which may be associated with common infectious diseases in childhood. This work has been nested within the Drakenstein Child Health Study, a multi-year, multidisciplinary birth cohort study following approximately 1,000 maternal child dyads in the Drakenstein region of the Western Cape in South Africa. The PI of the larger Drakenstein study is Professor Heather Zaar, and the PI of the psychosocial component of that study is Professor Dan Stein, both of whom are also MPIs on the H3Africa projects presented today. Within the Drakenstein study, pregnant mothers are recruited during their second trimester and then followed longitudinally through the birth of the index children and then at regular postnatal intervals during early childhood. Broadly, the Drakenstein study aims to investigate biological, psychological, and social risk and resilience factors for child health and development over time. To this end, comprehensive phenotype data as well as biological samples have been collected in the cohort, including maternal and child blood sampling for downstream omics analyses. Now, as part of the early omics work undertaken in the Drakenstein cohort, our group was particularly interested in exploring potential associations between a number of maternal psychosocial phenotypes and newborn gene expression. In a subset year of 150 dyads, we applied transcriptome-wide screens to umbilical cord blood samples from neonates of mothers with post-traumatic stress disorder or PTSD, depression, PTSD with comorbid depression, trauma-exposed controls, and unexposed control dyads. These analyses reveal distinct gene expression signatures which distinguish neonates born to mothers with a history of psychopathology versus those without. Specifically, and as demonstrated here, co-expression network analyses identified 2 depression-related modules which had previously been implicated in axon guidance and in mRNA stability, as well as 2 PTSD-associated modules previously indicated in TNF signaling and in cellular stress response. Interestingly, and also of translational relevance, these transcriptomic perturbations were actually also found to precede adverse neurodevelopmental outcomes in the same children when assessed at age 24 months, as demonstrated here in the cognitive and in the socio-emotional domains of the Bailey scales of infant and toddler development. More recently, we have also been interested in assessing potential associations between cord blood gene expression and TB infection in early childhood in the Drakenstein cohort. For these analyses, which are not yet published, the outcome of interest was tuberculin skin test or TST conversion at 36 months of age. As demonstrated here in this box plot, a principal component analysis, which consisted of those genes over an exploratory significance threshold, demonstrated statistically significant separation of infants with TST conversion from those who did not convert before age 36 months. We then repeated this analysis within the early TST conversion study population. This time, though, we were interested in exploring whether gene expression signatures at birth associated with the diagnosis of TB disease by 5 years of age within children who had acquired TB infection. Again, and as shown here, we found that gene expression at birth associated with the development of TB disease in early childhood. Building on this foundational transcriptomic work in the Drakenstein cohort, we've recently also undertaken RNA sequencing of umbilical cord blood samples, supported in part by our H3Africa research grants. Now, using RNA sequencing data generated at PE100 and yielding approximately 70 million reads per sample, we have run preliminary transcriptome-wide differential analyses. Separate models have been run for each phenotype of interest, including maternal PTSD, depression, and trauma exposure, with all models adjusted for relevant covariates or potential confounders. While no genes retained statistical significance after FDR correction in these preliminary models, the association between maternal PTSD and differential DISO1 expression in the newborns was found to tend towards significance. Given the multiple important functions of DISO1, including generating small RNAs, DNA damage response, and chromatin remodeling, this preliminary finding would certainly warrant further exploration in our context. Now, in addition to the RNA sequencing work being undertaken in our projects, we have also initiated and supported a number of community engagement activities in the Drakenstein cohort. These have included creating and staffing a school library at a local school, providing health promotion packages to our study participants, and implementing a number of COVID-related strategies or activities, including contributing to food parcel vouchers, mask provision, and health literacy and books being provided to children in the cohort. Secondly, we have also undertaken ongoing training, mentorship, and capacity building. For example, a number of doctoral students and postdoctoral fellows are currently and will in the future be able to leverage the data from these projects in their own projects. And a range of opportunities for early career capacity building and development have also emerged from this work. In the context of the larger H3Africa Consortium, we have participated in the steering committee as well as in a number of working groups, including the relatively newly formed Mental Health Task Force, together with 2 other similarly themed projects in the consortium. Looking forward then, we plan to undertake a number of additional transcriptomic analyses, including differential microRNA expression analysis, integrative microRNA and mRNA analyses, as well as potentially meta-analyzing the new RNA sequencing data with our existing expression microarrays. We also plan to undertake gene set enrichment analyses, as well as weighted co-expression network modeling, and we look forward to longitudinal analyses of RNA expression patterns and associations with infectious diseases, lung health, and other phenotypes of interest. In terms of the sustainability of this work, we have been fortunate to secure a number of new grants to support additional omics analyses in the Drakenstein study, some of which I've listed here on the slide. And we will, of course, continue to submit applications to further this work. We also look forward to new and continued collaborative research across disciplines, institutions, countries, and continents. And we will also remain committed to ongoing training, capacity building, and mentorship in this field. In closing then, I would like to say a word of thanks and to acknowledge our vast research team spanning UCT, Harvard McLean, Emory Yerkes, and many, many more. I'd like to acknowledge the other MPIs on the projects discussed today, including Prof. Stan Stein, Kerry Ressa, Elisa Wingo, Heather Zar, and Mark Nickel. I also want to acknowledge the H3Africa Consortium for all the collaboration and support received to date. I want to thank our funders, NIH, the South African Medical Research Council, and the Bill and Melinda Gates Foundation. I want to acknowledge our numerous co-investigators and collaborators across Africa and the globe. And last, but certainly not least, I want to thank and acknowledge all staff and participants within the Drakenstein Child Health Study, without whom none of this work would be possible. Many thanks and goodbye.
Thank you. Our next presenter is here, Stefan Jansen, who will be presenting intergenerational epigenomics of trauma and PTSD in Rwanda. And I would ask that once you're finished your presentation, join us here at the front. We're going to have a roundtable discussion afterwards. Sit here in the penalty box. Yeah. Yes, please.
Hello, everyone. Happy to be here. And thanks for the organizers here in Nigeria. It's the first time for me to be here. And it's a great experience. So I want to give a brief overview of the progress we are making in our project, Intergenerational Effects of Trauma and PTSD, following mothers who were pregnant during the genocide in Rwanda and their offspring. So we have a team of MPIs, which is Leo Mutesa, Derek Wildman, Monica Udin, and we have some brilliant, okay, thank you. And some brilliant other researchers like Segun, who is here, Clarisse Musanabaganwa, who is the PhD on the grant, Annette Uwineza, and many others. We have a big team. So PTSD is a mental health condition that can develop when you are exposed to extreme violence or extreme events. And of course, as you all know, in Rwanda, the genocide happened in 1994, and in a period of 100 days, 1 million out of 7 million population were killed. And really one of the biggest disasters worldwide that happened at the end of the last century. So we still see both on society and on individual mental health level in society, the genocide and the consequences of genocide are still present. And we are studying the transmission now to the next generation, because we see that also in the next generation, youth are struggling with PTSD symptoms, even though they are born after the genocide. And so we try to understand what is happening. Now, one of the hypotheses that we follow is when people are exposed to extreme stress, the hypothalamus reacts by releasing corticotropin-releasing hormone. That is then detected by the anterior pituitary gland that releases on its turn the adrenocorticotropic hormone, and that is then picked up by the adrenal cortex, who releases cortisol. This then generates a feedback loop and changing the way that the hypothalamus functions, changing the way that the anterior pituitary gland functions. And if you keep the stress for a long time, this can develop into, or not only long time, also very extreme, this can develop into development of PTSD. Now, the hypotheses that we are having related to this, so this is the HPA axis, so the stress regulating system in our brain. One of the hypotheses that we have related to this is when the stress is developing and starting to develop PTSD, there are methylation changes happening on the genes that are coding for the HPA axis. Right, so that's our first hypothesis. And the second hypothesis is that these changes can also be seen in the children that are born from, in our case, from the mothers that went through the traumatic event, right? So, so the main hypothesis, let me put a hypothesis here, and I'll just explain the design of the study. So, so the way that we are studying this in our study is we have a group of mothers who were pregnant during the genocide, who were living in Rwanda and who were targeted by the genocide, the Tutsi people. And they were pregnant during the genocide, right? And then we have a control group of also Tutsi people who migrated long ago, in our case, mostly to Congo, but also just to neighboring countries, who were not in Rwanda during the genocide, but after the genocide they returned. But they were also pregnant in 1994 during the genocide. So the one is our case group, the one is our control group. And now we are interested to see if the mothers who were exposed, if they have higher levels of PTSD as a reaction, so psychopathology, if related to increased PTSD, they also have a change in biomarkers related to the systems I explained before. So having lower cortisol, higher glucocorticoid receptors and mineralocorticoid receptors. So that are the biomarkers that are linked into this causal chain. And whether there is also methylation changes that are happening in the models. So that's what we are looking in. And then we look if those changes can also be observed intergenerationally, right? So we have a number. So I'm quickly going to talk through 3 studies that we did. This is the first one published on the existing. This was the first pilot study we did, happened in 2000. The data collection was in 2012. We biobanked those samples and then we reanalyzed them again. We had a bit of loss in the data. We had 70 samples originally. And I think after quality control, we remained with 41. In our original study, we looked at 2 genes specifically to see if there was methylation change in 2 genes of interest. In our new study, we did an EWA study. So looking genome-wide. And so what we saw, this is an example of one of the genes. These are the CPG sites. This is exposed to genocide, not exposed to genocide, and the children and the mothers. Right, so here are the children, here are the mothers, not exposed and exposed. So what we see broadly is a confirmation of what we saw in our pilot study. So we see that there is the mothers who are in the exposed group have certain genes, have higher psychopathology, and have, in this study, have higher levels of, this is comparing methylation status, right? So I confuse myself. So the methylation status in the exposed mothers is higher compared to the mothers in the control group, and we also see that effect in the children. Now, that's the first study. And of course, we look at all of our subgroups to test our hypothesis. Now, and I know this is the moment you've all been waiting for. This is results from the new data that we collected through the H3Africa project. And here we want to look at the psychopathology. So we build a structural equation model to analyze the data. So we want to know whether the mother's exposure, that's our main causality is mother's exposure, to what extent is it predictive for the development of the child PTSD, right? Our study is actually a bit more complicated, as I said before. We have the child that was born from the pregnancy during the genocide, but then we also look at the younger sibling of that child to see if that transmission during the genocide was extreme stress and the mother was living under extreme stress, which can explain the transmission. But our question is, does that stress also remain, and does the transmission also happen after the mother has developed PTSD, but is not anymore in an acute stress situation? So our first interest is, to what extent is the mother's exposure predictive of the child PTSD, and is this relation mediated through the mother's PTSD, right? So the mother's exposure predicts mother's PTSD, and the mother's PTSD predicts the child PTSD. And then we are interested between when the mother's exposure predicts mother PTSD, whether this is moderated by the mother's resilience, right? So we expect if the mother is more resilient to see less PTSD. And then we wanted to see if the mother's PTSD, to what extent is it predictive of the child's PTSD, and this is moderated by being the first child or being the second child. So The results are here completely in line, not completely in line what we expected. For the first child, you see a very strong effect of the mother's exposure, predicting much higher child PTSD. That is for the first child. For the second child, there is no effect. We didn't have a strong hypothesis on that going into the study, but we actually did expect to see an effect. So it's a bit surprising to see this. The second one is completely in line with our hypothesis. So you've got the mother's exposures and the mother's PTSD. You see, as the mother has more resilience, the PTSD reduces. So if the resilience of the mother is higher, she develops less PTSD. And the third relationship is a bit complicated. We see that the mother PTSD predicts the child PTSD, but only in the second child. So that's something that we will need to look deeper into to understand. We would have expected to see that effect in the first child, also in the second child, but certainly in the first child. So we will analyze some more to understand this more deeply. Okay, then our third study is to look at the biomarkers associated with PTSD in genocide survivors. So now we are looking at cortisol and GR and MR. And I'm just going to give you the overview of the whole exposed group, meaning the mothers and then the children born from the exposed mothers compared to the non-exposed mothers and then their children, right? So we of course, we did the analysis for all of the subgroups, but here you can see, as was expected, the cortisol levels, and confirming also our pilot study, the cortisol levels in the exposed group are significantly lower compared to the non-exposed group. The mineralocorticoid receptors are higher compared to the non-exposed compared to non-exposed. We expected to see the same in the GR, like the MR, but there is no significant effect. On top of that, we did, or during this event, we did community engagement. I think in previous presentations, we talked about it. It was very moving events. The mother were talking about, they were actually very motivated to join our events, as they said, there is something with my child and I know you are going to explain it, right? And still, because this study is quite fundamental research, so we are really looking to do follow-up research to actually help those mothers and help them and guide them with how they can help their children. Okay, so I went a bit fast. It's counterintuitive, and I have to point there and not here. Okay, so so then we are part of a number of the different H3Africa groups. So we are part of the harmonizing phenotype group, and especially Susan Rudahindra, who was at the university in USF, in Florida, with our partners in the States. Did a lot of work on this together with Natasha and the group from Dunstein, with others. Derek Wildman is active in the publication working group. It was mentioned also yesterday, Leon and the rest of the team, also Clarice, Aneto Ineza, all of them played absolutely key positions. We only have very few geneticians in Rwanda. And so they were really leading the effort of setting up a system to trace and get a lab ready with COVID. And so it shows really the importance, right, of the way that people are. Clarice is trained through this grant. And so you see the real effects that it has had on the country. Now, Segun, since yesterday, Prof. Otimi, he confirmed officially that he's one of the most brilliant African young researchers. We always knew before, but now, Segun, it's official. And so our next step is we want to finalize data collection. We were quite disturbed. Rwanda was very strict in its measures, so we couldn't continue with our data collection. This is planned. We are ready actually for in the coming weeks. We want to do the full EWAS, and if we find enough money, we want to do the full GWAS. At the moment, it will be partial GWAS. We also want to do a study on memory functioning. As we know, PTSD is also affecting the memory functioning. That's something that is clinically very much observed in Rwanda. And so that's another study we are doing. And we analyze the data using structural equation modeling. So it's the same model as before, but integrating the biomarkers. So that's it for me. Thanks so much.
Thank you, Stefan. If you could join us at the for the panel discussion afterwards. Our next presenter is Alashley, and she'll be presenting breast milk microbiota, influence on infant immunity and growth. Beaming.
Oh, thank you. Good morning, everyone. Good. We have the slides up. It's a better day. Oh, you're not gonna do that to me as soon as I said it. So I'll be sharing with you one of the projects that we have been funded to do, and is on breast milk microbiota, to see how it has any influence on infant immunity and growth. And this work has been done with using a birth cohort, a HIV birth cohort, with collaborators at the University of Cape Town in South Africa. Just to say that this particular research was nested in a birth cohort that we had established in Nigeria and in South Africa much earlier on, to just look at the effects of HIV infection in the mothers on infants. And we were particularly interested in looking at infants that were exposed to HIV. We knew a lot about the fact that when the infants are infected through mother-to-child transmission, there are some peculiarities that these infants have when they're infected, right? In terms of thriving and really mounting the appropriate pediatric response to pediatric vaccinations. Now, what we didn't know then is what happens to the infants as a result of the very highly active retroviral therapy that has been available to a number of the HIV infected mothers. What happens to those infants that's as a result of the moms being on ART and the viral load being so low, they get exposed to the low levels of viruses in mothers, but they come out uninfected. So they're not infected by HIV, but they have been exposed because mom is HIV infected, right? Do they have the kinds of limitations or the kinds of health challenges that HIV infected infants do? So that was the major question we were asking in this cohort. Now, we have already published that there is, in fact, a limitation in terms of the growth and in those infants that are exposed but not infected, that their growth is really limited. So when we got an opportunity to get the H3Africa funding, we wanted to go back and begin to look at mechanisms, ways that these infants might be different from normal infants who are coming from mothers that are not HIV infected. So the first study, the cohort, what the cohort did was give us an opportunity to enroll mothers. Is this pointer working?
Yeah. Ah, very faint. I can't even see it. Anyway, so it gave us an opportunity to enroll mothers when they're pregnant and then follow the infants from birth. So when they're 18 months old. And during that time, we had the opportunity to, because we had a biorepository, then a rudimentary one that has been built up through H3Africa to be a regional one. But we had the opportunity to really store a number of samples that now we were able to go back and interrogate these samples to address the goal and the specific aims that we had for the research activity and this funding. So what was the goal of the research project? It was to determine the influence of breast microbiota in the microbiome, in the gut microbiome, and to see whether that, certainly in a number of specific aims in the first one, we were trying to compare the ontogeny of the microbial structure and the metabolome in longitudinal breast milk. And does this influence the microbiota that we see in infants? Because earlier studies have actually shown that some of the growth limitation is also predicted by the type of microbiome that we find in the infant. So a number of these infants, as you know, growing up in sub-Saharan Africa, mostly mothers are encouraged to exclusively feed these infants. So we wanted to know what is the influence of the microbiota in the breast milk in determining the microbiome in these infants. And this study is led by Jonathan Blackburn and Heather Jasper. So for the microbiome, okay, I'm skipping. So for the second specific aim, what we try to assess here is to understand if the microbiome shifts that we see in specific aim, would that in turn explain what we see when we look at the immune responses in this exposed uninfected infants. So the ends are all linked together to try and kind of unravel this very complex relationship that's going on between the microbiota and the immune responses. And the last specific aim is really to look at the inherited HLA gene variants from moms in their infants and see what role do these inherited genes play in terms of the immune response that we're seeing in these infants. Because obviously, especially for the humoral immune response, you expect that the inherited HLA gene for the type 2 kind of immune response would influence that. So what I'm going to share with you in the next few minutes that I have is the progress that we've had so far. Maybe one of the areas that we haven't done a lot, one of the areas that we've done, we have enough data on is the immune response, because we had serum samples that were readily available and we started analyzing that very early on. But for our specific 1 and 3 that looks at the microbiota and the HLA, there was a lot of extractions that had to be done and optimization and shipping of samples between the two countries that was really truncated by COVID. So there were preliminary data, but the most mature is that of the immune response that we saw. So what I would like to also point out is that for all of the specific aims that we've worked with, we really have benefited a lot from the H3Africa fellows that have been associated with each of the investigators. So for M1, we have Anna Happel, Tarif, and Alex that worked with both Heather and Jonathan on that. For specific M2, Sophia did a lot of the work as a fellow, but I also have Flores and Evase looking at different aspects. Actually, Flores is more interested in the immune response to hepatitis 2. Sophia looked at tetanus and to see whether that's different, and Evase is interested in the response to BCG. And then for the third one, working with Clive, is Mikhail. So we're very grateful for the fellows that did this great work. And, so I want to recognize them now before I end. And just to point out that two of our fellows, Sophia and Florence, actually will be graduating this year on their PhD with work that they've done on this work, and all four of them have manuscripts almost ready for submission. So what are our preliminary results? So this is really briefly summarizing a lot of data that we have gotten, and that we found in the infants, when we compare the exposed infants and the normal controls, that really we do not see much difference in the, we do not see much difference in terms of the microbiome. So it was a microbiome that we were looking first and foremost, and that the gut microbiome tends to change with time. So I really don't have a pointer here, but what you see at the top here, the HIV positive moms in yellow, and then our controls in pink. And if you look at the pattern of the microbiome that you see really across the spectrum is very similar between both of them. And the only difference that we saw was that with time, this pattern tends to change. And then what else is here? And just to point out that, interestingly, the diversity we see in the South African population is much more complex than what we see in the infants. And this is in the infants in the Nigerian population. And this is really interesting because we see the reverse when we look at the immune responses. As I mentioned, the work on the metabolomics is really just been put together. We were really looking at the breast milk, we're extracting the RNA from there, and we're looking at the different profile of the metabolomes that comes out of that. So hopefully we will have that work done in the next year or so and have it published. But if we look at the immune responses that we're seeing, and again, in the interest of time, what I'm presenting here is really the summary. So this represents a very complex kind of interaction, but I do have a summary slide. So if this looks converse, it will get clearer. So on the left of you is the Nigerian cohort, and on the right is the South African cohort. And what you see off the bat is that when you control the HU are the infants from normals, and the HEU are the infants from the exposed infants. So what we're looking at is those that have the baseline immune response, and then the immune response following the 3 doses of tetanus that the infants have. And what you see off the bat here is that these infants have a really significant response to the tetanus at birth. And following vaccination, you really don't see that much of a difference. And this is the control. and the p-value shows here. But when you come to the South African cohort, although it's much lower in terms of the immune response you see, we see the same pattern, that there isn't much that happens following vaccination. However, in the exposed infants, we see a slight increase, but it's not significant. In the Nigerian cohort, on the other hand, the vaccination of exposed infants really seem to be detrimental in terms of their immune responses. We really don't understand this. At baseline, the immune response is really significant if you look at the median or the mean. And then after the 3 doses of vaccination, these infants even have a lower immune response. So that was really strange. And what we tried to do was go back to the mothers and see whether we can get any insight into what is happening. And this slide again summarizes it. And we're wondering whether what we're really seeing is the reflection of the immune responses in mothers, in these infants, and whether, you know, vaccination in the exposed doesn't seem to make that much of a difference. So mothers, uninfected mothers, have a really good high immune response. HIV infected mothers have a reasonably good immune response because they get vaccinated. When you look at the infants at baseline and subsequently later, the immune response all come down following vaccination, and even in the control infants. So we really are trying to look at this to understand what is going on. Maybe these high levels of immune response from moms is really blotting the ability of these infants to make the appropriate. Yeah, okay, so I need to finish up here. So this is a summary of really this complex work that we've done in terms of this. We find out that the mom's antibodies are definitely passively transferred to the infants. Significantly more are seen in uninfected than HIV infected moms. So somehow the HIV infection reduces the ability of moms to do that. And moms with higher antibodies is reflected in infants. And we're wondering what the vaccinations mean in these infants, whether we're just seeing moms' antibodies just being reflected over time and weaning off, or what exactly is the vaccination doing. So anyway, we're still following this, and the differences is really significant, but it's not our expectation. We expected the vaccines to work, but maybe not as well and the exposed as the uninfected, but what we see is the reduction in immune response. So we're following that. And this really is my last slide, I believe, is our HLA typing that we're doing at the moment. And one of the things that we want to look at is add up this immune HLA types and compare them with the kind of immune responses that we're seeing. And maybe we will begin to dissect rather than look at them as a whole group. So there are clearly some alleles that we see that pop up that really are good for humoral immune responses. And the other thing that we're seeing is also there are very significant differences between the allele cells in Nigeria as compared to our South African kids. So we're trying to put all of the 3 specific aims together to make some sense of it. So anyway, these are the challenges and some of the stuff that we experience, and just to acknowledge, like I say, our fellows, my co-investigators, the institutions and the funders that have allowed us to do the work. Thank you. This is for you.
Great. So save your questions for Ashley for the panel session. And our next presenter is Arissa Sabakaki. Ethical and Social Issues in Informed Consent Processes in African Genomic Research.
Thanks. Good morning, ladies and gentlemen. It's been hardcore science this morning. Let's just take a commercial break, dive into social sciences. That will cool the brain. Thanks very much. Who's helping me? Do I have a... Oh, thank you. So basically, this is, we're looking at the ethical and social issues in informed consent processes in African genomic research, and that's in Uganda. We had 6 objectives. Originally, there were 4. Then we were awarded a supplement, and we added 1. So basically, we assess the quality of informed consent documents to determine if they comply to our national guidelines. Then we also explored the knowledge, perceptions, and experiences of researchers on ethical and social issues involving biobanking. Then we also talked to research ethics committee members and regulators. Then the children, because the caregivers, that's for pediatric research, and research participants. For the supplement, we looked at the return of individual results in research involving minors, and to be specific, that is pediatric research. And we looked at the CAFGEN study, which has been running for the last 8 years or so. So their children, I mean their participants who were recruited as, as young as 5, 4, but right now they're adolescents. Some of them actually are adults. So we talk to them. So just a minute. Which direction is that? So it's frozen, eh? Okay, thank you. So we reviewed we reviewed 243 informed consent forms for biobanking and genomic research. Those are 243 protocols, and of those, 186 indicated that they would export samples across borders. Now, of the 186, only 77 had material transfer agreements on file. In Uganda, it's mandatory to have an MTA before you export. Then of the 186, only 56% had 30% had an export permit, and it's also mandatory to have an export permit before exporting. Now, the type of consent, the majority actually solicited for blanket consent. Only 11. Remember, in Uganda, broad consent is the one that is okay. Our national guidelines recommend broad consent, but the majority did not solicit broad consent. Now, overall, most consent forms were inadequate. We are talking about consent forms for sample storage for future use, and only 14% had a statement on benefit sharing, and only 51% had a statement on the risks. So we realized that actually most researchers and research teams do not know the risks that pertain to biobanking and genetics or genomic research. Then also the policies that govern the use of samples in future, the requirement for research ethics committee approval, that one was not also. The MTAs, we have guidelines that have the different components of a material transfer agreement, but actually most of the material transfer agreements did not did not conform to the national guidelines. And actually, as a result of this, our national regulatory authority, Uganda National Council for Science and Technology, is developing a template for material transfer agreements. And initially, it wasn't the role of the Research Ethics Committee in reviewing and approving MTAs was not mandatory. It was hazy, but at least as a result of our results, right now it's mandatory for data sharing agreements, material transfer agreements, and in some cases collaborative agreements to be reviewed by the Research Ethics Committee. Now, this is a summary. So basically, we had we had a convergent parallel. We had the convergent parallel mixed method design. So it's a lot of data, but I've just decided to to summarize it. So overall, there was poor understanding on the types of consent applicable to research, to biobank research. As a matter of fact, most researchers and their research teams did not know what broad consent was. And they didn't even know the different types of consent. And actually, as a result of this, we conduct regular trainings for research ethics committees and researchers. So right now, one of the things we're doing is trying to ensure that, we teach them the types of consent, broad consent, tiered consent, specific blanket consent, etc. Then many principal investigators are not conversant with informed consent processes in their studies. Because initially we wanted to look at, to talk to researchers to find out what they do. But after the first 5 interviews, we realized that actually principal investigators do not know what goes on. Everything is delegated to the study coordinators and the nurses. So we had to amend the protocol and included the nurses. So we had three focus group discussions with the nurses who actually actively participate in obtaining informed consent. But what we found that actually they have inadequate basic knowledge on genetics and genomics. Now, you expect a participant to understand, but the teacher who's supposed to teach doesn't also understand what they're teaching. Now, at the end of the day, you're going to have a participant who has not understood, not because they do not have the capacity to understand, but because the one obtaining the consent is also not well versed with the topic at hand. And there were also major challenges in translation of consent documents into local languages. Remember, in Africa, many of these words are new, they are novel. So for you to drive a point home, you need to tell a story because you do not have direct translations. And the problem with telling stories, you end up distorting the meaning. And this was a big problem. There was limited understanding of consent by research participants. Of course, that one was expected because even the people obtaining consent were not well versed with the studies. And in Uganda, use of checklists use of checklists in assessing comprehension is not mandatory. So you find that actually many of these studies do not assess, for understanding. And of course, this is a major weakness for the research regulators. And this was also this one we believe also contributed to the poor comprehension. And most researchers indicated that community engagement was very, very key in ensuring that the participants and the general population and communities understand what genetics and genomic research is all about. What were the challenges? Poor quality and inaccuracy of translations, inadequate understanding of consent, illiteracy, then using multiple informed consent forms. In Uganda, if a study involves genetics or genomics, you should, it's recommended that we use 3 consent forms. 1, consent form for participation, a separate consent form for sample storage, and then a separate consent form for genetics or genomic testing. And now, by the time a participant is through all those 3, they've lost concentration, and at times they just want to get through with it so that they go back home. And that, at least that we found, was a problem. Then some of these consent forms are voluminous, so much information, and information that has to be taken in and assimilated in a very short time. Then the language. Then we also had a problem. Some research ethics committee members and researchers had limited understanding of genetics. Now, there was also a problem of trust. Participants, researchers have some mistrust with collaborators. And of course, this might come from, the historical unidirectional flow of samples, then misinformation in the general public, as people selling samples for cash and etc. What are some of the recommendations to enhance comprehension? Community engagement to sensitize the general public and educate them on genetics research and its implication. Iterative approach, informed consent, where participants are given ample time to read. Consent information, ask questions. Permission, taking the consent forms home to, all these things are being, in literature they are there, but you find that at times, locally, some of these haven't been, okay? Then encouraging the use of simple languages and various media during informed consent disclosure, use of videos, role plays. In Botswana, they developed a comic book, some of them are being used at CAFG in Uganda. The need for harmonization of translations. So usually we translate consent forms into 4 or 5 languages, depending on where they study. But you find that since we do not have any uniform words for these translations, you can read 3 different, 3 people can translate one consent form, and you find that actually they differ. There is a lot of difference. So we need to harmonize and maybe come up with a dictionary of translated key. Okay. Then introduction of teaching of genetics at ordinary level education, then develop specific guidelines. And these ones, actually, we're developing them. Then, of course, we also have recommendation for the Ministry of Health, then training of research ethics committees. And we also, okay. So perspectives on biobanking. Overall, there was a positive attitude, but then importantly, respect for the culture, culture values and local beliefs. Then, of course, there is also a power differentials. So our project has supported 4 master's students. So this is 2 of them have graduated and 2 of them. So this particular student is already on a PhD program. And basically, she's still looking at consent in pharmacogenomic research. Then this one graduated recently. Basically, his study was on understanding of critical elements. These have all been published already. Their publications. Then we have, oh, sorry. Yep. Okay.
have one question right here. That microphone is.
Thank you. I have just two questions. Maybe it's not enough. Two questions for Stefan Janssen. Thank you very much for your presentation. First question, how did you measure mothers' resilience? Was it through questionnaires? And second, if you could please re-explain a little bit the confusing story about the first child. As I understood, you had more, you had association with mother PTSD, but no association with child PTSD for the first child. So you can please explain that. Thank you.
Stefan, are you? Yes, go ahead. Yeah.
Okay, thanks for the questions. So for resilience, we use the risk, so standardized questionnaires, and as we did also to measure PTSD. So it's all quantitative data collection. For the first child and the second child, I was taking notes when you ended your question. Is the question on how the design is made, what we are expecting, or what the results that came out?
When it's the first pregnancy, but you showed after that a slide where you show an association with the PTSD for the second child, but not for the first child.
So we are very sorry that we couldn't be there with you all in Nigeria, but are very excited to present on work that we've done in the last roughly 5 years.
Okay. My turn, or?
Yeah, go ahead, Stefan.
Okay. So the data we've seen is the one is about the mother exposure, and that is predicting PTSD in the child. That is one relation we are looking at, and the other one is whether the exposure from the mother is, or whether PTSD from the mother is predicting the PTSD from the child, right? And so in our results, we saw that going from the mother PTSD, what is predicted is that the second child, so if the mother has higher PTSD, for the second child, you also get higher PTSD in the children. So there it's correlated. But for the first child, the effect is not there, and we did expect it.
Great. Thank you, Stefan. I think what we're going to do now is pause questions, because in fairness, we have one last presentation to get through. So if we could have that video started. Thank you.
Be there with you all in Nigeria. So our project largely focuses on antibodies. Antibodies protect us against many different infections. They are, of course, unique and are antigen specific. So understanding the immunogenetics of these antibodies and how they arise is of crucial importance to vaccine design to make more effective interventions. The antibody molecule is a Y-shaped molecule that consists of two regions, the first of which is called the Fab and consists of the variable portion as well as a bit of the constant region. And this region mediates the ability of the antibody to block infection or neutralize a particular pathogen, does so through binding to the antigen. And the other portion is called the FC portion. This mediates a wide range of cytotoxic functions, many of which are indicated here in these little pictures. So they include cytotoxic popping or lysis of cells, trogocytosis or the nibbling of cellular membrane, and then phagocytosis or the engulfment of viral particles or infected cells. This region performs its function through binding to FC receptors and really allows for the recruitment of both innate and adaptive immune responses. So overall, our projects have really been focused on understanding antibody genetic diversity and then understanding the impact of that variation on antibody function. And this is really in the backdrop of South Africa being having the highest prevalence of HIV infection. And we really want to improve both, passive immunization and vaccine strategies. So I'm really going to give you a highlights package of things that we've achieved over the last 5 years. One very impressive one has been to look at variation in the variable region of the antibody that's indicated there in red on the little schematic at the top left-hand corner over here. And what we were able to do through work done by Elaine Marsden, and she's a PhD student that's mentored by Kathy Schippers, we were able to show that around 21% of these alleles that we were finding in this region are actually novel and previously unreported, and then about 80% of them are reported, and we see those across our population. So this is an unprecedented kind of level of diversity. Something else that Elaine's been crucial in is also reporting undescribed variation in regulatory regions of antibodies. So this is these are regions that are typically not looked at when sequenced, and 44.8% of diversity was actually seen in these sequences. So a large amount of this diversity was actually in the leader sequence. And that not only has implications for how antibodies are secreted, but also how antibodies are actually named. So both Kathy and Elaine have been very instrumental in actually altering how we go about naming these alleles. We've also brought in new technologies through our collaboration with Dr. Corey Watson at the University of Louisville, where Elaine previously spent some time. He has a technology where he captures the entire immunoglobulin heavy chain locus. So in chromosome 14, they're then able to kind of separate those out into genes and find that at least in 2 individuals where Elaine looked of these HIV infected individuals, they express genes in different combinations. And so this also has an impact on how variable the immune response may be. And so we're working quite hard to understand all of this new exciting data. Something else we're very proud of is the launch of NICD's iReceptor repository. It is an annotated international database for immune repertoires. And we also do, of course, submit data to other databases. But this is really exciting to us because it's very useful for future studies where the annotations are already put in. So go and check that out if you're interested. In addition to variation in the variable region, we have also reported now constant region genetic variation. So the constant region, as shown in this little schematic here, is highlighted here in black. And this essentially is different isotypes, which are important for different immune responses, usually innate immune responses. And this is work that was done by Ty and Holly, both master's students. We found 2 undescribed IgG1 alleles and 5 undescribed IgG3 alleles. Some of these changes are synonymous, so they don't have an effect on protein expression, as is the case for IgG1. But in IgG3, we see a large number actually of non-synonymous changes, and they have an effect on the changes the protein has. And then if we look at IgAs, we see similar things. So IgA1, we've seen 8 undescribed alleles, and then in IgA2, 5. And this was work done by Tai, and we can see that many of these are non-synonymous changes. And so we have very little idea about what these do functionally. And so this brings us to our next thing that we're very proud of, which is that this H3Africa as a whole actually helped to establish an FC effective function core across a number of different diseases. So we developed many of these assays. So this panel over here just indicates the slew of assays that we are able to actually run for HIV initially, but we've since run them in studies for influenza, for COVID-19, and for HIV-1. This core has 2 PhD students, 2 master's students, and a full-time technician. And we've generated 19 publications since 2018, which is the year after the grant was initiated. 11 where the FC core was actually first or last author. So we're very proud of this particular achievement. And then just to mention that this core has actually been crucial for the unfolding of the COVID-19 pandemic, and has made some very important findings. One of them being that FC effective functions are largely preserved against variants of concern in infection and vaccination. And that's, of course, in big contrast to what we see in neutralization, where the blocking of antibodies is less effective against different variants of concern. And so this has become a big focus of our laboratory as we move forward. In terms of the HIV work, this is work done by Holly Spencer. She found, as I mentioned, a number of IgG3 novel mutations, and we were actually able to show that one which is very close to the, that is directly involved in FC receptor binding, did also actually affect function. So this particular allele was more potent at performing phagocytosis as compared to IgG1. So these have very real functional implications and very real therapeutic implications. We've also since looked at broadly neutralizing antibodies for HIV. Those are antibodies that are able to target and prevent the infection of lots of different strains of HIV. And when we looked at different allelic variants of this particular antibody, we could improve neutralization compared to IgG1, which is typically what's used in clinical studies. We could improve vastly the phagocytic ability, and we actually had an interesting effect on the ability of antibodies to lyse cells. And so understanding this is now also one of our top priorities. So we have since also looked at a number of different broadly neutralizing antibodies instead of just the one, that target a whole range of different sites on the surface of the HIV trimer. And what we have seen is that when we make IgG3 versions as compared to IgG1, they show increased ADCP or phagocytosis, increased ADCC, and then increased neutralization. And very interestingly, if we test them in combinations, as IgG3 compared to IgG1 combinations, we can actually improve their neutralization as IgG3s. And so this has big implications for harnessing allelic variation for benefits in therapeutics. So in conclusion, we've reported unappreciated novel variation in both the variable and constant regions of antibodies. We've launched a notated immunogenetics repository. We found functional relevance of allelic variation in the constant region. We've shown direct implications of this on potency for clinically relevant antibodies for HIV. We've set up FC effective function core, which has made crucial discoveries specifically in the COVID-19 space. We, as a unit, have trained 2 PhDs, 3 masters, and 2 honors students in 5 years on this set of projects. And then ultimately, we've begun to understand how to harness genetic variation for real-world application to clinical intervention. And then just to thank the lab, of which this was a massive effort. The work was mostly completed by Holly Spencer, who got a distinction actually for her master's. Elaine, who is now doing
Hello. All right. Whether treatment could be confounding or impact. It reminded me of something that we're seeing with COVID, where people are being treated for melanoma or other malignancies are not responding to vaccination.
No, that's a good question, and we actually, fortunately for a lot of this mothers, they're on a very similar regimen, but we're concerned that we're seeing the same kind of lack of increase in immune response in even infants that are coming from uninfected mothers that are not on ARTs at all. But if you see the data that we have, there's really huge confidence interval. So what we're trying to do is really go in there and identify the clusters that are not responding and the clusters that are responding, and do a similar test in South Africa. But we see the same scenario in uninfected mothers who are not on ART.
Thank you. So a number of questions here from people in, go ahead. Yes.
My question goes to Elsa about ethics in Uganda. I'm not sure whether the study you presented today, whether you included the some study from the general population cohort at the MRC UVRI. And I would like to, I know that the process is that if you have to do if you have to do transfer some data or sample outside the country, you need to get the ethics from the UVRI and then go to UNCST. And in the process, after getting approval, is where you require an MTA. But it seems that your study, you want an MTA approved before you get the approval, which is not always the case in our case. So I'd like to know whether you have some data from the GPC for the study that you showed today. So my second question is about, not sure whether you mentioned this, but I would like to get your understanding about what you think constitute an appropriate best practice, or if there's some regulatory framework in Uganda for feedback for genomic study.
Can I answer the question directly? Okay, thanks very much. So with some of our participants were, parents and caregivers of children participating in genomic research. But specifically, this was at the College of Health Sciences, and that was at Baylor and some other research institution, but we did not go to UVRI. And about the MTA, ideally, the MTA should be reviewed and approved by a research ethics committee before proceeding to Uganda National Council for Science and Technology. But the problem was, it wasn't clear in the current guidelines. And when I presented this to the national regulator, actually, they sent out a memo to all research ethics committees requesting that from there on, moving forward, MTAs must be first approved by the research ethics committees before proceeding to Uganda National Council for Science and Technology, because they identified that was a big gap. Now, concerning the feedback of results, right now in Uganda we do not have any frameworks for it. Actually, most genomic studies are not returning these results. Maybe in my in my study, I think I came across only one person, and that was in a sickle cell study. So we do not have frameworks. And actually, one of the things, in our knowledge translation, we would like to come up with, of course, we'll benchmark, and we will benchmark on the guidance, the guidance documents that have been developed by the H3Africa Consortium. But of course, we have to, we have to adapt them to our local situation. Thanks very much.
Great, thank you. Are there any other? There's another question in the room at the back of the room.
Thank you. My question is for the last speaker. The study on HIV. Was it a multi-ethnic study or multiracial study? Or was there any influence of ethnicity on the results you got?
Yeah, who are you directing that question to?
Speaker.
Oh, I don't
know. The last presenter,
the
topic was on HIV in South Africa.
Yeah, I'm not sure we have that person live here, I'm afraid, sorry. I think we've got a couple of comments here by Dama Bukini. Are you in the room here? They're online? Okay, well, no, this one says in person, so.
Can I answer?
Go ahead, yes.
Yeah, no, a quick question. Your study is interesting, and Uganda is very vast, right? And some of the discussions we've had in terms of MTA, ethical approval, knowledge, and all of that, I would love to see, like in the individuals and the researchers that you interacted with. Are there mature researchers? Are there H3Africa researchers? Are there new ones? If they're H3Africa, because we've gone through these years of really trying to do community engagement, right, and everything, it would be nice to sort it out and just maybe with those that have had the experience and the benefit of of our ethics group within H3Africa, maybe they're able to do things a little bit better than those that are just starting or something like that. But just, it's a large country. You can't talk about all the investigators in Uganda, right? I mean, they're different.
To be direct and blunt, even H3Africa, some of the H3Africa principal investigators actually were not well versed with broad consent and some of the ethical issues. The problem is most of the researchers are just, this is my opinion. I'm more interested in obtaining research, okay? There are some of them who are interested in the ethical issues, but then others are not interested in the ethical issues. That's why right now it's mandatory for every researcher to undertake a research ethics training. That's a human participant protection every 2 years. And of course, this is a requirement, but it hasn't been enforced. But right now, because of the information we are getting from this, like this empirical information, our national regulator, is slowly changing the terrain in Uganda. So we hope everything is going to get better. Thank you.
Great, thank you. I'm going to ask the last question before we move on to our next speaker, and that's to Stefan. Your last slide showed that cortisol levels, you had 3 pictures. One was cortisol, I think glucocortisol was on there as well. But cortisol levels themselves were not different. Were not significantly different between groups. I was surprised by that. If I have that, am I remembering that correctly?
Okay, because the way I remember is they are different. So cortisol was different and was lower in the PTSD exposed group. The mineral,
it was lower. Sorry,
it's lower. Yeah, the mineral corticoids are higher, and then there was no effect for the glucocorticoid.
Okay, so I'm still surprised, but I would have thought cortisol levels would have been higher in the PTSD exposed group. That would have been my initial hypothesis going in.
Yeah. Maybe Segun can help me there. Also, Segun can't help me there.
Sorry, yeah.
Okay.
We can talk about this afterwards.
Okay,
Sorry.
yes, we can.
So we're going to go to our next speaker. Mamana is presenting, and I believe he will be presenting, showcasing the H3Africa HAA BioNet. And he is asked to do a online demo for those who are interested and want to stay during coffee for that. Is that right? So take it away.
Yeah, thanks, Philip. Hi, everyone. So I'm Mamana Mbiavanga. I am part of the H3 BioNet project based at the University of Cape Town in South Africa with Nikki. So I'll be taking you through a bunch of demos, and because this meeting is kind of special, so I took the gamble, the risk. Hopefully everything will be okay. So I'll just do a quick intro. Then the demo that we'll be doing will be, the first one will be on the African genomic variation medicine. The second one on HBD, also look at beacons. The third one will be on the H3Africa catalog. And then we'll look at the H3Africa array info site. And then we'll finish with the imputation service. So just to give you an idea of how these services are connected. So it's really trying to support the full life cycle of data. So starting from recruitment database to how we manage clinical data and store them, going to biospecimens, how they are served in the H3 Africa catalog, looking at the genomic data, how they are served in the version database, and how we also planning on using Beacon to share the data, and also the imputation service, the genomic medicine portal, and also the microbiome portal. So I'll start. So just to put the context in what we'll be doing. So let's consider a researcher who has a scientific question. And one of the questions, or maybe a common question that you ask is, I have a variant that I'm interested in. So a variant that is correlated with an outcome. It can be a disease or a phenotype in a given population. So some of the questions that you'll be asking as any study also looked at this outcome. Another question will be, has this variant been seen in any other population or studies? So if I'm doing a genome wide association study, I'd want to know if my variant is on any chip, and also maybe interested to know if any H3Africa studies have also looked at this phenotype, or maybe as some control samples that I can be interested. So the first tool that you can use to answer the first question, which is looking at a phenotype or an outcome, a tool that is developed by H3Africa is the African Genomic Medicine Portal, and I put the link there. So this is the landing page, and there you can, you can actually, before we go there, so what it is, it basically a platform that provide information about existing resources. So the data is taken from this GNet and the from KGB, and then created by researchers in H3Barnet. So what you can get from there, you can search for published SNPs or variants which been associated with diseases or even response to certain drugs, and you can filter that by genes, disease, or variants. So going there, you can do a search. So let's say if you're looking at malaria, you can just enter your search term, and you get some few suggestions. And then if you search, you get a list of studies that have looked at this phenotype. So in our case, let's say we're interested in cardiovascular diseases. So we can do that and search. Then we get one result. You can click and get more information. So you get the variants associated with this phenotype. You get the genes, gene or genes, and you also get the country where that study took place, and also available literature related to that. So you can click and go on and get more information about this. So let's see. So let's say I have this information and I'm interested to know more about this variant. Then that takes us to the second question. So I have this variant this variant been seen in any other population or study? So there we have two resources that can be used. The first one, the African Genome Variation Database, and also the beacons. So the African Genome Variation Database, it's a resource that serves, or that gives researchers the possibility of exploring genomic variation data within African populations. So there you can see more up-to-date, detailed information about variants in the context of African populations. So going there, the service, it's still in testing phase, so access is a bit limited. So you'll need to log in to get more information, and once you're there, you can start doing your search. So we say that from our previous search, just trying to move this. So we were interested in this SNP. So I want to know more about that SNP. Then I can take the SNP and use the variant ID and come and do a search. So doing that, I get some results, and then I can see in different studies or data sets all over the world what the frequency of this variant is, and then I can even go further For instance, if I go into 1,000 genome, I can see the frequency of these variants in various, African populations. So it also gives you, a nice map where you can see this variant. So with the H3Africa data getting into this database, it will be a great resource, to get information about variants. The third, another resource that you can use, it's what we call beacons. So it's an API, created by the Global Alliance for Genomics, G4GH, which is basically an API that you can deploy on your data or any database. So if you have your data, you can still keep the ownership of your data if it's hosted in your local institution, but then use this API to be able to share your data with the whole world. So basically, you'll go there and do a simple query, search about your variant, and you get an answer yes or no if that variant has been seen in any study. So upcoming versions, will provide more information about things like the number of samples with that variant. But right now, what you get is basically a response on whether or not that variant has been seen in any other population. So we can quickly go onto the Beacon network. So Beacon API by itself, you deploy it on your data is good, but it's more useful when you integrate it with a network. So which means when I do a query on a variant, I'm just not querying one beacon, but then I'm able to query, a number of beacons that are part of a network. So in this case, if you explore this website, you can see all the beacons that are here, but we can try to do a quick search on the variant that we found. And this is the genomic position of that variant. Then I can search for that variant. And then we start getting responses. I can try to show everything. And it's telling us basically that the variant that I've just searched is found in the first project, but not found in, for instance, BRAC exchange and so on. And if you scroll down or you do a search here, you'll see that one of the African data is found here. So if you look here, for instance, we see that we have the SHGP, which is an African project or data set, and we see that that variant is found there. So it's a very interesting tool where you can deploy this API and then connect it to this network, and people will be able to query in one query, be able to see if a variant of interest is found in your data or study. So, if you're looking at doing a GIS or interested, doing some genotyping, you want to know if the variants that I'm interested in is, for instance, on the H3Africa chip. So we've developed a simple tool that you can use to see if the variants that you're interested in is on the H3Africa chip. So it's we basically call it H3Africa chip info site. And if you go there, you can, let's go back and get our variants of interest. So if you go onto that website, you can do a search, then put your variant ID. So you can search by ID or region or even gene. And if I click search there, let's see if I'm fortunate. Yes, So the snip that we're interested in is actually on the H3Africa chip. So you can click there and get more information. For instance, one of the other information that you can get is why is this variant on this chip. So in this case, we see that this variant is on the chip on the PI request. So it's a very interesting small tool that gives you, a lot of information about what is on the chip. So I think I'll stop here for now. And for those who are interested, we can maybe take a 5-minute break, then we can come back and continue with the rest. Thank you.
I was just going to do one announcement and then. So great. So I think that's a great time for a pause. That's really cool. That's the first time I've seen the beacon used that way as well. So that's fantastic. So there's a photo. We're going to take a pause. We're going to have coffee. You're going to talk. Did you want to do the announcement about the photo?
We just sort of changed it. So that's what. So we're going to ask actually that everybody, go and get your tea, but do it quickly and don't linger too much and come back in here because we're running a little late and we do want to take a photo right before lunch and we do want to eventually have lunch not too late in the day. So so if people can come back in from tea, get it, come back in, you're going to do your demo as soon as you see people start to trickle in. And then we will try and get the next session started fairly soon. And then, so right before lunch, so after the next session, we'll all gather in the lobby for the photo.
Not really. Well, okay. They were completely.
conduct genetics and genomics research, researchers need to obtain human biological samples such as blood, saliva, hair, etc. From these samples, the DNA is obtained and studied to understand why and how we are affected by certain genetic diseases. They may also collect other measurements such as weight, height, blood pressure, blood sugar levels, etc. The process involves many steps and it is very expensive and requires collaborations between local researchers and researchers in other countries. Most of our local research institutions are often not fully equipped or are technically challenged to do all the genetic analysis. The collaborations between scientists across Ghana and other countries enable data to be obtained and analyzed to understand the genetic basis of diseases more quickly for the benefit of all. We will now take you through the journey of your sample, from sample collection and transportation to local laboratories, initial sample analysis and processing in the labs, and storage and transportation to more advanced labs, and finally to biorepositories. The journey of the sample starts from when it is collected in the community or clinic setting. The purpose of the research and the sample collection will be explained to you by the researcher or field staff. Your participation is your personal decision. If you agree, you'll be given a consent form to sign or thumbprint before the samples are taken. Blood is the most common sample collected for research purposes, but you may also be asked to provide saliva, urine, or stool. Blood samples can be taken by pricking your finger with a small needle and dropping the blood on a slide or special paper, or by putting a needle through your arm or vein and drawing a quantity of blood using a small tube designed for this purpose. The tubes are labeled with a number that is unique to you to protect your privacy. The quantity of blood to be taken depends on what it is going to be used for. It could range from about half a teaspoon like this, or as much as 10 milliliters, which could be split into 2 tubes. An average human has about 1.2 to 1.5 gallons of blood like this. And the maximum quantity of blood collected for research purposes is about 10 milliliters like this. The samples are carefully packaged and transported to the local laboratories such as the one in the Navrongo Health Research Center in northern Ghana or the Noguchi Memorial Institute for Medical Research local lab in Greater Accra for storage and initial analysis. When the samples arrive in the local laboratories, they are received by the lab personnel. The samples are stored in the freezer until they are ready for processing or shipment. Information about the samples is also entered into a computer so that researchers can locate the samples in the freezer easily. In addition, the samples are assigned a new ID separate from the field or clinic ID to further protect the privacy of participants. DNA is extracted from parts of the samples and divided into 3 different parts. DNA extraction involves taking a quantity of the blood sample or saliva and adding chemicals that will break down the cells and release the DNA in solution. 2 parts of the extracted DNA are stored in the freezer. The remaining 1 part is processed locally to make more copies of the DNA, that contains the genes of interest. In order to make more copies of the DNA, chemicals are added to the extracted DNA before it is put in a machine to make more copies. This process makes the gene of interest easier for us to analyze. Some of the information that can be generated from this analysis may include whether you have a mutation in the one gene that causes a disease such as sickle cell anemia or not. After these initial analysis, the remaining DNA is stored in the fridge or freezer for local use, and the processed DNA is stored until ready for further processing.
Yeah, I guess we're going to continue with our demos. If you're outside and interested, please, you can come in.
All right, everybody, we're going to try and get started soon. So hopefully people will be coming in, but I just want to make a quick announcement. Because we're running a little bit behind, Maimuna is not going to do the demos right now. Hello? All right, so we're not going to continue with the demos right now, but we will do that at the break later on this afternoon. Maimuna will be demonstrating the catalog and the imputation service. So just be sure, you know, at that time that you get your tea and come back in for that demo. Can anybody hear me? Oh, hold on. Is this better? Sorry, it's the mask. Okay. All right. I'm going to say that again quickly. We're going to get started with the next session. We're moving on. So anybody who's interested in seeing about the catalog and the imputation server, Maimuna will be demoing those in the afternoon break instead of the morning break. All right. Over to Audrey.
Hi, everyone. You've probably figured out that I'm not Charles Rotimi. Unfortunately, he's not able to be with us. We're going to get started on the session 7, which is the genomics of complex disease in Africa. And our first speaker is Joseph Ocheng. Joseph? Excellent.
Yeah, good morning. Yeah, I'm presenting our work from the genetics and genomics research in Uganda, and I'm from Makerere University. How do we move this? So our presentation will go through this outline. And we are sharing findings from our study. As I've said, it was the genetics and genomics research in Uganda. And we wanted to develop context-specific guidelines for ethical conduct of genetics and genomics research in the country. And this was our goal. And it was to be achieved through a number of objectives, but specifically wanted to assess stakeholders' awareness and the nature of the ethical and social concerns associated with the genetics and genomics research in Uganda. And then later would use the data generated to develop guidelines. So we carried out a number of activities, and these ones included the scoping review of literature. We did a survey. We did in-depth interviews as well as the focus group discussions, and all these ones would feed into a Delphi process for consensus building in order to generate the guidelines. These were our stakeholders. We had research regulators, we had the research ethics committee members, we had the patients, we had the individuals who had participated in genetics and genomics research, we had the researchers in genetics and genomics, as well as individuals from the grassroots community. So all these ones contributed to the data that we generated. And these are the detailed descriptions of what we did. For the scoping review, we did the documentary review and the analysis and synthesized with the focus on what guidelines are available within the African context, what policies are there to guide genetics and genomics research on the continent. Then we had a survey of research ethics committee members. We have 25 that time we had 25 ethics committee members that are accredited by the national regulators, and they had a total of about 250 members. So we surveyed 111 participants from those committees, and we also had a survey of genomic researchers in the country. to get their views and knowledge and input on the aspects. We also did in-depth interviews with the research regulators, specifically 3 regulators at the national regulatory agencies, as well as the 19 research ethics committee chairpersons. We also had 30 interviews with the genomics researchers in the country. We had focus group discussions. This involved 66 genomics research participants and 96 individuals from the grassroots communities across the country in different regions of the country, as well as 18 patients at the hospital outpatient unit. And then the Delphi process of consensus building. And following those activities, we have a number of results that we've generated. For example, the scoping review showed that the enforceable policies and for ethical conduct and review of genetics and genomics research in Africa are mainly deficient or lacking in many African countries. Actually, many countries do not have guidelines for this, including, of course, Uganda. And then we also found that there are existing international guidelines like those developed by the H3Africa working group that could be adopted to the local settings if countries really work towards that. Then in the survey, we conducted a survey, like I said, though it involved the researchers and research ethics committee members. And we looked at aspects like informed consent, community engagement, privacy and confidentiality, return of results, and counseling. And of course, respondents were provided with the questions, and then we scored them out of 15. So 61% of the respondents in the survey were considered to have good knowledge, and those are individuals who are scored 9 out of 15. And then 83% of the research ethics committee members and 77% of genomic researchers recommended the need for additional ethics guidelines for conduct of genetics and genomics research. Then the in-depth interviews, like I said, we had 30 in-depth interviews with the researchers in genetics and genomics, and 80% of these had done and conducted genomics research for at least 5 years. And they highlighted challenges of lack of ethical guidelines. No specific considerations were put in place whenever they were doing their research. Really, they just did the research the usual way, as if it was the ordinary research we normally do. 30% reported that they have ever attempted or conducted the return of results to participants. And they all stress the need for meaningful community engagement, guidelines, and of course, consideration of consent, privacy, and implications of the study. And these are some of the quotes. I would return those results to a patient if I knew those results are going to help a patient receive better treatment, or if I knew they were going to affect him. And then another one said, we need to have some guidelines and policies in place, especially for clinical research or studies that go deep look at genetics and genomics of individuals in the hope of coming up with better treatment options. Then the in-depth interviews with the research regulators were not much different from the ones we found with the researchers. Most of them had no experience of regulating and providing oversight of genetics and genomics research. They highlighted challenges of lack of guidelines, and they also had no specific considerations. Most of them had no specific considerations for genetics and genomics research. And of course, they stressed the need for guidelines, training, aspects on community engagement, genetic counseling, among other aspects. And this is one of the quotes, maybe I take the whatever. One of the ethical aspects we usually discuss is counseling and consent too. Counseling is very important. needs to be done as if you're going to return results because it has psycho-social implications. Then we had focus group discussions, 10 of them, with the individuals at the grassroots communities, and they all had no experience as in participating in genetics and genomics research. Almost 100% were supportive of participating in genetics and genomics research, but they all stressed the need for feedback of results because they said it will be useless for me to participate if you're not going to give me the results. Why would you test me in the first place? They also highlighted the need for meaningful engagement. They needed counseling and informed consent as you do that one. And of course, we also talked about issues of whatever extending feedback to family, and almost all of them said, yes, I would, at least one family member who is close should be able to know my disease. And then a few of them would say it would also be good to extend feedback to community, because many times if we fall sick, the people who come to our rescue are not normally the relatives, but the neighbors who are around, who could be, it could be good for them to know. And they give examples, for example, sickle cell. If you have a sickle cell and you don't disclose, and then you get an attack and you need help, it's usually the neighbor that will come to your rescue. Then these are some of the quotes. I want to know my results, because if you test for anything, you have to know your results so that I can know if I'm healthy or sick. We had the 6 focus group discussions with the individuals who had participated in genetics and genomics over the past 4 years. And they had some knowledge of DNA testing. Almost 100% were supportive, and they said, we will test again if given a chance to participate in a genetics study or genomics research. And they had issues on the consent. Here, they were not contented with the consent that was given to them. They didn't seem to understand the research they were participating in, and the genetic counseling was inadequate. So those are challenges that they raised, and they said these ones should always be stressed in such research. And this is a quote. For me, the challenge I experienced is that I did not get most of the things that they were telling me about as clearly as I'm getting them today. So it's like they were telling us, your informed consent, the information you're giving us is good, but the other time we did not understand. We had the 3 focus group discussions with the patients at an outpatient hospital clinic, and the 18 individuals participated, and most of them had known about DNA-related testing, because DNA is quite common in Uganda, particularly when it comes to paternity testing. So it's a very common thing. People talk about it. They stress the need for informed consent, counseling, and all those related aspects. They were ready to participate in genetics and genomics, as well as ready to receive results. What we realized that most of the individuals, apart from the researchers and the research regulators, the other stakeholders were almost 100% wanting to participate, as well as wanting to receive the results. So we need to change the way we look at things. And of course, they were ready for anything, because they said, whatever comes out of the testing, whether they are incidental findings, is part of my body. If you do not tell me today, it will still remain in me. And if it's a disease, it will come and affect me. So you rather warn me early so that I can prepare. And this one, I would ask the doctor to explain to my sister that I would have come with, and I would want us the two to know about it than others who may spread it. So that one is extending feedback to family. But then for the community, someone says, there are 2 things. The first one, knowing would be good, but the community has one problem. Instead of helping you to be around, it helps in destroying you. So that was the hesitance about sharing feedback with the community. So we've had 2 publications so far, but there are others in the pipeline that should be coming up very soon from this study. We've managed to engage some stakeholders, and one of the key stakeholders is the National Regulatory Agency, the Uganda National Council for Science and Technology. They are on board. They are currently revising their national guidelines, but actually they put on hold that process pending our input from the genetics and genomics study. So we are really confident that they're going to take up our findings, and we will really be happy to have them. I chair that committee that is revising the guidelines, so I'll try to make sure that we incorporate the guidelines in the national guidelines, but we will also come up with the probably different booklet that is sort of an addendum to that, which would have detailed guidelines on genetics and genomics. Most of the other stakeholders, particularly the researchers, are also supportive of the guidelines, and the research ethics committee members, and they have been demanding for that. So every time we talk to them, they say, yes, we are ready, we are willing, and we need to do a continuous engagement process. So we've requested for a supplementary to engage as many as possible, all the ethics committees involved and all the researchers involved in genetics and genomics, so that when the guidelines come, they really hit the road running. So basically, I think I've talked about most of these ones. The considerations include, for example, people need those ethical issues to be addressed. For example, genetic counseling. It's interesting that all researchers stress the need for genetic counselors. Ironically, there was no research projects, including those funded by H3Africa, that had a qualified genetic counselor in the country. So basically, I could confidently say at the time of our research, there was no qualified, well-trained genetic counselor in the country to do genetic counseling. And those are very important aspects, in addition to adequate informed consent that continues as a process, not a one-time thing. And that should be given before you test and before you give the results. There was a need for guidelines. That one, we're really going to work on it, and this is the objective, one of the objectives of our study. And there is need for training and the continuity aspects. So we're going to do that one. So we appreciate our funding from H3Africa. We are very grateful. We wouldn't have done this work without that, with that funding. We are also very grateful to our participants, all the categories that participated. And these are my other study team members. Thank you all. Yeah, thank you.
Thank you, Joseph. That was fantastic. I'll call you back up at the end to be on the panel for discussion. But for those submitting questions online, please indicate who the question is for if you can. And if you're in the room, there's no need to submit a question online because it's a little bit confusing. Thank you very much. Next talk is Duomo Adu, who's going to tell us about capacity development.
Thank you, Audrey. Okay, so good morning. I just wanted to talk very briefly about capacity development, and that's not across the whole of H3Africa, but across some of the studies, and I've picked out 3 or 4 studies. And capacity development is obviously important because this will shape the development of independent genomic research in Africa. I think that one of the things that perhaps we haven't highlighted as much is the importance of research ethics and compliance training, and in the UK it's called good clinical practice. And one of the things that all the researchers engaged in H3Africa have had to do is to do these courses on good clinical practice and get certified prior to being able to do any recruitment. And actually, these are renewable. So in our cases, every 4 years. So we now have a huge cadre of people who are trained in good clinical practice. There are now extensive opportunities for genomic training in Africa, and I'll only pick out some examples of that, and that's Abuja study, Hygenes, Siren, H3Africa, Kidney Disease Research Network, and then I'll end up just talking a bit about the West Africa Genetic Medicine Center. So if I look at the Siren training, that's the Stroke Investigative Research Network. So they have trained a large number of clinical research and biostatistics training. There are individuals undergoing master's training, PhD training. They've had grant administration training, and also research nurse and clinical coordinator training. So that's one of the studies, Siren, that has got a huge training portfolio. You've got Hygenes, it's a hearing and genetics training program. And again, they had PhD, awarded PhD training, grant administration training, grant writing training. And I think, and in addition, I've had a huge amount of training across several aspects of genetics, from genetic counselors courses, computer-aided design of small molecules, ensemble virtual and genome browser training, good clinical practice again training, and that again, as I said, is really, and good laboratory practice training in statistical genetics. So again, a huge portfolio of training that has been available through the Hygenes project. Abuja, again, has had a huge training portfolio, MPhils, PhDs awarded, PhD training. They've had not only grant writing training, but a lot of success in being awarded grants. There's a CVD supplement, Abuja supplement, the Cardinal study, which is a polygenic risk score study, and then also DSI Africa. In addition, I think what's been really important, because in order to do genetics, you need genetic studies, you need to have trained research nurses, clinical coordinators, and data clerks, and Abuja have trained over 50. So it suggests from Ghana, Burkina Faso, Kenya, and South Africa. And in addition, have 7 postdoctoral trainees. The H3Africa Kidney Disease Research Network, likewise, we've had clinical research training and biostatistics, the 30 of them, several MPhils and PhDs awarded, and also grant administrator training and grant writing training, and then over 100 field workers and laboratory staff in 14 centers in Ghana and Nigeria. And I think one of the really important things that we perhaps haven't discussed, that we've been able to have, I've called them interns in our genomic laboratories. So these are people who've come through for maybe 3 or 4 months and had training in genomics. So I tried to summarize all of this because there are quite a lot of disparate groups. So the clinical research and biostatistics, there are over 30 people have been trained. MPhil, MSCs awarded 13, PhDs awarded 13, grant administrator training, again, there have been over 10, and grant writing training, and almost 200 research nurses, clinical coordinators, and data clerks have had training opportunities. And in addition, there have been other training centers. So if I summarize the training domains here, make it slightly easier to see. So we've got clinical research training, and there've been over 30, MPhils awarded 13, PhDs awarded 13, and then grant administered training 10, and grant writing training. So there's been a huge amount, and this is just across the 4 centers. I'll just end up by talking a bit about the West Africa Genetic Medical Center. The director of that is Solomon Ofori-Akwa, and he gave me information on the students that have been taken on since 2019 when this program opened. So there have been 5 in 2019, 27, 33. So there are now almost 60 students who are doing training in genetics across a range of disciplines. And I must say that includes genetic counseling. So we've got 6 students who are currently doing either MPhils or PhDs in genetic counseling. So the director of the program is Professor Solomon Ofori-Akwa. It's an African Center of Excellence with World Bank funding, and the training is available to all African nationals. I've got to be careful, but if you look at their website, so there's a real huge training opportunity. And I think as we've gone along just over the 4 centers, it's become apparent that there are now tens, if not hundreds, of training opportunities right across Africa that people can then take use of in genomic medicine. So thank you for your attention.
Thank you, Joma. I'm going to call upon Mayowa Oblabi, who's going to, according to this list, is that right? Sorry, Michelle, it's you. Jumped ahead. Sorry. Yes, you are. You're now, Michelle. Sorry, it was me. Yes.
Hello, everybody. Very nice to be here again. And my task is really to talk about several different projects that have now done genetics and genomic studies. And this is a session on complex disease genetics. So I'm really going to highlight the work of 5 of the groups. And that is the Abuja study, the Kidney Disease Research Network, RHG Gen, Sickle, and Siren. So I'm speaking on behalf of these people, and it's been a great pleasure to review their genetics papers and to see where we are at the moment, but also where we're going. So I thought, just to start off, it's quite helpful to have a slide on the descriptors of the different studies, and then to show you some of the published work, not all of it, because of course 15 minutes wouldn't allow me to do all of that. So this is really giving you a sense for the size of the studies and the approaches that they've taken to trying to identify genetic contributions to complex diseases. So the Abuja study is a population cross-sectional study of over 10,000 individuals, and the main outcomes that were being looked at here were related to cardiovascular disease and metabolic disease. So what I've put in the sort of the heavy font are the papers that have been published, and the others are still in progress. And that sort of goes across all the different studies. So what you will see on the right-hand side in the column here is the arrays that have been used. So overwhelmingly, these studies have used the H3Africa array, which was really a consortium project. So the second study to talk about is the Kidney Disease Research Network, which was a case control study of over 5,500 individuals, looking at the main outcome as chronic kidney disease, and then the rheumatic heart disease study of over 4,800 individuals, which is also a case control study. The sickle cell study, which was more the complex disease aspect of it, was looking at fetal hemoglobin. And this was again a population cross-sectional study of 1,600 individuals. And then finally, the SIREN study, which was looking at stroke and also at diabetes in the context of stroke in over 3,400 individuals. I've just done these in alphabetical order for ease of just talking about it. So, so you can see that they're all different approaches and have different sizes in terms of their studies. So first of all, I'm going to highlight some of the papers that have come out of the Abuja group, and then I'll take them sequentially. So one of the first studies that was published was the genome-wide association study for carotid intermediate thickness, which is an early marker for atherosclerosis. And this was published by a former PhD student, now independent scientist, Pawende Boa, and this paper was published earlier this year and really looked at sex-specific effects as well as doing general genome-wide association studies. But I just wanted to show you some results from his earlier study, which was to look at gene-environment interactions and how that could predict carotid intermediate thickness. So this study was only done in West African men. And that was very interesting to us because we wanted to use our entire cohort, but that wasn't possible because very few women in West Africa smoke. So, so although this was a smaller study, he did find some interesting interactions. And you can see one here where on the left, the data is stratified. On the left are the individuals who didn't smoke, on the right are those who did smoke. And for this particular variant, you can see that if you're a non-smoker, the variant doesn't impact the thickness of your carotid artery wall. But when you are a smoker, if you happen to have the TT genotype, your carotid intermediate thickness is higher and your risk is higher. So I think that this is something really important that has to come out of our work is the gene environment interactions, because that also gives you targets for intervention. So the second study that I want to highlight is the genetic association with lipid levels. And this paper was published just last month. And what is interesting is that we're finding that straightforward genome-wide association studies are no longer enough, that we now have to meta-analyze with other studies if they are available. And of course, people are always looking for replication because that would boost your confidence that the results are actually meaningful. So this was done in Abuja in the first instance, and then as a meta-analysis, which took us then from just over 10,000 participants to over 23,000 participants from Africa. So what you'll see in the Miami plot on the right-hand side is that the bottom is showing genome-wide association study with LDL cholesterol, just in the Abuja study, and above is the meta-analysis. And what you can see is that many of the signals that have come up universally in lipid studies are still coming up. And some of the signals seem to be boosted with the meta-analysis and others are damped down. So that is interesting. And what we did find was one novel association that hadn't been found in millions of European participants where they've done genome-wide association studies for lipid levels. So what I wanted to show you in the two locus zoom plots at the bottom, one is on the locus on chromosome 19 for the LDL receptor. And what you can see in the top one, those of you sitting in the front, is that in European studies, there's quite a broad band of association, whereas in the African study, because of the lower linkage disequilibrium, you see a much sharper sort of signal helping you home in on the causal variant. On the right-hand side is the novel locus that we identified on chromosome 4. And what was really interesting was that in the Abuja-only study, it came up in a particular gene, but when we did the meta-analysis, it shifted the most significant SNP to another position. in another gene, but very close together. So we still don't quite understand what's going on here. We think this is a real study because it's highly significant, and we need to now think about the biology of the contribution of this gene to lipid levels in African populations, but not in other populations. Then we also published a paper on runs of homozygosity or autozygosity as like a marker for inbreeding effect. And we showed that there was a significant decreasing effect in men, but a significant increasing effect in women. And I think what this highlights for me in terms of cardiovascular disease and metabolic markers is that we need to do sex stratified analysis, because there are sometimes very important sex differences. So we showed that effects tended to be more significant in men and were more intense for people living in urban areas rather than rural areas. On the right-hand side is also the very recently published work on apolipoprotein and looking at association with albuminuria, but also low EGFR. And this work shows us that there are differences in the frequencies of the high-risk genotypes across different regions of Africa, but also that the APOL1 risk variants were associated with albuminuria, but not EGFR. And this seems to be a theme that is coming out in other studies as well. And what we showed is that there are potential differential effects for the G1 homozygotes when compared to the G2 homozygotes. So I think that pattern is just becoming more complex as we try and understand it better. So now moving on to the genetic studies from the Kidney Disease Research Network. And they have also looked at APOL1 as one of their first studies, although they are planning to do genome-wide association study for kidney disease in Africans and their descendants. So here they had a different study design, which was a case control study design. They had a wide age range, and they defined kidney disease as a GFR less than 60 and albuminuria as a UACR over 3. And what they showed is that the prevalence of chronic kidney disease, about 14% in Ghana and Nigeria, and you have to forgive me if I got anything wrong in my interpretation. And what they also showed is that Africans succumb to end-stage renal disease 20 to 30 years earlier than non-Africans. And I think this is really significant in terms of needing to understand that. And so, of course, because of that, it extracts an enormous toll in premature mortality and cardiovascular comorbidities. So the main conclusions from this analysis, which is now, I think, being submitted for publication, not yet published, is that 28% of all the participants that they analyzed had 2 APOL1 high-risk variants. And the prevalence of the high-risk variants was significantly higher among those with kidney disease compared to the controls. So it was 30.6% in the cases, there's a typo there, and 26% in the controls. They also showed that the G1 haplotype was more frequent than the G2 haplotype. And what they showed was that when they did the association analysis between APOL1 and kidney disease, that they got an odds ratio of 1.29 when they adjusted for age, sex, and clinical site. So really looking forward to seeing this in print soon. So the next study that I want to highlight is the main paper that's come out of the rheumatic heart disease genome-wide association study. And you can see multiple authors there, and the first author being a young researcher, which is what we want to see on these papers. So their main question was, is susceptibility to rheumatic heart disease heritable in African individuals? And what they found in their 4,800 participants was that there was one novel genetic risk variant on chromosome 11 that was significantly associated with rheumatic heart disease in Black Africans, but not in other groups. And then they managed to replicate another signal that was important. So this has impact in terms of that they've identified important polygenic component to rheumatic heart disease in African individuals. And some still remain to be found. So you can just see some of the results here on the right-hand side. So the next study that I'm going to talk about, let's just see if we can advance that. I'm pointing in the wrong direction. Has it gone? Okay. Hopefully it won't skip another one, seeing that I pressed it twice. But this is just highlighting two of multiple papers that were done on the genetics related to sickle cell disease. And as you can see, the first one is really about genetic modifiers for long-term survival of sickle cell anemia. And this involved whole genome sequencing of almost 200 African participants. And what they identified in the study are new gene sets that are relevant to variability in the clinical expression of sickle cell disease. And on the right-hand side here, it's about looking at kidney dysfunction-related genetic variants in sickle cell disease patients in Cameroon. And here they investigated 31 variants, and 6 of those were associated with kidney function in people with sickle cell anemia. And what you can see at the bottom on the left-hand side is the effect of alpha thalassemia on the hemoglobin F levels. And on the right-hand side, the effect of different haplotypes surrounding the sickle cell mutation on levels of hemoglobin F. And we know that this is very important in terms of the severity of sickle cell disease. So I thought that it would be important for me to look at the main generalizable conclusions. And just to say that the Siren studies still have their genetics in progress, so I couldn't present any results. But again, you know, we know that these are works in progress. So first of all, you know, what we've learned is that allele frequencies differ across African populations and compared to non-African populations. Novel complex trait associations are detected even with modest sample sizes in Africa. And when we do replication of known loci, very often the lead SNP is different to what has been found in other populations, but also the effect sizes are different. And of course, this would impact things like polygenic risk scores, because that's exactly what we use in polygenic risk scores. And we know that transferability of genetic risk is better if the discovery cohort is multi-ethnic or is matched to the target cohort. And what we're also understanding now is that there are regional differences across Africa, and this is perhaps not unexpected. And it means that, you know, as we're developing precision medicine approaches, they probably wouldn't be exactly the same for different regions of Africa or different ethno-linguistic backgrounds. So what have we learned? That we need larger cohorts. We've done an amazing amount of work, you know, on getting cohorts in terms of H3Africa, but we need more. We need longitudinal data because we want to be able to predict. We want to look at gene environment interactions. And I think that's really key. And that's often why we see differences between African and European studies. And we want to look at gene-gene interactions because this will allow us to identify novel biological insights and also to eventually look at translational impact in terms of complex diseases. So what about the future? It's all about collaboration, working together because we are stronger as we increase our sample sizes. And we want to make sure that our cohorts are sustainable so that they can go into the future and we can continue collecting data. So we have numerous collaborative partnerships here. I have to stack them up there. And we can look at things like kidney disease across multiple projects. We can look at anthropometry. We can look at other phenotype overlaps between our groups. Very importantly, we want to expand this whole meta-analysis of APOL1 variants and kidney function in the context of hypertension, for example. There's so many different questions that you can ask in different ways. We want to do GWAS for blood pressure. And what you can see at the bottom there are a number of larger consortia. And what we can do now is, as H3Africa, we can start contributing to these as well. And it's always interesting and gratifying how much value, disproportionate to its size, the African genomes are contributing. So what are some of the issues? So we've all struggled with delays in getting our genome-wide association data. We've worried about data QC, batch effects, that sort of thing. We've been concerned about imputation, what are the best panels to use, and there's a paper in progress at the moment really critically evaluating what should be used, not what should be used, but what happens when you use different imputation panels and how that affects your ability to enhance your data set. We've also had challenges with skills to analyze genomics data. We don't often have dedicated analysts, and we also are concerned about sustained funding going beyond the project closeout dates. But what we can say is that although there have been some delays in publication, there are many in the pipeline, and these studies are now coming into the public domain. More people will know about them, and I think as that happens, we will also have more opportunities for collaboration. So I hope that everybody will watch that space. Thank you very much.
Thanks, Michelle. That's incredible run through some of the results in H3Africa. And now I will call upon correctly Mio Owolabi to talk about the chair resource.
Thank you very much. Okay. Thank you very much. So I'm presenting on behalf of the chair group. That's on behalf of about 7 other PIs. Cardiovascular diseases since 2019, and we saw this coming, are now the leading causes of death on the African continent. And we need to do something about it. And this is the reason why we have the SHARE Consortium, which comprises about, right now, I think about 7 or 8 different studies, pulling all our resources together to better understand the reason and the basis, both genetic and environmental basis, for the rise in cardiovascular diseases on the African continent. And this will give us the opportunity of coming up with precision medicine solutions to tackle the burden. So we started out with a combined cohort of about 55,000 subjects, but that has since increased to right now over 70,000 and more than 13 countries. And looking at the entire spectrum of cardiovascular diseases from across the life course, from no risk factor to those who eventually have established cardiovascular diseases and target organ damage. We collect different cardiovascular phenotypes across the different study groups. So there was a special issue of the Cardiovascular Journal of Africa that was dedicated to this. And one of the publications there, which is Understanding the Rise in Cardiovascular Disease in Africa, actually was one of the most read CVJ articles for that particular period. So we also documented the profile of the cohort, the 55,000, which we started with in the International Journal of Epidemiology. Basically, the objectives of the SHARE is to enable us to do what we can't do within single groups by tackling cross-cutting research questions, which individual projects are under power to explore, examining genomic associations across different cardiovascular phenotypes across various African regions. And then, of course, this gives or enhances the potential for discovery, validation, and generalizability of African, and gives a Pan-African perspective with unique opportunities for collaboration and data sharing on controls, provision of validation codes and meta-analysis, some of which are already ongoing. And of course, capacity building across all the aspects of genomic science. And the ultimate goal is to be able to come up with interventions that will be cost-effective for diagnosis, prevention, and treatment of cardiovascular diseases on the African continent. The first major hurdle we had to overcome was harmonization of the data sets, because each individual study had their own CRFs, They have their own research, unique research objectives, and their own unique way of collecting data. So this was really a very tough job that took, I think, almost up to 2 years to harmonize all these phenotypes so that we can compare information across board. And that included socio-demographic and economic status, medical history, lifestyle data, family health history, clinical data, of course, on cardiovascular phenotypes and biomarkers of CVD relevance in blood and urine for 13 countries, more than 50,000 subjects for 67 phenotypes. We were able to harmonize 67 phenotypes that were collected by at least 2 out of the 7 different studies. So we began to put this to use to tackle one of the greatest burdens of disease that we have on the continent. You may be surprised, but that is actually true, that Africa is the global capital for hypertension, with the highest prevalence of hypertension. Even at the definition of 140, using the 140/90 cutoff, we also use the 130/80 cutoff, which was the new one at the time this paper was coming out. But even at that, at the 140/90 cutoff, according to WHO, for those who are more than 25 years old in Africa, 46% are hypertensive. And that's almost 1 out of every 2 African adults. So we need to know why this is happening. And the other problem is that only 7% of these are controlled, meaning about 93% are either not diagnosed, not on treatment, or not controlled. So we looked at the regional pattern of hypertension, and we also looked at that of obesity. And we were trying to understand if there was any association between obesity and hypertension. And indeed, we found that with increasing BMI, the chances of obesity, of hypertension also increased across different African regions, Southern Africa and East Africa, West Africa, and overall. And that obesity at least doubles the chances of being hypertensive. And this adjusted odds ratio for hypertension in those who are obese actually increased with age, both for males as well as for females. The other thing we looked at is we're trying to find out the other things that are associated with hypertension, which can be used for primordial prevention of cardiovascular diseases, or at least primary prevention of hypertension itself, which is actually more difficult. This is a new finding from what we did. This is actually between Cyrene and Awijen. And we found that we were able to establish a dose-response relationship between consumption of vegetable and occurrence of hypertension among Africans. Actually, this is happening for the first time. We're able to establish that for those who consume at least 12 servings of vegetables per week, they are more protected against hypertension. And we're able to demonstrate this also graphically. So those are some of the things that could be tackled. There are other things that we are doing, looking at dyslipidemia, looking at diabetes traits, and the kidney group, the Awijen group, the ACME group, the cardinal study which has been mentioned. Now, what are the emerging solutions for leading cardiovascular diseases in Africa? Of course, from what we've said already, for hypertension, and we're working with World Hypertension League, and this is actually in press already, call to action from the World Hypertension League. And part of the major things that we included, of course, is recommendation to increase vegetable consumption to reduce the burden of hypertension. And of course, weight reduction, which will tackle obesity and also help in reducing the burden of hypertension. And our publication on the proportion of hypertension on the continent was actually republished by Hypertension Journal. It was initially also published by Hypertension, but was republished under the call to action, which has just been published. And the call to action is going to be followed by the Achieve ecosystem, which is going to engage policymakers, patients, payers, general population, resolve to save lives, and all of that to ensure that we actually are able to control hypertension on the continent of Africa using findings from SHARE. Then we're also doing this for stroke with the Afrocentric stroke prediction model, and also with a video based on the findings of the SIRENE study, which is published in Lancet, on the risk factors for stroke and how to prevent stroke. So those are some of the things we are trying to translate. And we have taken this to the community, in the ARISES community, in collecting data, following up, and also screening for cardiovascular diseases, and also globally for stroke prevention. So basically, what solutions are we anticipating that will be emerging from the SHARE resource? Changes towards health, like lifestyle changes based on our findings. For instance, if we're able to control obesity, control diet, we might be able to reduce the burden of hypertension on the continent, and also be able to control those control hypertension better for those who already have hypertension. New biomarkers may come out, risk prediction models for hypertension. There was one that was presented yesterday for hypertension by one of the fellows, and of course the one for stroke we already showed. And these may have continent-wide applications using the SHARE resource. And then precision medication selection, rational polypharmacy, and tailored environmental exposure. So those are things that maybe, and of course, capacity building. We're doing this for neurology across Africa. We are also able to establish the African Stroke Organization. And of course, the World Hypertension League is leading activities to control hypertension on the continent. And leveraging our SIRENE network, we were able to establish the first, the African Stroke Organization, and we had a conference last year, and we're going to have one. And of course, our work has been recognized globally, even by the World Stroke Organization, with the 2021 award for best performance in clinical stroke research globally was given to our group. So I want to congratulate and thank every member of the SHARE Consortium for what we are doing, which is still work in progress. And hopefully we'll be able to do more to understand the burden of cardiovascular disease, which is now the leading cause of death on the continent, and to control it better using precision medicine products emanating from the SHARE resource. Thank you very much.
Thank you very much, Miowa. And last but not least, we have Ambroise Wonkum, who's going to tell us about the broader context of the impact of African genomics.
So thank you very much. I think previous presenter make my work extremely easy. So I'll try to summarize why we are here. So I'll try to convince you that the next frontier of global genomic medicine, not just in Africa, rests within the variation we are all investigating here. Yeah, there are three reasons for that. The first reason, we are all African. The second reason, variability of the ecology of Africa. The third reason, making genetic medicine equitable for all. Let's start with the first reason, and I will start with complex threat based on some of the data presented by Michelle. For example, these are evidence that African population at the moment in terms of the genomic have more variation that are not yet captured by the reference genome. This is a study of only less than 1,000 people living in the Caribbean island, and by using a pan-genome approaches, 10% on the content of their genome is not present in the reference. We know that there are some humanoids that never move out of Africa, unlike, for example, the Neanderthal that moved to Europe or Denisova to Asia. And some humanoid, for example, Lucia or Mona Lady in South Africa, mixed with us. We actually make children with them, but they never move out of Africa. And we know that the proportion of those variants that is currently called the ghost DNA can only be well captured if we investigate African population. This is important for health and disease, but we don't yet know how. We know that it is possible that the proportion, for example, or the DNA from the Neanderthal that is present at the 2% level in European and Asian have contribution to some condition, including amongst the susceptibility to COVID-19. The reverse is probably true. So those ghost DNA that we haven't yet investigated and that we need to investigate for the next 10 years to come within African DNA will also help all of us to know what is the contribution for health and disease. I think Michelle presented a version of this and you already saw it. Despite the whole effort that we have, only 2% of the global GYs contain people with African ancestry. And data from the UK Biobank shows that this not working very well using polygenic risk score and it's not very equitable in the sense that it doesn't capture the proportion, for example, accuracy within population of African ancestry. We know that we are all losing by not doing that because African population have shorter haplotype. That means that it makes file mapping easier in population of African ancestry. They account for 2 times more association as compared to their size of only 2% of participants. Some variants are very common amongst African and relevant to health and disease. I think this is the most common one used globally. A variant that is 200 times more common in African than in European and responsible 40% reduction of cholesterol and used for the development of medication for cholesterol. Some variants are specific. These are data from Charles Rotimi that shows that one variant that cannot be found in European and Asian monomorphic is associated with type 2 diabetes mellitus. This means that it has happened that this variant tomorrow is a target for therapeutics. For diabetes, it would never have been found if African population have not been investigated. If we even change the approaches and use rare variant association, study from South Africa shows that by investigating 1,000 people with African ancestry from co-ethnic group, they were able to found very, very rare, often deleterious, often de novo variant that are associated with susceptibility to schizophrenia. And those variants specifically target pathway of synapse. And this is important. We found the same in the study Michelle presented earlier on sickle cell disease. And actually, we wrote a piece of paper in the Nature Review Genetics last month, where we argued that actually this should be one approach to investigate complex threat. It's not only doing genome-wide association study, but looking for very rare variants that are easy target for therapeutics. Let's now move to single gene disorder after the complex threat. I think there a study that was long done by Michelle in South Africa that shows that if you investigate, for example, Huntington disease is one of the most dirtiest conditions in human genetics or medical genetics that is associated with degeneration of your function with age. If you test someone that have European and Asian ancestry, one single gene explains 100% of cases. You test someone with African ancestry, will explain only 70% of cases. So that means that genetic heterogeneity allows here to have a second gene that is not found in other population. Of course, this has implication for diagnostic, but it also have implication for the way we understand the pathobiology of this condition. I know there's some recent data from Amanda Cross that shows that maybe what we thought was linked to the multiple repetition and anticipation in this condition might not be the right mechanism. A very, very old study from Michelle, her group again, shows that in people with African ancestry, there is one specific variant that is more common for cystic fibrosis in Africa, but also in America. But I know that this specific variant is target for therapeutics now, specifically gene editing, to try to address some level of equity. Our own work on hearing impairment shows that GDB2 that explains 50% of children that are born deaf in Africa explain 0% in most children with African ancestry. And in this review, we found that only 30 out of 54 African countries have data, for example, of congenital hearing impairment. Let's imagine if we extend the study of all the 54 countries, that means that we will find much more variant and much more novel genes. The third reason we need to investigate, first reason of ancestry. The second reason is ecology. So if you look at the African continent, of course, have a lot of diversity because of migration within Africa, the migration out of Africa, the migration back of Africa. The second, the variable level of environment that is dictated by the north-south axis of the African continent. And this variable level of environment is also associated with variable of food and variable level of infection, and all of it make a mix that allow our genome even be more have more variation than anyone else, in addition to the 300,000 years of human genome history that we have on the African continent. This paper was published last week in the annual review. And we know, for example, that malaria was selected, sickle cell was selected to resist to malaria. And there are very strong evidence that this, my cousin, Tutankhamun, actually had sickle cell disease. He had some specific osteonecrosis that you can only find in sickle cell disease. In this study, they found that he was definitively infected at least by malaria a few times in his life. And that means that if this is true, the mutation probably gets fixed in the population at least 7,000 years ago, which means that for 7,000 years, it's highly likely that the sickle cell itself have put some pressure on our genome in addition to the environment. The second that everybody knows here, this is actually another H3Africa study that shows that APOL1 was selected against trypanosome. And this is one thing that we need to investigate. I think it's a hypothesis that we put out there in the literature. We did one heat map on all those variants that are selected in the African continent. We selected sickle, alpha thalassemia, G6PD, and APOL1, and on the side, in a separate study, piezo1. As you look at the heat map here, they all match the same region. That means that me standing here may have all the 4 variants. We know that each variant individually contribute to one specific disease, but if you put all the 4 together, what does that happen? What will happen to the person? So the epistasis of this variant and relation to the environment is critical to study. We may do this, for example, with African living in Africa, but also with African that moved recently in America. So living in a completely different environment with those that have been living there for 500 years, for example. And that is a completely new design that we haven't even yet started to look at. I remember in the H3Africa paper, the one we all published last year or so, there were at least 60 novel variants subjected to natural selection that have not yet been explored. So African genetic diversity is dictated by both ancestry, by both environment, by both 300 years of history, by both migration within the continent. That's the reason we are all here. So let's talk about equity. I like this map a lot. I would like to see it change in 10 years time, maybe less if possible. So 15% of people living on Earth live in Africa. We apparently are poor, 5% of the world domestic product, but I keep always saying that we are not poor. We are just being stolen to some extent, because 30% of the world resources are within Africa in terms of minerals. So there's something that needs to be corrected there. And then our investment in research is definitively very little. That means that we spoke about the capacity we have to explore our genome or in terms of research. If we do not invest, we are not going to be able to do it, even if we have already the competitive advantage of living on the continent. This was the map some 10 years ago on how research happened. I think this map had been corrected by H3Africa to some extent. You could only do this type of research if you connect with someone in Europe or in Asia or in America. And this again, actually I presented this data the first time when we had the meeting in 2009 in Yaoundé. Again, in that paper, it was easy to show that the study on human genetics using Cameroonian DNA was more interested of population genetic migration and variation than for disease that really matter in terms of genetic medicine implementation. Once again, I think for the past 10 years, H3Africa have changed this map. Of course, it is important that we interrogate what we should return and how we should return and what are the impact on genetic in terms of stigma. And that's what I believe H3Africa have done. This is some of the data we all publish. I will not speak to it. We know that we confirm that there are novel variants that are not present in the reference genome. We also developed this array that was spoken to before, in addition to the study presented by Michelle. These are numerous other studies that was reported from all of us, most of us using H3Africa chip. And we hope that this study will help alleviate one of the most common conditions on the African continent. And for example, if we want to benefit from gene editing in sickle cell disease, we need to change our public health offering. For example, there's only 3 African countries, actually only 2, that are able to do bone marrow transplantation. That means only 2 that can be actually now able to benefit to the possibility of gene editing. This is also a reason why probably we need to invest on technology. If we use gene editing the way we use it now, which is ex vivo editing, it will not be sustainable because you need to create capacity to do bone marrow transplantation. We will need to work together with the global community to do in vivo gene editing because of this map. Maybe to put some information on some of the things that Michelle spoke to, this is some of the data that we had on fetal hemoglobin using H3Africa chip. First of all, the phenotyping is good because we replicated BCL11 very well. We replicated MYB, but we also found a very novel variant. Some of it are obvious. For example, the KLF3 is a clear gene that is on the pathway of fetal hemoglobin. But one thing that we will have to handle to some extent, by using different imputation panel, we are finding different results. Actually, we use all the available imputation panel. We use the H3Africa imputation panel. We actually also design an in-house imputation panel using genome from this population. So the complexity of our genome and maybe the non-availability of a proper imputation that is relevant to the type of population that we are investigating will need to be one of the area where we need to invest in the next 10 years. Again, this is some of the data from our group showing the contribution of our study from various parts of the continent to the global knowledge of hearing impairment. And specifically, and this is very important paper because it's the first proof of concept that for those diseases that have genetic heterogeneity, there is a room for gene discovery in Africa. And in this study, we reported 7 novel genes, including this gene PAX8 associated with Waardenburg syndrome. And I'm highlighting this one because we already have a mice model, and the mice model is deaf. We have a second candidate for which there was a mice, and the mice was deaf. One of the candidates do not have a mice autolog, so we are still struggling to know how we're going to prove functionality. For the 4 other candidates, we are now currently developing a mice model, 1 knockout and 4 knocking. So we will be able to show you the data from those mice model in the few not probably months, they will take at least 1 year to come. So I think probably this is, maybe I'll go back to this slide. This is important because this will change our diagnostic offering, but at the same time, it will bring a global contribution to the pathobiology of hearing impairment for everyone else. For example, the mice are now being developed in the lab of Karen Steele in London. It's important because she's probably one of the most recognized mouse geneticists on hearing impairment. And I think I've never seen her more excited than that with this data. And we have such data from at least 7 different African countries. What we will have to work with, this is the bank that is managed by Alashley in Nigeria. And Moses Joroba in Uganda, or Elizabeth in South Africa. So we have all these data available now that probably will allow us to investigate some of the challenges we rose before us. These are some of the faces of people that will be able to carry the work forward. I think those are the faces from my own group. Some of them are doing their independent career. 4 of them are at the NIH. We probably sometime we need to find a way to continue working with them. Another important element of the contribution of what we are doing here, at least for those that are generated genome and exome, we'll have to feed into this database to make it more representative, so that when we infer pathogenicity is truly representative of what the global genetic community wants to do. For example, the one we all use is GenomeMat, and only 10% of African variant is present in GenomeMat. And we know that one African have 3 times more variant than the next non-African. So that means that in principle, proportionally, we should have had at least 3 times more African to make it more representative of the human genome. So this is the house that Dr. Francis Collins proposed some years ago of what is the base, what are the next element to be built after the Human Genome Project. I do think that the data that we presented now and that some of our colleagues are working on clearly indicate that the basis of this house, which is the Human Genome Project, still have a lot of cracks. We have to fill those cracks, otherwise we will not be able to build all the layer on the top of that. African genomic variation investigation is a scientific imperative before being an equity imperative. Equity is not new. The person in the yellow circle is me, 20 years younger, and that was in Bar Harbor in 2003. And this course was organized by the man in front, Dr. McChrysik. I can see Dr. Valley on the side of him holding the panel. Yes. And I remember that year already in 2003, Dr. McChrysik presented this slide where he already anticipated 20 years ago that the question of equity would be one of the more pressing challenges to be addressed for the years to come. And but I believe that probably he didn't anticipate that that would be actually be a scientific imperative. So this is what we proposed last year, that we need to sequence at least 3 million African genome in order to address that missing link that we don't yet have now. But of course, it doesn't need to be any African. Because if you sequence a Bantu from Yaoundé, it's not exactly like a Bantu from South Africa. Bantu from South Africa doesn't have sickle cell disease, and Bantu from Yaoundé has sickle cell disease. It has to be aligned along ethno-linguistic, along geography, along population movement, along what we know on the history of Africa at the moment. For example, someone, Nubian guy, for me, from Cameroon, it's not exactly like the same Nubian guy like Muntasa Ibrahim in the room. I'm not sure, but we need to test that. And to make sure that we really cover the whole continent in a certain way. This also have an important element. I think bioeconomy around the genome, we need to be prepared to take it on. If we don't, we're going to miss the next revolution of bioeconomy. This is an evaluation of the American Society of Human Genetics that shows that for every $1 that you invest in genetics, you will recover $4 more. So as we are thinking about it, let's also think about what goes with the economy of genomics as we do it. So that was my last slide. I would like to thank the Wellcome Trust, the NIH in particular. I think they have been so generous with my work. Please keep doing it so that we continue doing it. Thank you very much.
Thanks, everyone. Thanks to all the speakers who I'm going to ask to now come and sit at the panel. For some discussion, we're able to have a discussion until about 15 minutes past the hour. And I'm also going to call on Babatunde Salako to come and moderate that discussion. And straight after this discussion at 15 minutes past 1, we're going to have to go to the fountain as quick as we can to have the photograph taken, and then back from lunch at 2:15. Thanks.
Thank you very much. Please settle down. I believe we've had a very fruitful and interesting discussions. Unfortunately, we don't have much time. I know there will be questions, but let me just start off by asking the panelists, because we've been talking about sustainability, we've been talking about how to take this forward. Many of the projects that have been presented really, one way or the other, has a way of showing significant contributions to science, to the genomics of complex diseases. But in Africa, we do have a burden of some of these diseases that affects a lot of people. How do we engage the decision makers, the governments? What straight contributions can some of these projects that you have discussed has that we can actually put in simple language that they can understand, coming from the translational nature of some of them, especially that we are looking at Africa not being part of most of the genes used to determine some therapeutic or diagnostic procedures or devices in the past. And now that we are talking about the diversity of African genome and what we have done, what impact has this really made? And how do we translate that as an information to our government? Anyone who wants to start with that? Okay, Dr. Edu.
I'll start. I'm not sure the microphone is working. I mean, I think that as we begin to understand the consequences of these genomic abnormalities, we can begin to look at treatments. So as you will know, because you're a nephrologist like me, there are now molecules, drugs that will slow down lipoprotein L1 variant mediated kidney damage. And so this is 10 years is taken from discovering the variants to actually finding treatment. And so hopefully we should be able to persuade our governments that this is something worth investing in. And I think that we're not far off. Hopefully H3 kidney will be taking part in some of these drug trials to try and bring at least amelioration of kidney disease across sub-Saharan Africa.
Mayo, you were talking about vegetables.
Yes, thank you very much. I think already from the environmental determinants of some of these diseases, there are some precision medicine solutions that are already coming up for the control of cardiovascular diseases on the continent. I think, for instance, using hypertension as an example, one of the key things we found, and this was across the continent, was that obesity doubles the chances of having hypertension. Ordinarily speaking, we would think maybe obesity is not a problem in Africa, maybe poverty and perhaps even malnutrition, although obesity is also one type of malnutrition is a problem. But identifying that and targeting that could be one of the ways of reducing the overwhelming burden of hypertension on the continent. The other very interesting thing that we found, initially first only in Syria, but later on in the SHARE Consortium, was the effect of regular consumption of green leafy vegetables against cardiovascular diseases. We were actually pleasantly surprised because we checked so many other things. We checked fish, we checked fruits. They didn't have that kind of effect. And we've been able to replicate this and be able to actually put some value to it. For instance, consumption of 12 servings of vegetable every week clearly protected against developing hypertension. So, and these are some key messages that we could already inform the public about, inform government about, that if we really deal with our diet, we might be able to control risk factors for these cardiovascular diseases. And some of these we've already tried to disseminate in social media using video, from video cartoons based on scientific evidence that we've already unearthed from the SHARE resource. And we'll continue to look at this. We're looking at the relationship, the socioeconomic and diet and lifestyle determinants of other diseases like obesity, like dyslipidemia, like diabetes within the SHARE, and we're coming up with very interesting and actionable, actually immediately actionable findings. The genomic ones are going to come later, and they're going to complement what we are doing, so that we're actually looking with both lenses, not just the environmental aspect, but also the genomic aspect to enhance whatever we're doing. Polygenic risk scores, which may come through cardinal and through other means, other ways. For polygenic risk scores, for instance, for stroke, the ones developed in using other populations, we found that they are not effective in Africa. So we really need to do this ourselves, and because Africa is a completely unique population. So combining the genomic aspect with the environmental aspect will come up with solutions that are transformative. Thank you very much.
Thank you very much. Any comment, Michelle?
Yeah, thank you. I think you asked did you say one sentence? How do we take this?
And I say in a simple way.
In a simple way.
They will understand. We've been talking to ourselves,
yes,
and they don't understand that kind of language.
Yeah, so to me, what I think the simplest I would say is, yes, we've gained knowledge, and the knowledge is power. And from that, we can build on the other aspects step by step. I think generating knowledge, generating data is one important step, and it's the most important step. And then the others will come on using that knowledge and information. So simply I can say we've generated information or data or knowledge that we can share. Like he's saying, there is knowledge on, for example, BEST and the associated conditions. We can build on that and other aspects as well. So briefly, that's what I can say. Thank you.
Yeah, thank you. Michelle, anyway.
Thank you very much. I think.
Oh, we're coming back. I just want them to. We have a few minutes. Yes, please
That's
go ahead.
all right. Thank you very much. So I think, the simple message to governments is that complex diseases have both a genetic and an environmental component. And we can't change our genetic risk, but we can change our environment, which comes back to some of the issues that have been discussed by Mayowa. So I think, we need to stress the fact that we need to study, bigger cohorts. We need more data to understand the genetic risk, and then also to understand gene environment interactions, because I think those present opportunities for intervention. If you can stratify populations genetically according to those with higher risk, given a certain environment, that gives us a place to start to improve health. So I think the message should be more support, bigger cohorts to understand better so that we can come up with interventions for the future.
Thank you. Ambrose, any reflections?
Okay, I think the picture speaking at the end that can summarize everything. So I think there are 4 things that have to be happening at the same time. The first thing is educational training. We don't have enough geneticists on the continent. And this is at all level. Geneticist that does basic science, genetic counselor, and medical geneticists. So that we have to continue doing it, collaboration within the continent if possible, with other centers that are willing to do so. The second, of course, is research. We are trying to gather that. The third is advocacy. Probably we need to strategize better on how we get them the message across. We may want to have champions. Those champions might be patients. Those champions might be a politician. Those champions might be the media, and the fourth is technology. Probably we don't necessarily need to think that we need to follow the path of technology the way it's been implemented in genetics. We have to be creative. There are young minds here that will create a new way of analyzing. Probably we have to team with our engineering faculty to see how some of these technology can be applied, for example, for implementation as quick as possible. And that those technology being adapted to us. For example, we are running behind polygenic risk score for complex threat, but is it really the way to go? Because up to now, I haven't seen a single clinic in the world that uses it. So maybe we may want to think very, very differently, taking our specific environment. For example, I think I was having a conversation with Dr. Wandari yesterday night on COVID-19 susceptibility in Africa that happened to be extremely low. And he had an hypothesis that I found, I should have been speaking for him. Are you in the room, Gordon? Now, I had an hypothesis that for me very fascinating. He was speaking to the cytokine storm and malaria. And apparently, we all have had, living in Africa, those cytokine storm because of multiple infection of malaria, and train our body not to react anymore. And if when COVID arrived, we couldn't do it anymore. We already was trained not to do it. But those bodies in Africa that was not trained with malaria, for example, like Black South African out of the malaria zone, have much more higher COVID severity than us, and he has some data for that. So this is local knowledge and local technology, and we have to be creative and not necessarily follow the trend.
Thank you. I think we can take just 2 more questions, and I'll take Lashley and Shego. Okay, Charles is here. You're supposed to be the chair. So we're going to take you 3 people. Can we start from Malash? Oh, go ahead.
But we have to be holistic in the way we present it, because otherwise it may backlash. So you can't eat pounded yam 3 times a day and eat 12 vegetables and not exercise and have it work. And maybe that's the kind of backlash you'll have, right? Because you want to simplify the message and you don't make it holistic. So I think that's one additional thing that really putting out that information, that we try and be holistic in terms of the benefits of vegetables and environment.
Thank you, Shego.
It's just a comment.
My question also relates to Chair, and it goes to Professor Mayo and Michelle Ramsey. I can guess what would be the results when you do like a genome study of a range of cardiometabolic disease in Africa, probably next year or 2 years' time. Your conclusion will probably be that we've done GWAS of 50,000 people in Africa, but we need more GWAS to be done. That will probably be the conclusion. But currently in Africa, there are even more cohorts that have phenotype of CVD in Africa. Are you thinking about inclusive of all these other cohorts? An example is Adam cohorts that have been in a system for many years. There are many other cohorts that I know that we can actually think about something that is minimum of 100,000 samples across Africa. I probably mentioned this before, 2 years ago, but I think we should think about true collaboration, which is Pan-Africa, where we can be inclusive of all the study in Africa, and we can look at something that is probably much more bigger than what we have now. In the example that you give example of doing GWAS of lipid trade in 1 million people in European population, but we are now talking about 50,000 people. I think we should think about how we can expand that. Probably I can give you a name. You can have Share++ that can accommodate more cohorts across Africa. So that's the first question. Second question goes to Joseph. We'll talk about giving back genomic results in Uganda and also probably other part of Africa. I don't know, in the regulation that you are talking about, have you thought about when we give out like a genetic result to a particular participant and the implication that that result may have for family members? For example, if somebody have a particular genetic problem and what that might mean for their sister or for their brother or for a child. So in that kind of circumstance, you know, you say you're going to be the chair for this regulatory body in Uganda. So what should we be thinking about in that kind of circumstance? Are we supposed to be giving out genetic results to that participant? Because we know that it might have implication for other family member of that participant.
Thank you. We have someone online who wants to make a comment. Can you bring him on?
Hello, everyone. Thank you very much. This is Julie Makani from Wimbili University in Tanzania. I'm speaking from Johannesburg in South Africa. I just wanted to say how it's just been fantastic listening to the discussion. I wanted to focus on the way forward in terms of engagement and sustainability. And I wanted to share 3 experiences that we can build on as H3 Africa. The first is engagement with governments. There's been a lot of discussion that government hasn't invested in genomics, but I'd like to give examples, and these you can see in almost all African countries that are part of H3Africa, how much the governments have invested in universities, centers of excellence, as well as hospitals. If you go to Ghana, you will really see state-of-the-art hospitals. If you go to Uganda, you will see state-of-the-art centers of excellence. So I think we need to explore amongst ourselves if we are actually utilizing the resources that have been built by our governments, and we can leverage that. The second comment I wanted to make was with regards to engagement with global agencies. And here we're talking about the World Health Organization, WHO, the World Bank, which finances it, finances a lot of, for instance, the African Centers of Excellence, and organizations such as the World Economic Forum. Again, the WHO have really been responsive, and I think a lot of people from H3Africa participated in the WHO Science Council workshop series that was organized in November of last year. We had people like Daniel Masiga from Ian Bit, Fumi, a lot of us participated in those WHO Science Councils, demonstrating that there is interest and there is opportunity for H3Africa to organize ourselves so that we can engage with the WHO Science Council. So it becomes easier for us to have both a top-down approach from WHO downwards and a bottom-up approach when we engage with our governments. The same thing applies to the World Economic Forum and the World Bank, and we can share the experiences that we're trying to explore within sickle cell disease in terms of gene therapy. The third thing relates to the comments that Charles Rotimi made yesterday, as well as some of the people who've talked about this, is that how do we engage philanthropy and high net worth individuals or organizations so that they can invest in this? And a lot of the times we talk about Bill Gates and the Gates Foundation, and I think that's really good. But if you look at the experience that, for instance, Chris Happy had with regards to mobilizing resources for, I'll just turn off my video because my internet connection is not stable. So when you look at what Chris Happy was able to mobilize with the COVID-19 response, he was having discussions with foundations such as the Gates Foundation for maybe $5 million. And yet he was able to mobilize over $100 million from other philanthropic organizations. So I think we need to think beyond the usual as in with non-communicable diseases. And here Plus initiative, we've been able to get $1 million over 3 years for 5 countries to invest in NCDs, Pen Plus, working with the WHO Afro. So I think if we target, and this is from high net worth individuals and high net worth organizations. So I think if we're able to, as Amber said, identify champions, identify heads of state, we're able to go and mobilize resources at different levels within the African Union, Africa CDC. I think there is a lot of optimism with regards to how we can take the knowledge that we've built up in the past 10 years and translate this to have an impact on patients, populations, and research that we're doing in Africa. Thank you very much.
Thank you very much, Julie, for those reflections. Charles?
Yeah, again, thanks. Unfortunately, I had to step away this morning. But I just wanted to ask Jomoa, in respect to the kidney disease and the I want to say potential therapy in terms of the APOL1 effect. I wonder if this is an opportunity for us because it's such a unique story in terms of how the African environment shaped our genome, in terms of protecting us initially, but now putting us at risk, in terms of kidney disease. Do we know individuals, for example, in Ghana or Nigeria, who are high income, deep pocket type individuals who may have suffered the consequences of this carrying this 2 copies of this variant that we could go through to say we have studied this, we know how it works, and there's a potential therapy. We want you to be part of our voice or even a source of resources to do some of these clinical trials. I wonder if you know of such individuals and if you think they are approachable. I think maybe a very good story that we can tell.
I mean, Charles, thank you. That's really, really very important. And I must say, the last decade, we haven't really thought along those lines. So there are high net worth individuals in both Ghana and Nigeria who suffered from kidney failure, but I had never given that a thought. So we'll take that away from this 10th meeting, and it's something that we can definitely develop. And I know Prof. Salako will also know such individuals. So it was not anything that we've actually thought about developing, but we can do.
We'll try. I'll follow Ashley too.
Okay.
Mayo, you have
respond.
Quickly.
Okay, thank you very much. For the green leafy vegetable story,
don't take too long.
No, I'm not going to take long. What we presented were actually not based on just unadjusted odds ratio. These were adjusted for other risk factors, including other dietary risk factors and including exercise. And in fact, because there was no time to add all the slides, we had a digital abstract of a publication in International Journal of Stroke, which showed how other dietary factors influence the risk of stroke. And in our video, summary, which we have disseminated on YouTube on our findings, we also talked about other risk factors. So it's not in isolation, really, the green leafy vegetable story. Then in terms of inclusion, definitely, what we have in share, definitely we would like to expand it, and we would like to collaborate with others to have more power, to discover more, and to have more reach and more impact. So thank you very much.
Thank you very much. I believe what is left for me is to ask you to join me in appreciating the panelists.
Can we please go to the fountain area for the group photo, and then after that for lunch, and we'll have to reconvene at 2:15. So we need to do a quick job of it. Thanks.
The Institute of Human Virology Nigeria, IHVN, is a non-governmental organization that focuses on HIV/AIDS related problems in Nigeria. It was established as an affiliate to the Institute of Human Virology, University of Maryland School of Medicine, Baltimore in 2004. In 2015, International Research Center of Excellence, ISA, was established with Professor Abimiku as its executive director to promote collaborative research in diseases of relevance to the local community, non-communicable diseases, and emerging infectious diseases. IHVN moved to a 7-story, 2-tower edifice to support research activities in ISA. The Institute of Human Virology Nigeria, H3Africa, IHAB Biorepository Initiative project is directed by Professor Alashley Abimiku. IHAB is the West African Regional Biorepository for the Human Hereditary and Health in Africa Consortium. Established in 2009, IHAB supports the H3Africa Consortium network of African scientists by providing high-quality, well-annotated biospecimen to study genomics and environmental determinants of common diseases with the goal of improving the health of African populations. IHAB collects, processes, stores, and distributes biospecimens within Africa and beyond. IHAB receives both processed and unprocessed samples from researchers within and outside the country. Biospecimens include whole blood, urine,
saliva, stool, meconium, DBS, cervical swabs, nasopharyngeal swabs, breast milk, and so on. With over 150,000 collections, iHab utilizes robust FreezerWorks 2019 LIMS system to automate biospecimen, data, and freezer management in a secured system. Facility is well equipped, conducts sample processing for research projects from plasma, sera, DBS, DNA extraction and QC, cell characterization and PBMC isolation. Peripheral blood mononuclear cells, PBMCs, are made up of lymphocytes, which are B cells, T cells, NK cells, monocytes, and dendritic cells. iHab supports both manual and automated viable cell counting. The TC20 automated cell counter accurately counts cells in one simple step using an innovative autofocus technology and sophisticated cell counting algorithm. The TC20 enables accurate mammalian cell counting in less than 30 seconds. The BD Fax Caliber offers a unique modular approach to flow cytometry and allows users to perform both cell analysis and sorting in an innovative single benchtop system. iHab supports both manual and automated DNA extraction. The QIAGEN QIAcube HT enables automated mid to high throughput nucleic acid purification in 96-well format using silica membrane technology. The system provides reliable DNA, RNA, mRNA extraction virtually from any sample type. The user-friendly software allows for easy data management and documentation. It is convenient, flexible, safe, and minimizes cross-contamination. The NanoDrop 8000 allows scientists to quickly and easily quantify and assess purity of samples such as proteins and nucleic acids using an 8-channel pipette to dispense samples onto an array of pedestals to analyze 96 samples in less than 6 minutes. The qubit fluorometer quickly and specifically quantifies DNA, RNA, and protein. It is easier than ever to determine if you have sufficient nucleic acid or protein for your experiment. Qubit assay dye binds specifically to DNA, RNA, or protein, making it more specific than UV absorbance. eCube is an all-in-one DNA, RNA electrophoresis device, including gel electrophoresis chamber, LED illumination, live display, documentation by integrated digital camera, and software for image capture. Biospecimens are stored at -40 C, -80 C, and in liquid nitrogen tanks. iHab uses a combination of manual temperature chart and Tutela automated monitoring system to monitor the temperature of the storage unit. Tutela provides 24/7 monitoring status alerting system via phone call, SMS messages, emails to responders to track temperature excursion. IHVN has the capacity to produce liquid nitrogen. The hallway showcased the abstracts written by iHab staff, and the office is equipped with a conference room. iHab is a secured facility with CCTV camera, monitors, and trained security guards.
The African Neurobiobank for Precision Stroke Medicine project, particularly known as the ELSI project, seeks to identify, examine, and develop novel interventions to address the ethical, legal, and social implications of stroke neurobiobanking in sub-Saharan Africa. The project also aims at building capacities and careers related to genomics and biobanking, especially around issues relating to genetic counseling, biobanking, and regulations. Neurobiobanking is the storage of the human brain and associated tissues donated by individuals after death for the purpose of research. Neurobiobanking, although new to Africa, can in principle revolutionize the management of stroke, a cardiovascular disease of public health importance, of which Africans share one of the highest global burdens.
The ELSI project is a 4-year project which began in 2018 and is domiciled in 7 sites in Nigeria and Ghana with over 60 community advisory board members comprising stroke survivors, community leaders, religious leaders, and representatives from various sectors, including the public health sector, the legal sector, and the ethical review boards. Based on the project's objective to identify the ethical, legal, and social issues relating to neurobiobanking, a community-based participatory study was conducted using a mixed methodology to determine the perception of Nigerians and Ghanaians towards the donation of blood and brain tissue for genomics research and neurobiobanking.
While most people expressed a willingness to provide blood sample fractions for genetic research and biobanking, brain donation was a less familiar concept and fewer people were willing to donate brain samples.
The key findings from this study showed that across all groups, knowledge of concepts ranged from no prior knowledge to robust awareness.
project.
Hello? 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 Is it on? Oh, sorry. Hi, everyone. Welcome from a wonderful, wonderful lunch and having some air outside. We're running late. It's no news anymore, but what is news is that we're going to catch up during this session. So it really gives me a tremendous pleasure. Please settle down. Please settle down. It gives me a tremendous pleasure to welcome Dr. Judith Amumbo. She's going to talk to us about the Science for Africa Foundation. Please, Julie, Dr. I hope her slides are ready to load.
So as we wait for our slides, good afternoon, colleagues. I would like to just give you a little update on what's been happening in the sphere of the African science ecosystem. And for those of you who are wondering, who is this new kid on the block, the Science for Africa Foundation? Are we all here? I agree we did have a good lunch, perhaps maybe too good. So I'm glad that we're running and catching up time so that I can finish quickly and sit down. So my name is Judy Omumbo. I'm based in Nairobi, Kenya at the Science for Africa Foundation. Now, for those of you who's wondering what the SFA Foundation is, the acronym is SFA Foundation with the foundation, because apparently there are SFAs all over the internet. So distinguish. It's basically the programs that were formerly at the African Academy of Sciences have moved into a new entity called the Science for Africa Foundation. Now, this is wonderful news for us. It's the same team that were working on the programs before at the African Academy of Sciences. But now in our new entity, we're able to expand what we want to do and really work on the things that we have seen as an organization are very important for the ecosystem of science in Africa to work out. Now, that's not my presentation. And I'm filling in time. Yeah, who offered to sing once? I thought I could come and stand up and sing with her. This is another singing opportunity. Any sign of the slides? Celine is here. Yes, I did share them a while back. Let me just check with them.
We can continue after Dr. Amumba has made her presentation. The first STEM fellows, Grace, Anja, Labia, Harriet, Nankia, forgive me, please, Henrietta, Munye, Isaac Emmanuel Omara, Mamadou, Sangare, Maria Magdalene, Nana Ganda, Melaku Tilahun, Modibo Waiter, Nana Achia, Nicholas Okoko. Those are the first trainees with the speed presentation as soon as Dr. Omombo is done. Do we have her slides now? Welcome. Oh, you didn't have enough seated place? Can somebody just help us with one more chair? Can you grab a chair for yourself? Just one. Oh, two. I would have given you mine, but I need it. Oh, there you go. Thank you for being a gentleman. Dr. Oman, I think it's coming up.
Is it coming up? I've just been challenged actually, you know, to be able to talk about my organization. I can talk about my organization without the slides. Should that be required? I still see the previous slide on. So the Science for Africa Foundation is founded on the belief mission that Africa's solutions for development need to be generated and built at the point where the challenges are born. So therefore, needs to lead her agenda for science and her agenda for development. So at the Academy, at the Science for Africa Foundation, we have 3 mandates, or we can call them sort of areas of strength. Firstly, we're a science funding organization. We mobilize funds to invest in science in Africa. Secondly, we want to establish ourselves as an organization that sets the agenda for science first for ourselves. Now, this is it's a huge challenge. There's so many things to do, so we have to prioritize. And thirdly, and what our partnership here with the H3Africa is around is developing programs and implementing them so that we can meet some of the things that we have put down in the agenda. Now, if I could challenge you to just walk with me a little bit and think about, you're standing up, looking down at the earth and looking down at our continent. And there's a whole ecosystem about how things work. And as we understand an ecosystem, the energy is coming to that system. That's really funding for science and so on. How that energy is used, how it's distributed, how the different organs in this ecosystem are nurtured, cared for. They pass from one stage to the next. And indeed, as they grow older, they feed the younger ones so that that ecosystem is and it works. And this is the only way that the energy is used to sustain that ecosystem. The talk has changed from what it was. But we're looking at what is it that the African ecosystem of science needs to make sure that science can be practiced and the quality of the science can remain the highest, and that those who begin as scientists pass through that system, are mentored and nurtured to become the leaders and those who guide the direction of science, and ensure that the next generation is able to take that from there and carry the science on further. So that's really what we're all about. It's okay. So within that ecosystem, there's a whole value chain of science, from discovery, from its development, and then it's from its implementation. So we are really interested in making sure that all those stages can be practiced. We're very, very much conscious of the issues around equity. And without us addressing the need for Africans to be at the forefront of providing that science, it'll never be equitable. So this is what networks that are being built through the H3Africa program, because they're networks between African institutions. This is really a game changer for this continent. And this team here needs to be congratulated very much. We're also, as we strengthen, we aim to work with other institutions that manage science on the continent. We aim to strengthen our institution as well, to make sure that we're ready with proper systems for managing grants, for managing finances, for compliance and all those sorts of things. And we have a new office on Riverside Drive in Nairobi. Whenever you're there, please do come and look for us. We have a huge sign on the wall. And as Africa scientists, just knock on the door and do come in. We can even arrange a place to stay. So just quickly to look at our strategy. This is a lot easier than this. Am I going backwards? Oh, dear. It's stuck. It's stuck on this one in both directions. Right, so we take a holistic approach to science, okay? So as I mentioned before, we have initiatives across the entire value chain. We're looking at genomics, we're studying R&D, consortia, working in consortia, a lot of work around clinical research and trials, public engagement and innovations in science. Okay. I don't know why it's just going around in circles in the same slide. Yeah. Try. So just quickly, we've mentioned the programs that we have in place. H3Africa, which you're all familiar with. The Deltas Africa program is around building centers of excellence around the continent. We have a spoke and a hub and spoke model for developing centers of excellence that also focus on capacity building, building excellence in research, building linkages with other research institutions. We have a program around priorities. We were able, during the COVID pandemic, to convene scientists to identify priorities for COVID and design research programs and provide some funding for some of that work to be done. The Grand Challenges Africa program, which is around science and technology innovations. The Research Management Program in Africa, REMPRO, which is around strengthening institutions so that wherever a fellow is based in the institution, by the time they finish with that grant, the systems and processes for managing grants in that institution, we hope will become improved. And as an organization, we're going to in the direction of open publishing. So we have an Open Research Africa publishing platform. So I'm not going to say too much about this because you definitely know a lot more about it than perhaps I do. But those are the centers with a 30 million grant from Wellcome that the Science for Africa Foundation are managing. The Deltas Africa theory, as I mentioned, this is the theory of change for the Deltas program with outcomes in the 4 areas of scientific quality, improved research training, scientific citizenship, research management, and environment. So we're really keen on scientific citizenship. We really believe that scientists need to be the ones who make sure that the work in their own field remains at the highest standards. So just simple things, which are sometimes ignored: peer review processes, reviewing papers, sitting at meetings, listening to other scientists, mentorship, all those kinds of training will fit under that area. I'll leave that up just for a second so that we have a quick look. And these the first Deltas 1 from between 2015 and 2022 had 11 different sites across the continent, as you can see, spread out, just working in different areas of health research, including centers that are looking at genomics and bioinformatics. So these are networking 26 African countries already, 65 institutions, 11 PIs, and over 2,000 scientists trained from these centers. We have this Deltas is funded by Wellcome. We have a Deltas 2 a round of funding, which was currently being awarded, which will last until 2026. These are 14 different areas for 14 different PIs. Now, Deltas 2, aside from healthcare, will also be looking at environmental factors, climate, and finally, we shall have social scientists actually being PIs, because this is, we research on people, but we don't include those who research on people. So I think this is a significant gap, and I'm glad that this is beginning to be addressed by Deltas 2. So this is a sort of the round flow of what happened when we did prioritizing for the COVID pandemic. Most of this was done online, but the ability to convene scientists is a great gift, and I think this is a potential that all of us who are now networked can build on. When something emerges, let us gather the African scientists together to say, what are you seeing in your region? What do you think about this? Rather than hearing, well, there was quite a lot of information coming out of COVID. None of it was really relevant for Africa. But now there are ways that we can make sure that it's really homegrown solutions for problems as they face us on our continent. The Grand Challenge Africa is funded by Gates, and it has a focus in discoveries in different areas, many discoveries that are being used now in different countries, in maternal, neonatal, and child health, drug discovery, food security and nutrition, water sanitation, and quite a lot of work in antimicrobial resistance. Happy to share some of that information with you, either by email or afterward. The Grand Challenge Africa program has two components: an award that you get at the beginning, and then we work with a couple of years on that award to develop an innovation, and then there are awards that are offered for transition to scale. Now, the research ecosystem management system looks at institutional leadership, sustainability, standards, and individual capacity of those who work in research offices. A research office is not the normal thing in many African institutions. And this really causes a lot of problems for those of you who have worked in these institutions. And you receive your funding and you have to pass it on to the institution, and 90% of it goes into overheads, and you're stuck. There's basically nothing you can do about it. And there is a real need for this training, a real need for empowerment in this area. And our research management program looks at that. And publishing. We need to be able to access what we have published on this continent. So the open research platform publishes anything from data, from new methodologies, from interesting findings to actual full manuscripts. So this is a sort of a go-to place for African scientists who you may have started to work on something, didn't find anything, but you've got some very useful data. This is where you can publish that. So we're looking at many things, as you can see, but overall, the programs at SFA are about supporting you to make your research better. And also an opportunity for you as scientists to reach out and say, in these areas, we think we would like some interventions. So as an organization, we're here for you. And having gone through the same systems ourselves to the point, all of that, you study in Africa and go abroad, and experiencing those gaps and the challenges, I think we're well placed to address some of them with your help. So thank you very much.
Thank you. Do you want to hold on for a minute? Thank you for dealing with all the challenges. So we have one question for Dr. Judith. I know she's around. Yes, please go ahead. Right there.
Hi, thank you for your presentation. Can you please explain the relationship, if any, with the African Academy of Sciences?
So the programs at SFA were incubated at the African Academy of Sciences, but with the support of our funders, we decided to move those programs out to give us the freedom and latitude to work in an umbrella that is more Pan-African. So it was just in that context.
So AAS is not Pan-African? Oh.
The AAS is Pan-African, but the AAS has its own missions and its own goals. And this is the opportunity for us to pursue, the vision as I elucidated earlier.
Thank you very much. Eloquent presentation. My name is Paul Mahanetungu, and I'm a researcher from the University of Yaoundé 1, but also holding a position in the Navajo Technical University. The question I have is, how many African funders, or let me put it this way, how much of the money that SFA is getting is African money coming from our African governments, please?
And thank you for that question. I think this is really important. For us to gain agency and access and ownership, we must find a way of encouraging our African governments to put money into science for the continent. So I think the onus is really on us. We are a new organization. This is very, very much on our plate. We view partnerships in many ways. We can partnership with money, but partnerships also with thought processes are very important. And this is how we see we can also begin to partner with African organizations so that together we can build a program with which we can say to African governments, we would like to work with you on this, and can you partner with us putting some money into it? So as you know, and that's why you're asking the question, it's very difficult. And I cannot say, I'm not squeezing out of the question and avoiding it. The fact is, African governments are not funding African science, but it's for us to understand how we need to reach out to them and say, hey, you need to pay attention to this.
Thank you. I'm afraid we have to move on, but we really look forward to working really closely with the Science for Africa Foundation, because based on some of the discussions that we've had, obviously having a voice like yours as Pan-African is really going to help some of the ideas that H3Africa has put together. So thank you so much for coming and sharing that. And one of the things that Dr. Judith told us in her presentation is how important it is for us all to ensure that we pass the baton to the younger ones and we support and coach them. So for the next few for the next few minutes, where's Rolanda? You're supposed, okay. So Rolanda very efficiently here is going to help me. And we are going to listen to our future PIs as they make a brief presentation. A part of the
Let's wait until they get the slides up. So the first speaker for today, we will have 11 fellows in this group 2. We will have Usman, who was supposed to present yesterday, but because of a technical glitch, we're giving him a second chance. So he will be up first. This is not his presentation. All right, so Usman. Take it away. So yes, Usman is going to present to us studying the nuclear organization of telomeres in urinary circulating cells in bladder cancer.
Can I start? Okay. Thank you. It is a pleasure for me to present my work, which is studying the nuclear organization of telomeres in urinary circulating cells in bladder cancer. The bladder slide, please. Okay. 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 The bladder cancer is the third cause of hospitalization and the first cause of death in the urology department at the main hospital in Mali. The poor prognosis is associated with genomic instability and telomeric dysfunction. However, there are not biomarkers. This is the reason we set ourselves an objective. Yes. Very light. Yes. Okay. The aim was to assess the nuclear organization of telomeres in urinary circulating cells of patients suffering from bladder cancer. Next. Next. We enrolled 10 patients. We did cytological preparation of urinary circulating cells. We did 3D FISH technique of telomeres. We did 3D microscopy and 3D telomere analysis. Finally, we did statistic analysis. Next. As you can see, the normal cells have small nucleus, fewer telomeres, dense chromatin, and regular outlines. In contrast, cancer cells have a large nucleus, more telomeres, irregular punctures, and large nucleus. Next. We evaluated 6 parameters, but you show you 2 parameters. Here, we measure the number of telomeric signals. The blue ones are shorter than the red ones. That means the cancer cells have more telomeres than normal cells. Next. And then we measure the index of telomeric nuclear distribution. We obtain that the telomere nuclear distribution index are higher in cancer cells than normal cells. In conclusion, next. We develop a new methodology assess the nuclear organization. To assess nuclear organization in bladder cancer, we found differences for the parameters defining telomere profile between normal cell and cancer cell. We show tendency, telomeric profile, and patient prognosis. As next step, we need to recruit more patients to confirm our funding. Thanks.
Thanks so much, Usman. You may have a seat. All right. Next. For all the speakers, we're suggesting that the projectionist advances your slides. Is that all in order? So if you need to move from one slide to the next, please say next slide. All right, thanks. So our next presenter, can we have a presentation up, please? Our next presenter is Grace Ajolar-Bigafa. Great. So Grace is going to present to us her work on participants and stakeholder views on feedback of genetics research findings of the Kidney Disease Research Network, Ghana. Thanks. Take it away, Grace.
So for almost a decade now, the H3Africa Kidney Disease Research Network has been investigating the genetic cause of kidney disease. And there's evidence that variants of the APOL1 gene increase the risk of developing kidney disease in Africans and people of African descent. And we also know that, next please, we also know that families of relatively chronic kidney disease may be at risk of developing chronic kidney disease. So the question is, should we tell these research participants, should we tell their family members that they are at risk of developing CKD? And if you have to tell them, what findings should we feedback? And then who does the feedback and how do you do it? So this research sought to find out from the participants, their families, ethics review members, and then the researchers on the subject. Next, please. So this research employed is purely qualitative, specifically Africa bioethics, and using focus group discussion and then individual in-depth interviews. So for this research, 20 research participants of the Kidney Disease Research Network were interviewed. 9 researchers were also interviewed, and then 2 focus group discussions for family members and 2 deliberative workshops. The data was statistically coded and analyzed thematically with the in vivo software. Next, please. Okay, for results on what to feedback, on whether to feedback, there was a consensus that we should feedback to the participants and then the community, as stated in the quote. And then on what to feedback, though participants preferred to receive their individual results, researchers and ethics review board members suggested feedback of aggregate results. And then on who to feedback, next please, who to feedback, participants prefer the researchers or their doctors to feedback, and they don't mind a genetic counselor being around, as stated in the quote. And then the sort of ways to feedback to the communities, various mediums such as the media, debates, and religious gatherings were suggested, as stated in the quote. Next, please. So in conclusions, there is ethical imperative to return validated, clinically relevant individual genetic research results to the Kidney Disease Research Network participants, their families, and then aggregate results to the communities. And we recommend that research institutions should make efforts to improve genetic literacy among research participants, healthcare providers, and community. just to demystify some of the ideas behind genetics and then to improve the understanding. And then to also train genetic counselors and healthcare providers to support the feedback process. Thank you very much. Next, I want to thank everybody who make this possible.
Thanks, Grace. So the next presenter we have is Harriet. Harriet will present to us the stakeholders' perspectives on community engagement for genomics research in Africa, a review of literature.
All right. Thank you so much. My name is Harriet Nancha, a PhD fellow on the Tripanogen project, and I'm looking at stakeholders' perspectives on community engagement in genomics research. And this is basically a review of literature. So community engagement is recognized as a facilitator in addressing a number of ethical and social issues that pertain to genomics research. However, the knowledge base on how to conduct this community engagement is limited, especially in the African context. So this study basically, first go back. This was to explore perspectives of key stakeholders, and these were genomics researchers, community stakeholders, and research regulators. And this was aimed at informing effective community engagement for genomics research. And in total, we reviewed 59 articles published between 2011 and 2022. Next. So community engagement is perceived to allow continuous interaction. And this allows informed consent because continuous interaction is a requirement for informed consent. And through the interactions, there's comprehension because of the sharing of the science. And this allows informed decision-making in participating in genomics research. Then it allows understanding of community norms, and in a way it informs the design of the study and also the acceptance of the study by the communities. It also shows the respect a researcher has to the community norms and community values. And it also gives a way of anticipating and mitigating of potential risks and misconceptions that may pertain to genomics research. Next. No, go back. So no, yeah, community engagement still is perceived to have challenges, especially in genomics research. And these include limited local and international guidelines on community engagement in genomics research. And usually when there are no guidelines, there's a resort to getting guidance from the research ethics committees. However, in Africa, there's limited technical ability by the RECs or research ethics committees to review genomic studies. Then the other issue is difficult in explaining genomics to communities, and also the resistance by communities due to the unfamiliar sampling procedures that pertain to genomics research, like the collection of large samples, the storage and sharing of data and sample are concerns mainly by these communities. Then there is the delay or never return of genomics findings to communities, and this portrays a lack of reciprocity for study participation. And also there's a difficulty in involving communities, especially under conception. Usually researchers go to communities when they want to collect data, so they miss the step of involving them at the start. So in conclusion, assessing community stakeholders' perspectives highlighted the strengths and weaknesses in fostering community engagement in the African context. So the implication of these findings is they will inform the designing of our research tools to collect empirical data using Uganda as a case study that will inform further the development of a framework for effective community engagement in genomics research in the African context. Thank you so much. I would like to thank my funders, the supervisors, and H3Africa Consortium.
Thanks, Harriet. So now we call on Henrietta. Henrietta will present to us the attitudes towards genetic testing and associated factors among adult eye clinic patients in Ibadan, Nigeria.
Thank you very much. Good afternoon, everyone. I'll be presenting this topic under the Eyes of Africa Genetics of Blindness study. And the rationale for presenting, next slide please, for carrying out this study stemmed from the fact that molecular diagnosis is becoming, next slide please, is becoming very important in evaluating patients with eye diseases. And also the fact that the Eyes of Africa and Genetics of Blindness study has provided a lot of opportunities as well as resources that can help support the establishment of ophthalmic genetic services in this setting. However, there is a paucity of data on patients' perceptions of genetic testing in this setting. So our aim was to evaluate the attitudes of adult eye clinic patients at the University College Hospital Ibadan, and we carried out a hospital-based cross-sectional study in all. Next slide, please. We recruited about 223 patients with a mean age of 49.4 years, and we found in our results that there was a high level of awareness of that some eye diseases had genetic basis. This was in 88% of participants. However, there was a low level of awareness and knowledge of genetic testing. So we found out only 9% of participants were aware of genetic testing, and of this, only 21% of patients knew really what genetic testing was. Next slide, please. In terms of reproductive choices, we found that 20% of participants were willing to undergo prenatal testing, while only 7% would consider pre-implantation diagnosis. Then for willingness to test and willingness to pay for genetic testing, we found that about 92% of patients were willing to undergo genetic testing if this could be carried out for free, but a lower proportion, 79% of participants, were willing to undergo genetic testing if they had to pay out of pocket for this testing. On the whole, about 48% of participants believed that genetic testing will be useful in this environment, and a majority of the remainder of participants were undecided as to whether or not this service could be useful. For the significant determinants that we found that tertiary education preferred a greater odds, while having other children preferred a lower odds. In conclusion, we found generally that there was a generally good awareness of genetic eye diseases, but that the knowledge and the awareness of genetic testing and attitudes also towards genetic testing was quite poor in the studied population. And so it's important to create awareness about the implications, the benefits, as well as the limitations of genetic testing, with consideration to patients' perceptions as well as their characteristics, in order to maximize uptake of this service when it's established. Thank you.
Thanks so much, Henrietta. So next we call on Isaac Emmanuel O'Mara. Isaac will present to us the genetic diversity of Bundibugyo Ebola virus from Uganda and the Democratic Republic of Congo. Thanks, Isaac.
Thank you. As already said, my name is Omar Isaac Emmanuel from Makerere University. Pleasure to be in Nigeria. These are my supervisors. So from this slide, it's quite compressed, but I'll pick out the main points. Yeah, so the genesis of Ebola virus dates back to the year 1976, when there were 2 consecutive outbreaks. That's in Zaire, currently the DRC, and then in Sudan. So from that map, these have been the hotspot countries since 1976. So there are 6 strains, known strains of Ebola virus, with the Zaire strain being the most lethal strain. And with the transmission, there is a new pattern that has been identified in how Ebola viruses spark off. That is following a paper that was published last year. If you see down here, in September last year, there were 2 outbreaks in the Republic of Guinea and in the DRC, whereby survivors are actually turning out to be reservoirs for Ebola viruses. Hold on. Please go back. Yeah, so in our study, we considered 2 local and scientific accounts of 2 past Ebola virus outbreaks. That's the Bundibugyo outbreak in Uganda when it first emerged. Then 5 years, there was re-emergence in DRC. So the question actually here is, was the 2012 outbreak in DRC a spillover event from nature, or it was through latent infection? We looked at that by identifying variants from these isolates across the 2 epidemics. Next slide. So these are preliminary findings. Generally, the Bundibugyo Ebola virus was genetically diverse, and we so far identified overall from the 2 epidemics, we identified 14,632 genomic variants from the 2 epidemics. We can see that the DRC outbreak had more variants, which was almost close, double the number from the variants from the Ugandan outbreak. And one interesting finding is from the Ugandan isolates, we identified the variants that caused high impacts on the genome, and these were mainly within the L gene region. Now the Elgin region is the most conserved region and is the target when designing PCR primers. So these findings, these were mainly frame shifts, and these findings further throws more light on the emergence of the Bundibujo in 2007, since it was a novel strain by then. So from the DRC isolates, more variants in nature, viruses undergo evolution, which can be due to environmental changes, host immune responses. So this suggests some sort of evolution that is after the 2007 outbreak in Uganda. And this is further evidence in the phylogenetic tree that we drew. The Ugandan sequence is clustered separately, close from and distant from the DRC clades, which forms a separate cluster. So at the moment, we're looking at determining the evolutionary rate to see whether the evolution during the 2012 outbreak lies within what is known for Ebola viruses, and this will further draw more conclusion on the 2012 outbreak in DRC. So in conclusion, I'd like to thank H3Africa for the funding that enabled me to enroll for my master's in Makerere University. And it's through the skills that enabled me to do this project. So basing on these preliminary findings, there is need to strengthen public health. And as H3Africa, can we develop new therapeutics to clear latent infection? Thank you.
Thanks so much, Isaac. Next, we have Mamadou Sangare. Mamadou will present to us the structural characterization of phosphate-glycerine decarboxylase as a promising therapeutic target of PF prediction of its potential inhibitors.
Thank you. Next slide. As you know, Plasmodium falciparum as an protozoan responsible for the most severe and deadly form of malaria. According to the last malaria reports, there were in 2019 227 million cases of malaria against 2,041,000 in 2020, and also 409,000 deaths in 2019 against 627,000 deaths in 2020. The statistics are alarming. The resistance of this parasite to last anti-malaria drug has been reported and warrants the situation, as you can see on the map. The discovery of new drug is a necessity. It requires the identification of new therapeutic targets. Our present study aims to propose the 3D structure of phosphatidylserine decarboxylase, which catalyzes the formation of phosphatidyletanolamine by decarboxylation of phosphatidylserine. These steps are crucial for parasite development and differentiation. Next slide. And to predict the potential inhibitors. To achieve this goal, we adopt the following methodology. The first one is to identify a protein of interest from TDR target database, to construct the model with AlphaFold, validate the model with ProCheck, identify binding pocket with Frank Web Server, virtual screening with Autodoc Bina and Zeng database, and ADMA evaluation with Swiss ADMA. This methodology allows us to obtain the following results. As you can see at right, this is the structure, 3D structure of phosphatidylserine decarboxylase. You can see in the binding pocket the ligand. And at right also, we have a validation step with Ramachandran plot. As you can see, most of our residue in dot black are in the red area, favorable zone. That means that our protein has good quality. Next slide, please. Here, in this table, we can see the result of virtual screening and ADMA evaluation. From more than 6,000 molecules, after virtual screening and ADMA evaluation, we select the top 10 molecules, which has lowest energy and most respect Lipinski rule of 5. Next. For conclusion, this study allows us to identify a phosphatidylserine decarboxylase as a promising new therapeutic target of Plasmodium falciparum, to predict its 3D structure model, identify its potential inhibitors. The next step of the study is to test experimentally the obtained inhibitors to evaluate their anti-malarial activities. Thank you.
Thanks so much, Mamadou. So next we have Maria Magdalene Namaganda. Maria will present to us the low abundance HIV-1 drug resistance mutations among patients experiencing virological failure at the time of therapy switching in Uganda.
Thank you. It is quite agreeable that since the emergency of HIV, antiretroviral therapy is one of success among the efforts to manage HIV. It's one of the medical advancements in the medical management of HIV. However, the efforts of antiretroviral therapy have been jeopardized by the emergency of drug-resistant variants that are present among the patients. If you look at this cartoon here, it's showing a leaking bucket. If you pay attention to only the big holes, if you pay attention to the big holes, the smaller holes will still continue to drain to contain the contents of the bucket. And that is what minority variants are in this case, which is the focus of the study. What we are looking at is has been neglected for a long time. These minority variants are usually present in less than 20% of the viral cache species and have been reported to have an impact on drug resistance. So if we do not focus on them and miss them out, we are going to continue having the burden, which is represented by the leaking bucket, and as such elevates drug resistance among the patients. To perform this work, we did total viral RNA extraction, performed sequencing on the MiSeq as pictured, as a technique called next generation sequencing, which is a high throughput. It is able to give us many reads that are able to give us the variants that we're looking at in the variants. And then we perform data analysis using the Hydra pipeline, accompanied by the Stanford database to annotate the mutations that were in these samples. Overall, we found 757 low abundance variants in the samples, where K65R was the most prevalent, and the others are the 10 selected mutations that were present. All these are low abundance variants. The interesting bit about this work is most of these variants cannot be detected using Sanger sequencing, which is a gold standard, and as such are only able to be detected by next generation sequencing because it's high throughput. When you look at the bar graph here, you look at their red bars, their gray bars, and then there are the gray bars that have the blue dots. So each of these blue bars that have each of the gray bars that have blue dots are representing what the drug resistant variants are. And then the ones that are highlighted red are showing the unusual mutations that were present in our samples. And we therefore find that next generation sequencing is able to detect these minority variants among the patients, but also there is need for continued surveillance of the drug-resistant variants that are present in the samples, because this could have synergistic effects and could have additive effects towards the antiviral resistance. Thank you so much to my funders and to everyone who was a part of this work.
Thanks, Maria. Next, we have Malaku Talahun. Malaku will present to us the popular perception on genomic studies and hereditary disease in Ari community, South Omo Zone, Ethiopia. Take it away.
Good afternoon. Thank you for the opportunity. This rapid ethical appraisal was conducted to explore genomic study in Ethiopia, and this study was conducted before starting participant enrollment for the main study division Africa, which aims to unravel susceptibility to tuberculosis in Africa. So the study was conducted in Ari community. Ari community is found in the Southern Omo Zone. Southern Omo Zone is the home of 16 ethnic group, and in terms of population size, the Ari community is the largest one, and TB is highly prevalent in the Ari community compared to the others. So using in-depth interview and focus group discussion, we explored popular perception and awareness to genetics, genomic research and hereditary diseases with the members of community elders, health officials, as extension workers, TB patients, and apparently healthy individuals. So the RE community explains the concepts of inheritance using phenotypic traits encountered in their daily lives, and they also considers few diseases as hereditary diseases. By observing familial aggregation of diseases in certain families, they consider diseases as hereditary diseases. But the RE community has limited understanding of the role of gene-environment interaction in shaping one's cells, but this is exceptional in case of asthma. And despite their understanding about the role of genetics in health, some of the study participants has expressed their willingness to not to participate in genetic study because they are afraid this genetic study may prove scientifically the genetic background of these diseases, because these diseases are stigmatizing in the community. So the study participants are afraid of this, so they express their willingness not to participate in genetic studies. So overall, we have got this study. The RE committee has a limited understanding about hereditary disease. Therefore, community engagement and public awareness programs should have to be conducted before starting any genomic studies in that community. Finally, I would like to these funding organizations and agencies. Thank you.
Thanks so much, Shanaka. Next, we have Modibo Goita. He will present the profile of individual telomere length of a rare chromosomal abnormality in chronic myeloid leukemia in Mali.
Thank you. It's a pleasure for me to present to you the profile of individual telomere length of a rare chromosomal abnormality in chronic myeloid leukemia in Mali. So chronic myeloid leukemia is a myeloproliferative disease caused by a translocation between the chromosome 9 and 22, leading to the formation of the Philadelphia chromosome. At chronic stage of CML, complex chromosomal abnormality are uncommon, as well as derivative 9 chromosome inversion are extremely rare. Besides, it's known that telomere shortening is correlated to the worse prognosis of CML. So our aim was to describe the telomere profile of the derivative 9 pericentric inversion in CML. To do so, firstly, we did cell culture, followed by the CML fluorescence in situ hybridization, then multicolor fish, and we ended by quantifying the telomere profile using the quantitative fish techniques. After CML analysis, we found an atypical pattern showing a missing derivative 9 signal, as we can see in an interface nucleus image here. To investigate this missing signal, we did a multicolor fish. As we can see, instead of evolving the long arm of the chromosome 9, a part of chromosome 22 was located on the short arm of the chromosome 9, and then cytobine analysis revealed that it was resulting from a pericentric inversion that confirmed the missing signals. And then after following the fluorescence quantification, statistical analysis show that the telomere length of the homologous chromosome 9 and 22 was heterogeneous, as we could not infer from here if the shortening was significant or not. Furthermore, we have measured the telomere of a classical CML cases in order to compare both, as we can see here, showing that the telomere length of the derivative presenting the inversion is shorter than the classical case. In summary, this is the first time to profile the individual telomere length of a derivative chromosome 9 pericentric inversion in CML, and this variant may be involving in a therapeutic resistance in CML. As next step, we aim to characterize the breaking region involving in this translocation using the whole exome sequencing. I thank you.
Thanks so much, Madiba. So next we have Nanaya. She'll present to us the genetic association of a polyvariant and the development of preeclampsia.
Right, thank you. Next slide. So slide, please. So preeclampsia is a hypertensive variant that occurs during the second trimester of pregnancy, and it is usually characterized by the elevated blood pressure of greater than 140 or 90 meters of mercury. Now, racial disparities account for the variation in prevalence globally. Now, it has been known that women who develop preeclampsia tend to develop cardiovascular impairment or chronic kidney disease at a later stage in life. The maternal phenotypes alone cannot be able to explain condition development, and as such, there is the need to explore other alternatives, most especially in the genetic field in African populace. So as such, we hypothesize that variants of the apolipoprotein, a gene which is known to cause an increase in chronic kidney disease, may also cause an increased risk in preeclampsia development. And as such, we aim to determine if infant APOL1 stratified by maternal APOL1 risk would cause an increase to the development of the condition. Next slide, please. So this was a paired case control study where participants were recruited at the OBGYN unit at the Kolebu Teaching Hospital. So maternal venous blood, DNA was isolated from maternal venous blood as well as cord blood, after which we employed a low-cost efficient genotyping technique, which is post-PCR LDR assay, to assess the genotype distribution among the study participants. Next slide, please. So for the APOL1 genotype distribution among the maternal group, we didn't see any differences in genotype distribution. However, with the fetal study population, with the fetal study group, there was a significant difference in genotype distribution. Next slide, please. So we went on further to check for the associations of APOL1 risk allostatus, either by high risk or low risk, to the association to development of preeclampsia. However, in the maternal group, there was no significant association. But in the fetal group, there was an association for fetals that had the high risk allo. And that we observe an odds ratio of 2.56 in this population or in this study group. So in conclusion, babies who carry two APOL1 risk allo have mothers with an increased risk of preeclampsia. And if this is indeed so, then not only mothers are at risk of developing kidney disease, but then the babies as well at a later stage in life. Thank you.
So next we have Nicholas. Nicholas will present to us the comprehensive benchmarking of copy number variant detection tools using whole exome sequencing.
Thank you very much for introducing me, and greetings to you all. So copy number variation is a type of genomic structure variation that contains segmental duplication or deletion of a DNA fragment. Its size ranges from 1 kilobase to 3 megabase. It has gained a significant attention because it has been known to contribute to complex phenotypes and disease vulnerabilities such as Down syndrome, schizophrenia, osteoporosis, and certain types of tumor. Among the approaches for obtaining samples, the whole exome sequencing is an attractive alternative due to its cost and the fact that disease-causing variants are thought to lie within the exome. Now, developers now use 4 various or main approaches, that is the read depth, read pair, split read, and de novo assembly to build tools to detect these CNVs. But among these 4 methods, the read depth is gaining much attention because it provides the opportunity to set a sliding window in which you're able to assess the mean of the reads and make sure that you employ either statistical or mathematical methods to be able to detect the CNVs. Though there are several methods that have been developed, the challenge now is there is currently no tool established as the best tool for detecting CNVs. So for one to make an input, the first task is to try and assess the performance of the tools based on certain metrics or criteria. For us, we try to do that using the number of CNVs detected, the accuracy, the time, and the memory usage. So to be able to do that, we shy away once again from benchmarking published papers. We try to use European data sets by using 10 ESOM data set, 5 each from Nigeria, the Yoruba group, and Gambia, the Madinga group, based on the phase 3 of the 1,000 genome project. And we try to follow their documentation and assess all pre-processing protocols. Obtaining the data set, we try to use 8 published tools as shown in the tools section: CANU, CNP mobs, ESOM Code, CNV kits, the CNV Contra, and ESOM copy. It was shown that when it comes to predicting CNVs, each tool have its own number of CNVs that it will be able to detect it. For the CNVs that were detected, ESOM copy, ESOM Contra, CNV kits were shown to predict a higher number of CNVs. So we went ahead to try to check its accuracy. Based on the precision, almost all the tools try to have above average precision, with the exception of ESOM copy. But when it comes to the recall, most of the tools were lacking it, but ESOM copy was having a very higher recall score. We went ahead to also assess its time and memory usage. The time for Codex was the highest. They tried to take at least a day or some few minutes for it to finish running, while the other tools ranges from 3 to 8 hours for us to get our results. The tools were run using a standard 16 gig ThinkPad, Lenovo ThinkPad laptop. Although there have been a progress in this field, we still see that there is the need or the urge to look for novel, robust method to be able to build new algorithms to detect these CNVs. Thank you very much. And I would like to acknowledge my supervisors, founders, and institution.
Thank you very much. Can we give them a round of applause? They've done. I think this group has done exceedingly well, and a lot of us in this room can learn from them. You've done so well. In the interest of time, we will have just a few questions, and can we just follow their trend and be very brief with our questions, and they will be very brief with their answers as well. So do we have any immediate questions to our panel? Yes. Jen.
Don't worry, I don't have a question for each of you. I have one question, and as many people as want to can answer it. So thinking as I'm waiting for my COVID test and hoping that the results come in really quickly, I'm thinking there's a huge difference between clinical timelines and research timelines. And so how are, or should we be addressing that gap of maybe 7, 8, 9, 10 years before there's anything even to feed back?
Okay, we'll just take one more question, and I have a few so that you can all be thinking of that wonderful question. Do we have any other burning question from the audience? For I, and I just wanted you to clarify what you mean by genetic testing, because a lot of the clients didn't understand what it was for, and just to clarify that. Mamadou. Mamadou, you talked about pockets of conserved region within the malaria falciparum. And I just wondered whether you see a similar one among the other species of malaria, or you haven't looked at that yet. And I believe Maria mentioned NGS as a really effective method of getting the no abundant variants, the kind of not your very obvious mutations. And I was just wondering whether you were thinking of other easier methods of of getting to that than NGS, because a lot of our labs in Africa don't have that. Maybe you have an idea in terms of networking that would help us with that. Malako, one of the things that you said was your audience were a little afraid of even getting the results from genetic testing. And I just wondered maybe you can share with us some of the ways that you have been able to reassure them in terms of what genetic testing is about. So the rest of you, I'll ask you questions when we either are dancing or when we are having tea or coffee. Okay, so all right, I'll start with you. You remember that next question is for all of you.
Okay, so I would say build more institutions and then put systems in place that will speed up the process. Thank you.
Thank you, Prof, for your question. So next generation sequencing is quite expensive, like you mentioned, for the African countries, and I agree. However, the other methods that I would suggest still have sequencing in it. So for now, sequencing or NGS is the run to. What we can do is we can make it policy so that people are able to access these services, just like they're able to access the rest of viral load testing by the government. But if it's on a personal basis, it's expensive for the general population, but it is efficient for the minority variants. Thank you.
So can you repeat the question again?
Yeah, I thought somebody was going to ask that. I wasn't quite sure I understood either.
All right, I was trying to keep it brief, but I will be a little, okay. So when they take my sample to do a COVID test, they're going to do that test today. When we start recruiting patients to be in a clinical study or any sort of study, research study, especially a genomic study, we're going to take their consent now, we're going to take their sample maybe later. That sample is going to sit in a freezer for a really long time until we have thousands of patients. Then they're going to be sent off to get the sequence data. Then that data needs to be cleaned and analyzed all together as a group, and results need to be analyzed to see what is significant before we even know if there's a result that could be fed back. And so thinking about the projects that we all work on here, there are genomic results that aren't yet in yet from projects that started recruiting 7 or 8 years ago, right? And we don't have their genomic results yet, or those are just coming in, but they haven't been analyzed yet. So when you're talking to your community about feeding back findings, are they understanding that this is not a quick turnaround time, and how are you thinking about explaining to them how that might be done in 10 years, not tomorrow?
Thank you. That was crystal clear. Thank you. Please go ahead.
Okay. If I understand, the question is about the samples, how can I say, the storing sample in 10 years ago, how this sample can be. No. So can you be a bit concise?
So she's saying, okay, you collect blood now, okay, and with all the genetic testing and analysis that needs to be done, okay, you may not get the results until 5 years from now.
Okay.
So as you engage with your client, how do you prepare them for the amount of time it's going to take to get the results? Did I do good? Kind of, she said.
Okay, try to answer.
I don't know if I get you clearly. What you're trying to ask is, no, this is what I get. Please bear with me because I'm trying to get what she's saying. She's talking about a turnaround time that takes a longer time, for example, 10 years, like you mentioned. But what are we putting in place to make sure that in 10 years, not tomorrow, in 10 years, you're able to get immediate results. And I think—
No.
Okay.
If you're doing a clinical genetic test for somebody, you're promising them the result in a week. If you're enrolling a participant in a genomics study and you're talking to them about returning results to them, the timeline for which it will be until they get a result is very much longer. And you might not even be able to find them to give them an individual result in that timeline later. So all the people that are thinking about both what are your implications of your results for your communities, and how does research progress? When are you actually going to have something important about someone's health to tell them? And what are you promising them? And is there a gap between those two things? Or what are they expecting? Maybe not what you're promising, but when you talk about returning results to them, what are they expecting in terms of a timeline versus what a timeline might be?
And can I just clarify that you don't all have to answer it? Those who understand, please answer so we can move along. Okay. Do you want to pass it on? Okay, you have.
So I would say, don't overpromise. Be realistic. So you tell them, for now, for kidney disease research network, you tell them you can get your biomedical results, which obviously will come early. But for the genetic, it's going to take a longer time because we might not have the institute systems in place now, but it will come. So it will come maybe after 5 years' time before you get it. And whilst they are waiting, you can engage them, community engagements. Yeah.
So Jen also wants to know that, will you tell them that you may not have a result to share with them?
Yeah.
And that's okay.
Yeah.
Okay. So let's go to the gentleman.
Okay, let's retry. In my case, it's like I'm doing chronic myeloid leukemia. The result doesn't take such time to be given to the patients, and some almost 10 days patients are getting their results in my case. Thank you.
Thank you.
Okay, it's mine now.
Turn by turn. Do you want to add something? Yeah? No, the guy, yeah. It's okay if you, yeah, go ahead.
Hello.
Okay, go ahead.
Yeah, I don't know if I've understood it well, but what I think is it's important to sensitize communities about the importance of research, which I see it's a very big challenge in Africa. Now, like during COVID-19, for example, I happened to work on a COVID-19 program in Uganda at Uganda Virus Research Institute. There are studies that were involving clinical trials on the vaccines, but people don't want to go out. They don't want to participate. Yet it is through research that clinical decision is made. So I think it's important to sensitize communities the importance of participating, being involved in research.
Okay, thank you. And you, okay, you have your light on. Go ahead.
All right, thank you.
Yeah.
I think you need to be very clear from the start that this is research that is different from clinical care.
Mm-hmm.
So because with clinical care, you may have results early, but with research, most of genomics, it may be hard to get validated results. So you'll be clear from the start that your results may not be back soon, or they may not even be back to forever. So you be very honest at the very start. However, I think researchers can have strategies in place during that time, from that time, like you can continue engaging your community. If you have things like you can boost them with like knowledge, or capacity development in that they don't forget about you by the time you happen to get your results when they finally come back. So you continue engaging them, but you be honest from the very start so they consent to participate whether they will get results back or not. Thank you.
Thank you very much.
Okay.
Thank you.
Okay.
Anybody else, if you have any addition to what has been said, but I believe, is it Henrietta? You have a specific example because in your own case with the eye infection, you did tell them your study was about whether they were interested in genetic testing. So in terms of really engaging them, how have you engaged those clientele? And I'll come back to you. I'll come back to you in a minute.
Okay, so we're actually trying to find out what people really knew about genetic eye diseases, genetic testing, different aspects of testing, counseling. I just presented a small part of the study. So in terms of genetic testing, we wanted to know if they had even heard about genetic testing.
So when you engage them, what do you tell them genetic testing is?
Okay, so we tell them that there are some eye diseases which have a genetic basis, and that some of them are, I mean, we can actually find out really what's the main cause of this disease, what's the mutation that's responsible for this disease, because for so many, we also give them some instances where or examples where this knowing the exact mutation that's responsible can influence management, can also help us counsel in terms of the prognosis, visual prognosis, the risk to other family members and things like that. So if they understand, so if ordinarily we find out if they've heard of testing, then what they know about testing. And so, yes. So that's
Thank you very much. Your turn.
If I understand, you ask about if I see the same region in other species.
Oh, falciparum, yes.
Yes,
You talked to
for
us about-
Plasmodium falciparum. Yes, we see the same region in H3 coli because, yes, to construct the model, we're doing the homology modeling. The sequence of H3 coli has been used has template to construct the model.
Wonderful.
Yes.
All right. Thank you all for this wonderful engagement. And thank you. We'll continue during tea. You've done excellently well. So we'll take a break now and listen to a presentation by Grace Ajuon. Is Grace Ajuon in the room or is it? Yeah, please come over. It's and give us your presentation. And then after your presentation, we'll go to the second cohort of trainees. Thank you.
Thank you very much. Good evening, everyone. Yeah. My name is Grace Ajuon. I'll be talking about the tools that we need to enhance the visibility of our research. So this is the outline. Please, next slide. Yes, your paper is worthless if no one reads it, uses it, or cites it. It's useless. It's not meaningful. So, in the traditional setting, you conduct your research, you write the papers in journals that have high impact factor, and then you are okay, it's fine. But in this day and time, you have to do more work. And that is why I'm here. I am a librarian. I'm not going to be talking about genomics. I'm not going to be talking about biorepository. I'm not going to be talking about, I'm not a scientist. I am a health librarian. Next slide, please. Diseases. So I'm going to be talking about citation, but I'm not going to waste time on this because we already know what citations are. And we know the citation styles. There are several of them. If you are using EndNote, we have over 5,000. For those that are using Zotero or Mendeley, we have over 1,000. Different journals have their different referencing styles. And then we have this citation analysis. And we are using online bibliographic databases like Scopus, Web of Science, even Google Scholar to measure this, to do these metrics. We also use our metrics. I know some of us may know Publish or Perish and Plumex to do the analysis. We are familiar with H-index. We are familiar with I-10 index, which is common with Google Scholar. It's only with Google Scholar you have I-10 index. Actually, it was developed in July 2011. It's only in Google Scholar you have this. And what does it tell us? It shows the number of a researcher's publications that have been cited at least 10 times. That's what it does. And among us, someone like me, struggling researchers, please cite my work. Cite my work because I'm still struggling. And you can see those young ones, they are coming up. So what is visibility? Remember, my first slide said, if your work is not used, is not cited, it's not useful. So what is visibility? It's about creating an identity for yourself. A presence. The extent of which, as a scholar, your work is visible online. If you are not online at this age and time, your work is not going to be visible. That's the truth. Connections. Are you connected? Reputation. Reputation doesn't just come. You build it over time. And then groups. You have to collate to groups. If you are not connected to groups, your work is not going to be visible. Conversations, you have to engage. And then sharing. Many of us are on WhatsApp. So when you publish your paper, start to share it. Don't expect the journal, that journal is going to do it all for you. It's not going to happen. So, research visibility, the process of making research finding visible to the global scientific community. Using academic social networking sites, social media platforms. So, get more citations. Every morning when I wake up and I want to search my mail, the first thing I want to do, who has mentioned me somewhere? Who cited my work yesterday or even now? Those are the things I look out for. That makes me happy. I don't have too many publications, but if I have, I want them to be seen and cited and used. So how? How? Make sure you publish in open access journals as much as possible. Share your data as much as possible. If you have data, share it. Create profiles. Be consistent with your name. My name is Grace Ajuwon. The next time I'm publishing, I write G Ajuwon. Grace Ada Ajuwon. You have different variations of your name. You are not helping yourself. Yes. Now, there are repositories, institutional repositories. Ensure that as much as possible, you don't sign off the copyright of your work so you can deposit your work in your institution's repository. It will help your work to be visible. Then try and join academic professional networks such as ResearchGate, LinkedIn. Many of us are on Twitter, but we tweet other things, we don't tweet our work. Yes, we don't tell the people following us that, yes, I'll publish an article, read it. We don't do that. We're on Facebook. All we do is to take photograph with our new car and post it. But when we get published, we don't do that. So why is it important to be visible? When you are visible, you'll be invited to collaborate with those who are working in the same area with you. It will showcase your work. You will have opportunities. I remember when COVID-19 started, those in the area of infectious diseases, they were being invited by radio stations, television stations to come and speak. If you are not visible, if you are not known, nobody will invite you. You will have high citation, and your citations will be making impact because people are seeing them, they are reading them, they are citing them. You have opportunities to collaborate with your peers and your colleagues across the globe. So how do you do this? What are those strategies? For those of us, the older people, maybe this is no longer useful because you're already there. But those who are just coming up, start from the point where you are writing the manuscript. Start from that point. Your title, the title of your work, make sure it's concise and relevant. The abstract, the keywords or concepts within your abstract, repeat them so that the databases, the search engines be able to pick them when someone is searching. So use attractive technical terms or keywords. Subject headings. Publish in reputable journals. Publish in open access journals. Open access journals, I can't say it enough. Open access journals, that's the way to go. If you also want to publish, publish in journals that have both print and electronic versions. It's good. Many of us presented papers here. Present your papers in conferences. Then create profiles. Research identifiers, create them because they have their benefits. Academia.edu, Facebook, LinkedIn, My Science Work, Pen Profile, ResearchGate, Twitter, create profile there. All of them can help you to raise your profile, all this. Social media. Don't be shy from social media. Be part of it because they will help you to share your research work. ORCID ID. For those whose names have variants, the same name, but you write them in different ways. If you don't have an ORCID ID, there's nothing that will bring all your papers together. But if you have ORCID ID, it will help you. Please, I want everyone listening to me. If you don't have an ORCID ID, do that today. Do that today. You need it as a researcher. LinkedIn will help you. SlideShare. Many of us, when we want to make presentations, we go to SlideShare to get, but we are not putting our own slide there. Our slides are not there. Develop your slide, share it. When you do that, you are known. Your work is speaking for you across the globe. How many of us use it? Reference manager. Mendeley is a reference manager. It's free. It's now owned by Elsevier Publishers. It is not just a reference management tool. It is also a social networking application. Use it. It does a lot. You can use it. All of us, every one of us can form a group and we begin to communicate. We begin to share our publications. When you publish, I get to know if we have we have group. We form a group under Mendeley. That way, we are going to be able to connect with people, share data, share our publications, and then the word will go out and that will become more visible than ever before. Research ID, Google Scholar, create profile in Google Scholar. Some institutions now, if you want to get promotion, they say, go and create Google Scholar. I want to see how you are doing. If you are Google Scholar, if your profile is not high up, your H index is not there, you are not going to be promoted. Create your own digital footprint. Don't be analog. Yes, digital footprints. Have an identity for online presence. You need it. In your institution or your university. If you're investing, if there's a website, make sure you have your own page and that your publications are there. Professional networks, LinkedIn profile, create it. Academia, create it. Create ResearchGate profile. In social media, be there. Share your slides. Share your work. You are on WhatsApp. When you get published, there's something that interests me. Whenever someone publishes a new paper in my group, they share it and we are all happy. So that motivates other people to write. And then when you look at the paper, you are motivated to also share yours. Even your email, if you publish in an open access journal, and you just send the link after the signature of your name, somebody will want to know what's this link about. You open the link and see that it's a publication. You will read it. If it's of interest, you will cite it in his or her own work. Yes, this is my own Google Scholar profile. I don't have too many papers. My H index is just 9. But I created it. I'm also on Plobons. This is my OKID ID. ResearchGate. Academia. I am there everywhere with my few number of my papers. Yes. I'm there. Yes. LinkedIn. So my advice for researchers, get an OKID ID today. Take metadata seriously. What do I mean by metadata? In the past, we call it bibliographic details, author names, the title of the research, the title of the journal where the research is published. Those are what we call metadata. DOI, Digital Object Identifier. If it's a book, the year of publication, the date of publication, the publisher, all those details, take them seriously. Then use Twitter to share your work. Keep track of your digital footprint. Keep track of your papers. Every morning, I track my paper. Who has mentioned me? Where is my name mentioned? Every day, I track who read my work, who downloaded it. Blogs, if you are good in blogging, if you are not, if you think you can't do this, get someone to do it for you. It's as serious as that. These young people, they are very technology savvy. Get them to do it for you, and you will see how far your work will go. You become visible everywhere. Your work will be speaking for you, and you'll be tired of people calling you for collaboration. Yes. Now you need a sensible email address. Some email addresses are not sensible. Use professional email address. So other ways of boosting your visibility. Discuss your research. Join social networks. I'm saying it in different ways so that it will sink. Inform your colleagues that you have a paper published, creates visibility in social media with all metrics. How many of us know about all metrics? How many of us have heard of dimensions? These are tools that will help you. Please, they are very relevant tools. So using our metrics stand for alternative metrics. We know the traditional metrics we use in measuring our work. Here, our metric use to measure and monitor the reach and impact of scholarship. And how does it do that? You can increase the visibility of your research in social media with what we call our metric explorer. Article level metrics. It tracks research that is visible in social media or other platforms. That's what our metric does. So how does it do that? It goes to the article data set using those identifiers, those that you have created, either in ResearchGate. It goes there and picks them from DOIs, from PubMed ID, and then collect dimensions and begin to, every morning, be telling you, your name was mentioned there. Your name has been mentioned there. So, and we give you names of those who mentioned your name and where your work has been cited. So you can track. So the checklist, use unique name, be consistent with the name you use. Eh? Create digital IDs, create research profile, English. Research is in English. Yes, that is where research is, English language. Collaborate, participate in conferences, use the right publication type. When it comes to research, it's journal article. So publish in journal, good ones. Choose appropriate publishing channels. Some channels are not appropriate, that's the truth. Choose and cite well-cited journals. There are some journals that are not well-cited. I've said all this. Thank you very much. I want to thank the National Library of Medicine for sponsoring me to this place. I want to thank members of the Network of African Medical Librarians, organizers of this conference. I say a big thank you. The College of Medicine, University of Ibadan, where I work, for releasing me to come, and my family. Thank you so much.
Thank you very much, Grace. Thanks for a wonderful presentation. And thank you also for leaving some time for questions. No, here, yeah. So I'm sure that our fellows have a few questions. I'm going to give the privilege to the fellows first and then anybody else in the audience. Yes, at the back, please. There's someone at the back and you.
Thank you very much. Name is John Mukisa from Uganda. Wonderful presentation about scientific communication, basically, for me to put it. But my key question to you would be is, many times as scientists, we get the fatigue of being online. What's your experience in trying to maintain that consistency of scientific communication while online? Thank you.
Thank you very much for that great question. I do many of these things early in the morning. That's the best time for me. So you don't do it when you are tired. Do it at your best. So when you do it, when you're already fatigued, you'll be it's not going to be fun to you. Do it at your best. Your best may be early in the morning or late in the night. Do it at that time. Thank you. Any other question? Yes.
Sorry, it's more of a comment. Is this on? Can you hear me? It's just a comment about predatory journals. One of the perils that we often get into as researchers is, I know my mailbox is full of invitations where they don't even spell my name correctly. And I'm an expert in everything in the world. I could write about engineering or Shakespeare or who knows what. So often when we have inexperienced researchers, they get caught in those traps, especially if they think that their research is great and they've got the ResearchGate profile and people are asking them questions, and then they start getting the invitation. So I guess it's more of just a little bit of a warning. It's great to publish open access, although it's very expensive, but also, if somebody offers to sell you a diamond ring online, don't buy it.
Yeah, it would be one short contribution and one question. My contribution is I was advised by someone that I respect many years ago. It's the same advice that I give to the younger than me. Don't worry about the impact factor of the journal. Don't go for Nature. It takes 5 years for one paper. Publish as much as you can. If you would PubMed Nikki, you see the number of papers she has. How many times would you take to look for Nature papers among the papers she has? The only thing you would see is the number of papers. At some point in the career, the impact of the journal, how many Nature, it doesn't count. It's how many papers this person has been able to publish in his career. This is what people really get in mind. And my question is, Jennifer, I see Laura, is there a limit for the number, the amount of money you can budget for publication? Because the article processing charges are actually really expensive. When you are writing and submitting a grant, is there a limit amount of money you can budget for APC and open access publications?
There's no limit as long as you have grants that cover the payment of article processing fees and the number of publications you want to push out in a particular year. There's no limit. But if you are going to be paying this out of pocket, I don't know how, because in Africa, how many researchers are able to pay on their own? It's difficult. It's really difficult, except there is a grant and you channel it through those grants and get your papers published in open access journals. But as much as possible, let's try. Because it is through open access journals that our work is made more visible. I hope I've been able to answer your question. Thank you. Now you talk about Nature. In the past, previously, what matters is that you publish in a high impact journal. Beautiful. But my second slide shows that 48% of articles published in Nature, that's research was actually conducted by Nature publishers. They wanted to find out how many of those publications were cited. And 48% were not cited, never ever cited. And they wanted to know why. Meanwhile, there were others. Some of those publications, breakthrough, well-announced research, but they didn't make the first 100 cited work. Why? Those whose works are not so well-known, celebrated, were busy. They're everywhere, pushing out their work for people to read and cite. Those whose work are novel just ended it there after publication. When you end your work at the point of publication, you have only done 50% of the work. The remaining 50% will just be there. Nobody will do it. You now take the next step of making sure you disseminate as much as possible. Create profiles, push your work out. Let people know you have done beautiful work. So here I'm in Nigeria. I want to read what someone in Makerere University has, the research going on there, University of Zambia, University of Zimbabwe, all across Africa. But if we leave it that the journal will do that, it's not going to happen anymore. We're in the 21st century, the things are different. Thank you very much, Mark.
Thank you. Round of applause again for her. Thank you very much. And thank you, second cohort, for coming up before we even call your names. It's really helpful because we're running out of time. Rolanda has done such a great job with the first cohort. I'll hand the mic over to her to start us off with the second cohort. Thank you.
Thanks, Alasha. All right. So for this group 3 for today, we call on Alfred to present to us QuasiFlow, a next flow pipeline for analysis of NGS-based HIV-1 drug resistance data.
Thank you very much. Good afternoon, Africa. Alfred Sekajidi here from Breca Makerere. And I'll be briefly telling you about next flow pipeline that we are developing for analysis of NGS-based HIV drug resistance data. Next slide, please. Right. So next generation sequencing has been proven to reliably detect low abundance resistant HIV variants. And this is important because some of these have actually been implicated, associated with treatment failure. We got some bioinformatics challenges in our ecosystem, some of these being the scarce bioinformatics expertise and also unreliable internet connectivity. And this is a point because most of the user-friendly tools are mostly web-based and require good internet connectivity. What we're doing about this is to actually come up with this pipeline that we think is more user-friendly and doesn't require all-time access to internet to get things done. So what we have actually done is to put all the tools that are used to analyze the data into a single tool and away from quality control through data processing until the point where we generate drug resistance reports. We are using Nextflow to automate these processes. We're using Docker Singularity to make sure that this tool runs well across all computing environments. So we are testing this and trying to validate it using publicly available data. And what we have right now is that we have up to 90%, 99% agreement between what we are developing and one of the mostly used algorithm for this work. So we think after everything we're trying to do, QuasiFlow will help us effectively manage this data and have prompt results from this analysis. Thanks to the team that is helping out, and I thank you.
Thanks so much. Thanks so much, Alfred. So next we have Andrews. Andrews will present to us today benchmarking of long-read assembly tools for analyzing African human genomes.
Thank you very much. Okay, so yesterday, Prof. Nikki made mention of the fact that Africans are underrepresented when it comes to the human reference genome that we have. And it kept me asking several questions. Next slide, please. Yeah, so I kept asking myself, why? Was it because in the time of the reference genome generation, the sequencing cost was very high, or probably maybe many Africans were not born or are not born as at that time. But how about now, when where we have the sequencing cost being reduced, when we look at the complete human reference genome as at today, which still we are underrepresented. But the good news is now we have many African genomes being sequenced, as Nikki made mention yesterday about the African reference genome. And the challenge that we often face is we have so many tools available with different algorithms that are used to build these tools. And selecting a tool for your analysis is often a challenge for research teams, which tool will work best for your data set. And as of now, most effort that have been put in place to look at these tools have focused on bacterial genomes, not even talk about human genomes and even African genomes. So the work was to access the performance of 10 commonly assembly tools that are being used to analyze human genomes, and also to assess the performance against certain matrices that will enable us to know that whether the tools are of good quality or not. So we took data set from the human pangenome, African data set from the human pangenomics repository, and we also work on a cluster of memory 500 gig and 34 threads of CPU. And many of the tools were based on hi-fi data set, and others were in generally for long read. So after doing the analysis, the results that we obtained, the green color shows the good performance of each of the tools that we had, whilst the orange colors also look at the worst performance of the tools that we accessed. And looking at the results, we find out that Fly came out to be the best among all the tools that were tested, while some of the tools couldn't even run to completion, for instance, MDBG. And also looking at it, we realized that Fly also had so many mismatches in there, which was also based on maybe the algorithms and the framework that were used to generate it. So in conclusion, we realized that Fly was the reliable assembler. And also depending on the kind of analysis that you want to do, whether you want to have only haplotype or you want to talk about diploid kind of genomes, then if it is only the haploid genome, then you can go ahead and use the Fly. But if you're talking about diploid genomes, then you can go about looking at other tools that also perform best. And reference-based statistics that we also look at, when you look at the tools, they were all comparable. And also the computational result, with exception of Kano, which was very fast, and the averages tool, which also took longer time to perform, all the tools were all comparable. So in the end, we realized that we can use Flyer, or you can go ahead and look at other tools that were also comparable to do your analysis. Thank you.
Thanks, Andrew. So now we have Carlin Fiona. She'll present to us the impact of COVID-19 pandemic on the sputum conversion rate and treatment outcome of TB patients at some diagnosis and treatment centers in southwest regions of Cameroon.
Thank you. Okay, before COVID-19 break out, we know TB was top ranked as causing disease from a single infectious agent. But with the outbreak of COVID-19, COVID-19 is now top ranked in front of TB. And WHO states that the COVID-19 pandemic will disrupt the measures put in place so far to improve the TB diagnosis and treatment services, and may lead to a drawback in achieving the 2035 NTB goal. As there is a lack of information on the effect of COVID-19 on the treatment outcome in Cameroon, and that's where the study comes into outline the possible effects of COVID-19 pandemic on the sputum conversion and treatment outcome of TB patients in Cameroon. As method, we reviewed data of patients in 9 health facilities in Cameroon from before COVID, that is from January 2019 to February 2020, and during COVID period, that is from March 2020 to 2021. And as a result, we observed that at 2 months, the sputum conversion rate of TB patients was higher during the COVID-19 period. That is 98.3% higher as compared to before COVID-19, and this difference was statistically significant. And there is the same difference at 5 months, with the sputum conversion being greater during COVID-19 era. As compared to the treatment outcome of patients, we observed that the mortality rate of TB patients was higher before COVID than during COVID-19, and equally a very high proportion, that is 13.8% of patients, were lost to follow-up during COVID than before COVID-19 period. And equally, this difference was statistically significant. So we concluded that patients were lost to follow-up during the COVID-19 pandemic, and equally, COVID-19 didn't really affect the sputum conversion rate as we thought in the different centers. So we called on the health system to improve on strategies to retain patients in the directly observed therapy during COVID-19 by reallocating the various resources towards TB to ensure good patient compliance. Thank you.
Thanks so much, Karen. Next, we have Shik. Shik will present to us osteogenesis imperfecta in a Malian family, clinical and genetic features.
Thank you very much. I would like to start by thanking H3Africa for giving me this wonderful opportunity to present my work. My topic is about osteogenesis imperfecta, clinical and genetic aspect in a Malian family. Osteogenesis imperfecta, also known as brittle bone disease, is a group of genetic disorder affecting connective tissue, leading to bone fragility and frequent fractures with minimal or absent trauma. The main clinical features include bone fragility and deformity, short stature, dentinosis imperfecta, and hearing impairment in adulthood. The condition is inherited in an autosomal dominant or recessive manner, and in 95% cases, mutations occur in these two genes, COL1A1 and COL1A2, encoding for type 1 collagen. Few genetic confirmed cases have been reported across the African continent, but none in Mali. The objectives were to clinically characterize a patient with OI and then identify the underlying genetic defect. The patient and her family member were enrolled after an informed consent and assents were obtained. Bone X-rays and blood chemistries, including vitamin D, were performed. DNA was collected from peripheral blood for panel gene testing. As you can see, a 7-year-old girl from a consanguineous marriage was referred to our neurogenetics for multiple fractures. She's from an eventful pregnancy and delivery, and she presented with short and broad legs and arms, blue sclerae, dentinosis imperfecta. We performed bone X-rays that showed multiple fractures and demineralization occurring mostly on long bones. We can also notice the fractures on the skull. According to the clinical and radiographic findings, osteogenesis imperfecta was the more likely diagnosis. The next step is to perform panel gene testing, including autosomal dominant and recessive genes that are COL1A1, COL1A2, LEPI1, PPIB, CRTAP. Additional genetic testing, such as whole exome sequencing or whole genome sequencing, may be needed. Functional studies will be done if you come across new gene. In conclusion, I would say this is the first clinical description of OI in the Malian family, and hopefully the genetic testing will uncover the genetic defect in this family and expand the genetic epidemiology of osteogenesis imperfecta. Thank you for your attention.
All right, we move on to Diana, and Diana will present to us the prevalence of advanced HIV disease in people living with HIV on antiretroviral therapy in the Abuja Regional Hospital and co-infection rate of Mycobacterium species and Cryptococcus species.
Ladies and gentlemen, you're all welcome to this presentation titled Prevalence of Advanced HIV Disease in People Living with HIV on Antiretroviral Therapy at the Abuja Regional Hospital and Co-Infection Rate of Mycobacterium species and Cryptococcus species, presented by me, Kolegu Chemini. So advanced HIV disease is defined as presentation with a CD4 cell count of less than 200 cells per microliter or a WHO clinical stage 3 or stage 4 event. Advanced HIV individuals have a higher rate of mortality, which is accelerated by opportunistic infections such as tuberculosis and cryptococcal meningitis. There is limited knowledge on the burden of advanced HIV disease within our setting. So to keep track of the commitment made by the government to end AIDS as a public health threat by 2030, knowledge of the burden as well as the risk factors of advanced HIV disease is essential. In our study, we hypothesized that advanced HIV disease was driven by opportunistic infections such as tuberculosis and cryptococcal meningitis. The aim of our study was to determine the prevalence of advanced HIV disease in people living with HIV and to determine the proportion of tuberculosis and cryptococcus in advanced HIV disease treated patients at the Abuja Regional Hospital. So in order to attain our main objectives, we conducted a hospital-based cross-sectional study between February 2020 and February 2021, in which 3,229 people living with HIV uncared during the study were screened for advanced HIV disease. 327 suspected advanced HIV disease patients were enrolled into our study. We investigated for tuberculosis and cryptococcal infections as well as other opportunistic infections and conditions. Chi-square and logistic regression were performed on SPSS at a p-value of 0.05 was considered as being significant. Our results indicated that 39.8% of our participants presented with advanced HIV disease, and it was more common among females and those within the age group 35 to 49 years old. The most common opportunistic infections and conditions present in participants presenting with advanced HIV disease were tuberculosis and cryptococcal infection. One participant presented with both tuberculosis and cryptococcus. When we conducted a logistic regression to determine the risk factors for progressing to advanced HIV disease, and we saw that advanced HIV disease was associated with male gender, and being on treatment for more than 12 months was protective for advanced HIV disease. So based on our results, we concluded that advanced HIV disease is far beyond the UN AIDS reduction target of 10% by 2020. So an improvement in the management of people living with HIV is warranted, and special focus should be laid on those people living with HIV during the first 12 months of treatment. So we recommend that further research should be done in order to add more strength to our implications. My references, thanks.
Thanks so much, Diana. So next we have Edwin. Edwin will present to us the gastric microbiome predictive model for gastric carcinogenesis.
Thank you very much, Rolanda, for the introduction. So gastric cancer is the 5th most common cancer in the world and the 4th leading cause of cancer-related death. H. pylori infection has been identified as the #1 risk factor for gastric cancer. But however, only 1 to 3% of people who are infected with a Helicobacter pylori actually proceed to get gastric cancer. And that as the disease pathogenesis happens, when one person moves from atrophic gastritis to gastric cancer, H. pylori suicide actually happens in the later stage, and other carcinogenic bacteria actually comes in to cause the disease. So what we did was to perform a comprehensive analysis of the gastric microbiome and gastric carcinogenesis to be able to unravel the other microbial communities that might actually play a key role in gastric cancer, and then also try and identify potential microbial signatures that could help us predict gastric cancer to help in the treatment and management strategies. So we use publicly available data, and we use 6 different studies which span across 985 samples. We employed a data workflow to infer microbial communities. We controlled for batch effect with a Hermann package in R. And then we built our model using the lasso technique, and then bacterial co-occurrence patterns were also elucidated. So in the microbial diversity, we found out that when we employed the Shannon index, we found out that the moving across the various histological stages, microbial diversity actually differed, and this was quite significant, and which gave us the first clue that different microorganisms may actually be involved. The microbial composition, we also found out that the gastric microbiota was actually dominated by 5 different phyla, and moving across the various histological stages, actinobacteria phyla actually increased with increased pathogenesis, and proteobacteria decreased with increasing pathogenesis. So in the co-occurrence patterns, we decided to focus on the gastric cancer individuals. And one thing we found out was that transient oral bacteria actually dominated this particular community, and most of them co-occurred with Fusobacterium nucleatum, which has been identified to be involved in the development of colorectal cancer. So it gives us an indication that probably the microorganisms that are actually involved in gastric cancer may actually be transient. So at this point, we were ready to try and identify microbial signatures. So we used the lasso technique, and then our model was quite accurate. We had a mean AUC of 0.9, and then Bacteroides dori was actually robust in discriminating healthy controls from gastric cancer. So at the end of the day, we concluded that microbial diversity and composition differed significantly, and then transient oral bacterium dominated the gastric microbiome, and then the Prevotella species co-occurred with Fusobacterium. Our next stage is to try and validate this using African population. So we've started collecting samples where we are going to infer the microbial communities from Ghanaian population. I would like to acknowledge my supervisor and all of these groups for helping me to prevent this. Thank you.
Thanks so much, Adrian. So next we have Elvis. Elvis will present to us the novel MARFAL2 variant associated with postlingual autosomal recessive NSHI co-Stangeneous family.
Thank you. Good afternoon to everyone. I'm Elvis Chimesi Abuja from the West Africa Center for Cell Biology of Infectious Pathogens at the University of Ghana. I'm happy to share with you our work that identified a novel variant in a postlingual consanguineous family in Ghana. So after screening this particular family of the most common variant that is associated with hearing impairment in Ghana, we used whole exome sequencing data and actually identified this marbled variant to be segregated with the phenotype in the family. And as you can see from the audiogram, the individuals that are carriers of the variant have a normal hearing, whilst those that are homozygous for the variant have the condition. So we use a bidirectional Sanger sequencing to actually validate this variant in the family. And we can clearly see from the chromatograph. So we move forward to actually look at the structure of this protein. So using a solved model of the protein from PDB, we used homology modeling to actually model the mutant protein using and viewed on pi mode. So as you can see, the fig A shows the wild type, the B is the mutant, and the C has the superimposition of the mutant on the wild type. And the D actually also gives us the prediction of this variant that actually leads to a truncation of the protein. So the domain colored red is actually what is truncated to result in the condition. Sorry, so we also move on to actually see how conserved this amino acid is in other species. And we can see that at this particular position, 354, where the valine is substituted for serine in our mutant protein, is conserved in all the species that were analyzed. So this is actually a first case of a marbled mutant report in Ghana as well as in Africa, although we have other reports from other families across the world. And this is the 13th report. And this mutation is novel, segregated with the condition in the family. And we've actually taken delivery of a construct to actually further validate this finding using animal studies. Thank you.
Thanks so much, Elvis. Next, we have Esther. Esther will present to us the vitamin D binding protein gene polymorphism and free serum bioavailability among tuberculosis patients and household contacts.
Thank you so much. Good afternoon, good evening, everyone. I'm glad to present to you part of my PhD work on vitamin D binding protein and bioavailability of the serum. This is part of the manuscript, which is in submission very soon. So vitamin D, as you know, is a molecule that is important in innate immunity and adaptive, and genetic variants influence the distribution of vitamin D, which leads to vitamin D deficiency. And there is an estimate of about 1 billion people who are living with hypovitaminosis D, which is a risk factor to tuberculosis. So we wanted to determine the association between the D binding protein gene and vitamin D bioavailability among the TB patients, latent, and individuals without TB infection. And after ethical approval, we got 103 participants from a previous study and then carried out DNA extraction. We did PCR, and then the figure there shows you the results of the PCR that were taken for Sanger sequencing to look out for that RS7041 and RS4588 SNPs using bioedit tool. Vitamin D was determined using the ELISA method. We did spectrophotometry, and then we interpolated our result unknown from a curve. So in our results, we predominantly found what we call the GCIF in our population. That is the genotype of the D binding protein gene, and the list was the GC2. We wanted to see the relatedness of these, our population, and we really see that they were closely related by the phylogenetic tree there. When we did a correlation of the vitamin D and the D gene and then the vitamin D levels, we found a very weak association between the two. And as you can see that in the GC1, the highest vitamin D levels were found there, and the lowest was in the GC2, and those were TB patients. So we come to a conclusion that the D binding protein gene, the GCIF, was predominantly found in our population, but there was no difference in the three groups. But due to the homogeneous population, we didn't find a significant association. So we recommend that maybe heterogeneous population can be used to find those minor alleles and then try to associate with the D binding protein. So I would like to acknowledge H3Africa and the funders of this study, that is Mopronano and Happy. Thank you so much.
Thank you so much, Esther. So next we will have Fatoumata, who will presenting to us the docking of human band 3 anion transport proteins with their Plasmodium falciparum interactors based on short linear motifs.
Thank you very much. I'm Fatoumata Fofana, and I'm from Mali. So I would like to share with you my study docking of human band 3 anion transporter protein with their Plasmodium falciparum interactors based on short linear motifs. So the human protein boundary anion transport is a man-man protein which role is to exchange anion across membrane plasma. And it's also considered as a main pathway for the invasion of parasites to enter the erythrocyte. And the parasite protein we choose here is a tryptophan-rich protein with less information on his function. But according to review literature, we found that it's a protein we can use as drug target or vaccine targets. And the short linear motifs are short protein sequences that mediate host-parasite interaction. And the goal of this study was to screen for candidate Plasmodium falciparum interactor as potential drug or vaccine target for the treatment of malaria. So to achieve this goal, we first of all identify the host parasite interaction. Sorry. Okay, so to achieve this goal, first of all, we identify host parasite interaction and host protein interactor using APID program. And secondly, extract the protein sequences from UniProt, characterize the common SLIMs of this protein using PathMat program, and predict the host protein, parasite protein interaction using David, and data functional analysis also using David program. So the second workflow was to predict the parasite protein structure was unavailable on PDB. And we did that using UniProt. We extract the sequence, parasite protein sequence using UniProt, did a template check using blast needle emboss, and predict the model using modular. And we did a drive-in protein-protein docking to see exactly if it interacts using the specific SLIM we identify with HADOC program and evaluate the protein-protein docking using COCOMAP. So as you can see here, the result, the figure in the left side is the template built under HADOC. So the green structure is the parasite protein structure that we predicted, and the white structure is the boundary anion human protein. So here is to highlight the importance of the SLIMs, which link these two proteins, and it's very important during the interaction process. So SLIMs can be used as a path to avoid infection during protein and human interaction. And it can also help us to predict candidate proteins. And you can see here in the right figure, the importance of SLIMs and the surface it's covered during the protein interaction between human and Plasmodium falciparum. So what we want to say here, it's the mechanism I propose here can be also take into account to avoid infection between Plasmodium falciparum and human protein-protein interaction. So it can be a way to identify new therapeutic targets also. So the next step will be, you can notice that all this work is done in silico. So the next step will be to validate experimentally this interaction we identify to confirm exactly that the SLIMs, the specific SLIM we identify is important during the interaction, and it's a good link. Thank you very much.
Thanks so much, Fatimata. Next, we have Vincent, and Vincent will present to us the molecular markers of Plasmodium falciparum, resistance of sulfadoxine pyrimethamine during prophylactic treatment in pregnant women in Muthenji, Cameroon.
Thank you, Rolanda. Good afternoon, everyone. I'm Vincent Intwe from the University of Abuja. I'll be presenting on molecular markers of Plasmodium falciparum resistance to sulfadoxine pyrimethamine during a prophylactic treatment among pregnant women in Muthenji, Cameroon. Malaria remains a huge problem in pregnancy. And to control this group, WHO emphasizes on the use of intermittent preventive treatment, that is sulfadoxine pyrimethamine, to control and prevent malaria in this vulnerable group. With increased resistance and spread, the SP resistance is partly attributed to treatment failure. And so the effectiveness of this drug is inevitable. And so it is therefore relevant to monitor the spread of this drug and its effectiveness. So our aim of this study was to determine the current status of these molecular markers resistance to SP and the effectiveness of the intermittent preventive treatment in 2015 and 2020. So prior to the study, we obtained ethical clearance, informed consent from the participants, and we recruited 300 participants. And we collected venous blood, which we performed blood aridity, we performed PCR after DNA extraction, and also we genotype these markers, the K540, S108, and 581. Interestingly, when we compared the groups of pregnant women on SP and the ones that are not taking SP, there was an overall improvement in the infection, which therefore implies that SP helped in protecting the women against the infection. And also, there was an increase in the S108 allele, as well as the 581, with zero prevalence in K540. So we therefore conclude that IPT SP seems to be relevant in this study population, and hence, and also there's an increased prevalence in the S108 allele. So we therefore recommend an investigation to determine the association between SP resistance markers and the efficiency of the drug. I'd like to acknowledge all the women who took part in this study, my supervisors, PAMGen and H3Africa. Thank you.
Thank you very much. Wonderful. Thank you all for this great presentation. Let's recognize this effort again by another round of applause. The good news I have for you for the second cohort is Jen was pulled out for an important meeting during your presentation, okay? So that's the good news. But we do have a few questions for you. Are there questions from the audience? Some people, I'll come back to you, but okay, so let's have a different person. I'll come back to you guys.
Thank you very much. There's our presenter who presented on microbiome. I'm interested in if you can comment on, if you look at a disease like cancer, sometimes it alters the immune system, it gets altered. Do we know whether the microbiome you find there is a result of the disease or is the cause of the disease? Is there a way that we can know that? Thank you.
Okay, we'll get all the questions and then you'll answer. And the ones you don't want to answer, don't answer it. Oh, no, I'm just kidding. Okay, you please, go ahead. The gentleman with the mic. Short question, and I think
quick question to Cisse. He presented about the osteogenesis imperfecta. Given the possibility of identifying novel genes in African, do you think that it's worth doing targeted testing, or would you rather suggest going for exome, for instance?
Thank you. Next question.
My own question is to Tui. You saw efficacy for SP, yet you saw some very high values for the resistant genes. So S108, for instance. Were you very concerned about these, or do you think that maybe there need to be some kind of an association for those molecular markers to form a particular signature that would be attributed to this kind of resistance?
Thank you. And the last question at the back.
Good evening. My question is directed to the scientist from Mali. I'm curious, what happened to the guy, to the boy that had all those multiple?
Okay, thank you.
Fractures, yeah, multiple fractures.
All right, our panelists, please go ahead. Yeah, press the button in front. There you go.
Right. Thank you very much for the question. With respect to the fact that microbiome as a cause for the cancer, we are not in a position to state that emphatically, because one thing we look at is the microbial composition actually differs, but we can't say with certainty that the difference is the cause of it. Besides, dysbiosis itself has been, microbial dysbiosis has been associated with gastric cancer, so in colorectal cancer and other cancers. But we are not able to say emphatically that this is the cause of it. And it might also need a lot of validation to be able to come out with a specific microorganism that is actually causing the disease.
Thank you. So association, really difficult to establish cause. Next.
Thank you. Thank you very much. I will start with the second question. It's not a boy, it's a girl. Okay, as you said, it's a very awful situation. The last time I saw her for a follow-up, she was complaining chronic. She was experiencing chronic daily pain. So she's just receiving painkillers. She undertook a orthopedic treatment, and she's receiving vitamin D because she has increased vitamin D. And she's not living with her own parent. She has been entrusted to her grandmom. So yeah, she's experiencing pain every single day. And we took advice with our colleague, the surgeons, and unfortunately there is nothing they can do for her right now. The second one, as we said, the condition is inherited. In 95% cases, it's inherited in an autosomal dominant manner. But here, the LEPR-E1 gene for the autosomal recessive, we think we might find this gene because there is a paper published by Professor Charles and Sarah Tishkov and colleagues, where we have found a mutation in LEPR-E1, especially in Ghanaian and Nigerians. So as we said, if you don't find anything, we can panel gene testing, we'll go for whole exome sequencing.
Thank you.
Thank you.
Next, please. Let's keep the comments brief. We're trying to get to break.
Okay, thank you, sir, for the question. Yes, I am. I'm concerned with if at all there's any, maybe there's a particular signature that is responsible for that high increase. But we can, I can't conclude, or we can conclude, because with the three alleles that were genotyped for the two genes, we have just S188 with a very high increase. So for further investigation, we intend to evaluate this across the country, because this is just one study site. So in that, we might be able to conclude if there's something driving this particular increase.
Thank you. I guess we've covered all the questions asked from the audience. Okay, we're kind of running late, so we will round this session up. But before we go, can we give them a round of applause? You are all going to be very terrific PIs because you do know how to be very concise.
you, Ashley. Thank you to the presenters. Just a few announcements. Can I please ask the PIs and the steering committee to join outside this hall in the foyer for a group photo? So that's the steering committee for a group photo. And then we need to break for 15 minutes and come back super quick because we have to catch up. Some folks that are talking in the sustainability session are.
So what are genes?
So I do appeal to
Genes are the
come
recipe
back,
for making your body the way that it
speedily.
is. Our genes provide instructions which tell our bodies to look and function in specific ways. Your genes are inherited from your mother and her family and from your father and his family. Because you inherit your genes from your parents, you may also inherit some of their traits. For example, if your mother has brown eyes, you may also have brown eyes. Or if your father is very tall, there is a chance that you could also be very tall because you inherited the feature for tallness. So exactly where in our bodies can we find these genes? Your body is made up of billions of cells. Cells are found everywhere in the body. There are cells in your blood, skin, muscles, and organs, for example. Every cell has thousands of genes inside of it. The genes are made up of a material we call DNA. DNA sort of means the same thing as genes. Every gene is responsible for providing specific instructions, like a recipe for baking a cake. Each instruction in our genes tells our bodies how to grow, work properly, and stay healthy. Your parents each pass on one copy of their genes to you, so that you have one copy from your father and one from your mother. Just because genes are passed on from your parents does not mean that you will look and be exactly like them. For example, if both of your parents are tall, it is still possible for you to be short. This is because our genes are randomly passed down over generations. For example, your grandparents might all be short, and even though your parents are tall, you may get the genes for shortness from your grandparents. Genes do not only make up how we look. Genes also provide instructions for things inside of our bodies which we cannot see, but that we need to be healthy. For example, we have genes that influence the shape of our heart or how strong our muscles are. So genes are really important for how our bodies look, and they are important for keeping us healthy. But genes can also make us sick. There are some diseases that are always caused by mistakes in the genes. Sickle cell disease is an example, but that isn't always the case. Our genes do not stay the same. Over time, small changes occur. We call these changes mutations or variations. Most of the time, these small variations or changes don't change anything in our bodies, but sometimes they can cause our bodies to not work so well, or they can make us ill. Most often, however, the small changes that we find in the genes mean that some people are at greater risk of developing a disease. That doesn't mean that they will get the disease. It only means that they could get the disease. For the disease to actually happen, other things also need to happen. For example, if you have genes that mean that you're at risk of developing diabetes, then that does not mean that you will definitely get diabetes. If you exercise, walk everywhere, and eat healthy food, then you can stay healthy. But if you eat unhealthily, don't move, and have genes that mean you can develop diabetes, then you could be at greater risk. This is the same as saying that there is a risk you can end up in an accident if you drive a car. Saying that there is a risk doesn't mean that you will end up in an accident. And you can take precautions, for instance, wearing a seatbelt and not speeding. In the same way, you could increase your risk of ending up in an accident, for instance, if you use your phone while driving. Similarly, genetics can also tell us whether people are at greater risk of developing a disease. It can tell us about things that people could do to avoid developing this disease, or things that could cause the disease to develop. But most of the time, genetics cannot confirm that we will definitely get sick.
You look like some members of your family, both generations past and present. This is because our genetically determined characteristics, such as height and skin color, are passed on from one generation to the next. In other words, we inherit our traits from our parents and the generations before them. We also inherit how our body will function or work. This is why certain diseases run through families. Your environment, diet, and lifestyle also contribute to how your body functions. The process by which our traits and function are passed on from one generation to the next, known as inheritance, is studied in a scientific field called genetics. The information on traits and function that we inherit are obtained from molecules, which are small particles found in our body cells called DNA. Specific segments of our DNA containing the information that helps to determine our physical traits, including observable characteristics and how the body functions, are called genes. All the DNA, including the genes that make up an organism, such as a human being, is called a genome. Genes store the information needed for the cell to assemble proteins, which eventually contribute to our specific traits and function. There are over 20,000 genes in the DNA that make up the blueprint for a human being. There is also a lot of additional DNA, some of which regulate how and when genes are turned on and off. The length of the entire DNA in a single human cell is about 2 meters. In order to fit this DNA into a very tiny cell, which is about 6 micrometers, it is tightly coiled or supercoiled around specific proteins to form chromosomes and stored in the nucleus of a cell. Every cell in an individual's body contains 2 copies of each chromosome, which carries the genes. This is because both mother and father contribute a copy at the time of conception. During conception, the DNA of your parents' egg and sperm mix in a unique way, and this process introduces variations or changes in humans. As a result, the genes can be inherited in different forms, and some forms work differently than others, which is why you end up with different traits. While a few traits are due to only one gene or a form of this one gene, for example, albinism and color blindness, most human genetic traits are the product of interactions between several genes. For instance, a trait like height or eye color is the result of a number of genes working together. DNA is also copied each time a cell divides as we grow, so that all cells contain the same DNA for its growth and function. Like most endeavors, nature can also make mistakes or changes during conception or in the process of making copies of the DNA for growth and function. These changes are scientifically referred to as mutations. Most of the changes do not cause any harm, but in some instances, they can result in a change in one gene causing a genetic disease. We refer to this as monogenic or one-gene diseases. And an example is sickle cell anemia. The change can also affect a group of genes that contribute to the risk or potential risk of having a disease. A genetic disease that arises from the contribution of a group of genes is called a polygenic disease and can be complex. Complex diseases are not only affected by the genes. Environmental factors, including lifestyle and diet, can play a major role in the onset of the disease. Diseases such as cardiovascular diseases, diabetes, and asthma are examples of complex diseases. Genetic and genomics data obtained from complex diseases studies are not always conclusive, but rather they serve as a guide for people at risk to balance a good lifestyle and improve their health. We hope this basic concept of genetics will help you appreciate why we collect blood and other human biological samples for biomedical research.
Africa is the oldest humanity. That means that we have 1,000 times more variation in African genomic data than in the youngest population. There will not be a good understanding of human genome without African genome.
African populations have been overlooked in the drive for precision medicine. Most genomic research has been undertaken in other populations, specifically European populations.
The effects of sequencing technologies becoming more accessible to African scientists is leading to a drive for more precision medicine within Africa. Programs like H3Africa are really helping to bring genomics to Africa.
The Human Hereditary and Health in Africa initiative, also known as H3Africa, came about from the African Society for Human Genetics and is funded by the National Institutes of Health and the Wellcome Trust. The broader goal is to improve the health of African patients. To do that, one had to improve the research infrastructure so that the researchers, African scientists, had the capacity to do this large-scale genomics research. When they developed the white paper for H3Africa, there was a need for a robust bioinformatics network to underpin the consortium, to manage the data, and to make sure that the data was going to be properly analysed and interpreted. We had an informal bioinformatics network through conferences that we've run every 2 years through the African Society for Bioinformatics and Computational Biology. When the H3Africa call came about, we were perfectly placed to address the call and to be able to implement this network.
H3Bionet comprises of 28 nodes in 17 African countries and 1 US partner. The personnel in H3Bionet range from master's students all the way up to professor level. We have expertise in various areas ranging from systems biology, to bioinformatics, to GWAS, to microbiome, to structural biology.
In order to do a lot of big genomic studies, you either need to do whole genome sequencing or you need to do genotyping bioarrays. Genotyping bioarrays is still cheaper than whole genome sequencing, and the data is much smaller and easier to manage. We started looking at which arrays existed, and we found that most of them were actually designed for non-African populations. Because we all came from Africa, African genomes have had a lot more time to evolve. So the diversity is bigger and the haploblocks are much shorter in African populations. So we decided to design a new array for the H3Africa Consortium, and H3Bionet was tasked to do this.
We developed the chip by first gathering whole genome sequence data of African genomes. We then developed a next generation sequencing pipeline to process the data, and after that we went into the process of selecting the content of the array.
The H3Africa array has more than 2 million genetic markers which cover the entire African genome. The array will improve the quality of the outputs because it has a lot of ancestry informative markers which helps us resolve the African subpopulations. This in turn helps with genome-wide association studies.
The chip has been manufactured for consortium use in the first instance. Already we've seen at least 15,000 to 20,000 samples being run on the chip. Illumina is very happy with the quality of the data. Our great aim is to empower African scientists to analyze African data in Africa. We need to train bioinformaticians to be able to analyze data as bioinformaticians. And then we also need to train scientists to know how to analyze and interpret their data.
H3Bionet offers data management and analysis training in a range of ways. We have both online and face-to-face training that we offer.
The way that we reach numerous audiences and remote audiences around Africa is that we opt for multiple delivery training models. Our Introduction to Bioinformatics training course is done with a multiple delivery approach. We have online classrooms that register with us, but within that classroom we also have a teaching assistant and a systems administrator for on-site support.
H3 Bionet is supporting H3Africa research projects by helping them to analyze the data. We're providing tools and workflows for them to actually run the data. And then when they finish analyzing their data, they send it to us. We then map it to ontologies, harmonize it, and then we help them to submit it to the public repositories.
We study the bacterial composition of the microbiota in African children. We're particularly interested in the respiratory microbiota, and we believe that the composition of that microbiota is key to maintaining respiratory health and lung health in African children. When we do microbiome studies, we generate huge quantities of sequence data which need to go through well-validated and established pipelines. And H3 Bionet's helped us set up those pipelines, help train our scientists in how to run those pipelines and understand the outputs of those pipelines.
My research focuses on mainly genomic variation in African population from a medical point of view. We, at the present time, study sickle cell disease. With H3 Bionet, we have some training, a collaboration specifically in bioinformatics and in computer science and in population genetics. We hope to have a genetic risk model that will allow us, when a child is born with sickle cell disease, to adapt our intervention.
We've managed to build a critical mass of bioinformatics on the continent. We're building a reference data set and an infrastructure as a platform from which people can then do better science, better genomic medicine, and have the skills to do that.
If African scientists get trained to analyze their own data, they are able to deliver precision medicine to their own local populations. That's where H3 Bionet has played a key role, taking that ownership of African data back to African scientists.
The Institute of Human Virology Nigeria IHVN is a non-governmental organization that focuses on HIV/AIDS related problems in Nigeria. It was established as an affiliate to the Institute of Human Virology, University of Maryland School of Medicine, Baltimore in 2004. In 2015, International Research Center of Excellence ISA was established with Professor Abimiku as its executive director to promote collaborative research in diseases of relevance to the local community, non-communicable diseases, and emerging infectious diseases. IHVN moved to a 7-story, 2-tower edifice to support research activities in ISA. The Institute of Human Virology Nigeria H3Africa IHAB Biorepository Initiative project is directed by Professor Alashley Abimiku. IHAB is the West African Regional Biorepository for the Human Hereditary and Health in Africa Consortium. Established in 2009, IHAB supports the H3Africa Consortium network of African scientists by providing high-quality, well-annotated biospecimen to study genomics and environmental determinants of common diseases with the goal of improving the health of African populations. IHAB collects, processes, stores, and distributes biospecimens within Africa and beyond. IHAB receives both processed and unprocessed samples from researchers within and outside the country. Biospecimens include whole blood, urine, saliva, stool, meconium, DBS, cervical swabs, nasopharyngeal swabs, breast milk, and so on. With over 150,000 collections, IHAB utilizes robust FreezerWorks 2019 LIMS system to automate biospecimen, data, and freezer management in a secured system. Facility is well equipped, conducts sample processing for research projects from plasma, sera, DBS, DNA extraction and QC, cell characterization and PBMC isolation. Peripheral blood mononuclear cells, PBMCs, are made up of lymphocytes, which are B cells, T cells, NK cells, monocytes and dendritic cells. IHAB supports both manual and automated viable cell counting. The TC20 automated cell counter accurately counts cells in one simple step using an innovative autofocus technology and sophisticated cell counting algorithm. The TC20 enables accurate mammalian cell counting in less than 30 seconds. The BD Fax Caliber offers a unique modular approach to flow cytometry and allows users to perform both cell analysis and sorting in an innovative single benchtop system. iHab supports both manual and automated DNA extraction. The QIAGEN QIAcube HT enables automated mid to high throughput nucleic acid purification in 96-well format using silica membrane technology. The system provides reliable DNA, RNA, mRNA extraction virtually from any sample type. The user-friendly software allows for easy data management and documentation. It is convenient, flexible, safe, and minimizes cross-contamination. The NanoDrop 8000 allows scientists to quickly and easily quantify and assess purity of samples such as proteins and nucleic acids using an 8-channel pipette to dispense samples onto an array of pedestals to analyze 96 samples in less than 6 minutes. The qubit fluorometer quickly and specifically quantifies DNA, RNA, and protein. It is easier than ever to determine if you have sufficient nucleic acid or protein for your experiment. Qubit assay dye binds specifically to DNA, RNA, or protein, making it more specific than UV absorbance. eCube is an all-in-one DNA, RNA electrophoresis device, including gel electrophoresis chamber, LED illumination, live display, documentation by integrated digital camera, and software for image capture. Eye specimens are stored at -40 C, -80 C, and in liquid nitrogen tanks. iHab uses a combination of manual temperature chart and Tutela automated monitoring system to monitor the temperature of the storage unit. Tutela provides 24/7 monitoring status alerting system via phone call, SMS messages, emails to responders to track temperature excursion. IHVN has the capacity to produce liquid nitrogen. The hallway showcased the abstracts written by iHab staff, and the office is equipped with a conference room. iHab is a secured facility with CCTV camera, monitors, and trained security guards.
To conduct genetics and genomics research, researchers need to obtain human biological samples such as blood, saliva, hair, etc. From these samples, the DNA is obtained and studied to understand why and how we are affected by certain genetic diseases. They may also collect other measurements such as weight, height, blood pressure, blood sugar levels, etc. The process involves many steps, and it is very expensive and requires collaborations between local researchers and researchers in other countries. Most of our local research institutions are often not fully equipped or are technically challenged to do all the genetic analysis. The collaborations between scientists across Ghana and other countries enable data to be obtained and analyzed to understand the genetic basis of diseases more quickly for the benefit of all. We will now take you through the journey of your sample from sample collection and transportation to local laboratories, initial sample analysis and processing in the labs, and storage and transportation to more advanced labs, and finally to biorepositories. The journey of the sample starts from when it is collected in the community or clinic setting. The purpose of the research and the sample collection will be explained to you by the researcher or field staff. Your participation is your personal decision. If you agree, you'll be given a consent form to sign or thumbprint before the samples are taken. Blood is the most common sample collected for research purposes, but you may also be asked to provide saliva, urine, or stool. Blood samples can be taken by pricking your finger with a small needle and dropping the blood on a slide or special paper, or by putting a needle through your arm or vein and drawing a quantity of blood using a small tube designed for this purpose. The tubes are labeled with a number that is unique to you to protect your privacy. The quantity of blood to be taken depends on what it is going to be used for. It could range from about half a teaspoon like this, or as much as 10 milliliters, which could be split into 2 tubes. An average human has about 1.2 to 1.5 gallons of blood like this. And the maximum quantity of blood collected for research purposes is about 10 milliliters like this. The samples are carefully packaged and transported to the local laboratories, such as the one in the Navrongo Health Research Center in northern Ghana, or the Noguchi Memorial Institute for Medical Research local lab in Greater Accra for storage and initial analysis.
Welcome back, everyone. Could we settle down and close the doors? We need to get started. Thank you so much. Please close the doors. Okay, so we can start. Is it? So we've put a short dedication together to acknowledge the colleagues and friends and family that have been lost over the course of the project. Many of them worked side by side with all of us and helped make H3Africa successful. We regret that they are not here to share this moment of celebration with us. Some of you have sent photos and tributes. We acknowledge that there are many others for whom we do not have pictures, but let us just take the next few minutes to remember them, thank them, and carry their spirits with us into the future. Thanks. Thanks so much. So we'll have Jumo and Alashley chairing the next session, the sustainability. Wow.
Alashley and myself will share this final session, and we changed the presentations around a little bit because of slides. So first, I'd like to invite Xavier David to talk about unlocking the African genetic diversity.
Yeah. Okay, good afternoon. Bonjour à tous. It is a real pleasure for me to give you this presentation for many reasons. Firstly, I have been involved in this H3Africa consortium since 2018, and I had the chance to participate in 4 different conferences: Rwanda, Tunisia, Ghana, and this year, Nigeria. Secondly, I presented to the steering committee the V2 of the chip. It was on Tuesday, 21st April 2020. And that day was special for me. And that's why there is a special relation between H3Africa and myself. So my talk will be split in 3 parts. So first part, Illumina your partner since inception. Secondly, what has been achieved. And thirdly, what's next? That can be part of the discussion. So based on the conclusion that the content of the chips at that time which were available did not fit the particularity of African genetics, you, as a H3Africa Consortium, decided to build up your own chip with Illumina. The first step was to get out of the data set out of whole genome sequencing for more than 3,500 whole genomes for 17 countries and more than 17 ethno-linguistic groups. The first version of H3Africa chip was developed in 2016 and released in 2017, and a version 2 was developed later on and was used from 2020. The chip was accessible to any academic study using African samples at a consortium fixed price of $55 per sample. Building a chip is complex and needs a close collaboration between Illumina team and the end user. In the development of the H3Africa chip, Nikki's team worked together with our ILS team in San Diego, which was in charge of the different steps of the development of the chips. The first point is to check the design score of each probes to be related to each SNP. The second point was as well to develop the gen train to be able to deliver the final manifest and the classifiers. As some probes design were new because they covered new regions of the genome, it was sometimes quite challenging. Here is the contents overview of the chip. Genome-wide backbone around 2, which was the most important part of the content with 2 million, the clinical research part 200, and the quality control. It is a high-density chip with more than 2.2 million of markers, and as a consequence, there is only 8 samples per chip. Another examples of close collaboration was between Illumina Lab and H3Africa Consortium, where the use of our lab facilities in San Diego for the first project using H3Africa chip. Why? Because in 2017, there was no platform available on the continent. So more than 21,000 samples have been processed in San Diego during 2017 and 2018. What has been achieved together? The first aim of the first goal of this project is to produce accurate genomic data for African population. So let's have a look on the figures. More than 62,000 of samples have been processed with the chip until end of 2021. You see, until 2019, it was with the V1, and from 2020, it was with the V2. 2 main years of project, 2018 and 2021, and we can expect until 2022 that the distribution between the V1 and the V2 will be the same in terms of distribution. What is the difference between V2 and V1? It is the same chip, and as a difference as the V2, you decided to add 3,000 markers, which were quite relevant for particular regions of the genomes. Another achievement is building capacity on the continent. 2017, 1 eye scan has been donated to H3Africa. You decided to place it at CPGR, Cape Town, South Africa. So since end of 2019, despite of COVID time, about 5,000 samples have been run there, which means, which is until now the most active platform involved in the H3Africa genotyping project on the continent. But besides this decision of H3Africa Consortium, it has an impact for all the continents. 2018, you see only one iScan, one platform for genotyping. It was CPGR. And you see 4 years later, some more in Nigeria, in Gambia, and in Egypt for array-based solutions. And besides this, since beginning of the year, on the continent, there is now capacity built for whole genome sequencing in South Africa, in Nigeria, in Egypt, and Senegal. Oh, sorry. Another achievement is all this data were used for research publication. So it was a real booster in terms of number of publication based on data generated with H3Africa. More than 250 have been done, which help to bring more diversity in human genomic in human genetics data provided by the research. Even it's a good achievement, we still know that there is still a lot of effort to be done, as Ambroise mentioned it this morning. And what's next? The Illumina continuum has been used for the H3Africa chip. On one hand, you discover you use whole genome sequencing to discover the novel variants, and then you use array-based solution to screen with a massive power of scale. We can look on things at the population level. But what's next? We have two options. There is maybe more, but let's discuss two potential options. The first option is to develop a new tool dedicated for fundamental research. That means, as Michel Ramsey told us this morning, there is still a need to continue the GWAS studies projects. So there is a need of high density of markers. That can be an update of H3Africa content to keep consistency, and to include the latest discovery to improve the cover of African diversity. That's the first option. The second option could be a dedicated tool for translational research. That means we don't need so many markers. We can focus on clinical research content. Then in that case, we have more high throughput workflow with more samples on the chip and capacity potentially to deliver end-to-end solution. Of course, a most cost-effective test due to the size of the number of markers per sample. But do we have enough covering of African diversity, either for GWAS or to validate the clinical content on efficiency on the African populations? If we look at the map, it was how the 3,500 African samples were dispatched at the beginning, at the origin of the data set used for development of the chip. You see there is some gap, north of Africa and central of Africa. So there is a need to fulfill the gap, and some particular ongoing projects, whole genome sequencing, will be necessary, and we are already ongoing for the next years, such as Agenda, for example. Just to conclude, I would like to just to detail the Illumina commitment. Come back. With Africa. So, and it is a consequence of the H3Africa Consortium experience. We increased our presence. Before, no Illumina employees were based in Africa. Now we have people in South Africa, we have people in Nigeria, we have people in Egypt, we have people in Kenya. Secondly, as you see, there was some infrastructures built either for array-based solutions or whole genome sequencing, or even for small genome for microbiology. Human genomes, so you see now there is capacity even in Africa to do some whole genomes on human genomes, not only with a high throughput instrumentation, and that's based on a short read technology, even if by the end of the year we should launch a long read solution, which will be functional on our actual platform. And lastly, we beside human genome, there's as well I would say involvement in infectious disease. We did it during the COVID time, and we did it as well we do it now as well for TB and HIV. So thanks to our common experience with H3Africa Consortium, we are contributing daily, always more and more, to unlock the power of diversity in genomic data. So thank you especially for Cora Vacher, who was the person who participated in the development of the first version of the chip. And thank you to all the people who helped me to gather all this information. Thanks for your attention. Merci de votre écoute.
Xavier, so thank you very much for excellent presentation. I gather that you have to leave now, and so we'll say a big thank you to you and to Illumina, and we hope to continue our collaboration. Thank you. A couple of questions.
Yeah, I can take. I'm already late, so I can take one or two questions.
Can I ask a quick one here? Chair. A quick one. One of the things that H3 Bionet has done is to really demystify the ability to work with large data. And I know Illumina is really working very hard in being great partners. But I think one of the things that would be, like during your presentation, if you could also share representatives that you have in the different countries, so that we have that kind of engagement and more engagement with Illumina that would help those who want to really get into the field to be able to do that. So some trainings as well, in addition to what H3Africa is doing.
Yes, I agree with you. We have some initiative in terms of training. That's something that we are starting to do. And there is some plan as well for Africa. But for that, we not reinvent the wheel. We already set up a lot of infrastructure, such as Ashraf Ionet, but a lot of things. We just, if we can contribute in some materials or experience, that we are ready to implement that.
Yes, merci Xavier. Lots of us in Africa, we relied on the MiSeq, so we are quite keen in Africa to have low throughput to middle throughput sequencing. Now here you present the iScan for arrays and also the NovaSeq 6000, but in the middle of this, I mean, what do you have, and will there be a successor to the MiSeq more or less?
So in the middle of MiSeq and NovaSeq, you have the NextSeq 2000, which is already the good compromise for exome and microbiology, I would say. And there is as well some access for data analysis inside the sequencer. But so far as know, because we are the last people to be informed, because we are on the field. There is no new successor to the MiSeq. Maybe sooner, but at the moment, no. It's still the one which is the most popular in Africa. You're right. There is, I think, more than 100, something like that, which is a good, I would say, compromise in terms of price of capex and capacity to run small genomes.
I think maybe one last question. Okay, thank you. So getting the MySeq is one thing, and getting reagent is another thing when you are in Africa. So are there any plan from the company so that some countries that are not covered by for the moment by your company, do you have any plan to make access to reagent easy for researchers like me from Mali?
No, that's a very good question. Just to give you a small background, until 2020, there was only one distributor for all, two distributors. One is a distributor for all Africa except South Africa, and was based in Dubai. So you can imagine the way to distribute was not optimal. Now we have 10 different channel partners based locally, African channel partners with African employee. Based here in Nigeria, here in Senegal, here in Kenya, in Egypt, Tunisia, Morocco, etc. Uganda, Rwanda. Of course, there is still some gap. We are working to fulfill this gap and to have at the end one distributor per country, because you have some in Africa, even if you are not only diverse in genetics, but as well in custom rules, in importation rules. It's each country has its own administrative rules, and you need to have a local person to deal with that with efficiency. And yeah, with efficiency. So no, it's a good question, and we are working on that, but it takes time.
Thank you, Xavier. I think it'd be really helpful if you could put the companies representing you on your website, so there's no conflict. So we can go to your website and find out who's representing you in Ghana. I think that'd be very helpful.
Okay.
Thank you. Thank you very much, Xavier. Really appreciate it.
Have a nice evening.
Thank you. We've changed around the program a little bit. So I'm going to be calling on Professor Michelle Ramsey to give us a talk about the future of genomics in Africa. And I think just for correctness, I should say that Ambrose had to leave early. But Michelle, you've been with us since the beginning, and we look forward to hearing your thoughts.
Okay, is this better? Excellent. So the first thing Ambrose did when I saw him after 2 years of COVID was to say, hey, Michelle, I have a job for you. Because he knew in the beginning that he would be leaving before the session would take place. But I'm really very happy and honored to share with you my reflections about the future of genomics in Africa and realizing the potential of genomics in Africa. So we're all here because we believe that there is tremendous potential for translational genomics in Africa, and that this could be transformative in reducing the burden of disease on the continent and alleviating poverty that is exacerbated by the high prevalence of infectious and non-communicable diseases. So my reflections today are going to touch on 3 different areas. So I'm going to talk a little bit about discoverability brought about by access to genome sequences from people living in Africa. I'm then going to talk about a view from the ground. What does this really mean for families with genetic disorders? And then I'm going to talk about the potential for accelerators of genomic and precision medicine in Africa. So first of all, in terms of discoverability, so African genomes harbor the highest genetic diversity and potential for novel discovery compared to those from other populations worldwide. So despite increasing data from African studies in global reference databases, data from just over 400 whole genomes from 50 African ethno-linguistic groups yielded over 3 million previously unreported variants. And whereas European and Asian individuals on average each have about 4 million differences from the reference genome sequence, African individuals have 5 million. But understanding the link of this variation to disease susceptibility would not only benefit Africa, but it most certainly will benefit the vast African diaspora and all populations globally. So as an example, a recent COVID-19 host susceptibility study for severe disease published by Huffman et al. In Nature Genetics this year showed that European patients had a protective haplotype of about 75 kilobases on chromosome 12, and that this area was derived from Neanderthal genomes. Using trans-ancestry mapping, and specifically due to the low linkage disequilibrium of African genomes, they found that a single splice variant in the OAS1 gene is responsible for a 23% reduction in the risk for becoming critically ill from the SARS-CoV-2 virus. So the protective G allele has a frequency of 58% in Africans, but only 32% in Europeans, and likely predates the split between Neanderthals and modern humans about 500,000 years ago. So when planning large projects, there are many decisions that need to be made. And we're all familiar with the tensions. Do we prioritize quality over quantity? Do we go for depth rather than breadth? And this applies to developing plans for a grand African genome project on a scale not yet imaginable. But first, we need to think about the state of play with regard to genomes globally. So last year, we saw the first telomere to telomere haploid genome, but a set of diploid genomes from one individual is within sight, but has not yet been achieved. I think people often misunderstand that, and they think, we can have many telomere to telomere genomes. So the reference human genome build 38 is widely used to call variants, but its accuracy is being challenged. And a recent study by Wagner et al. In Nature Biotechnology revealed errors in the reference human genome sequence. And when they mask those errors, the accuracy of variant calling improves by at least 8%, and much higher for some types of variants. So I think our ability to find these variants is still not optimal. So the Human Pan-Genome Reference Sequence Project proposes a multi-layered graph to enhance variant calling and accuracy. And there's been a keen awareness that this project will require the inclusion of diverse world populations, especially those from Africa, to better represent global genomic variation. Therefore, we need to consider what approach would give the highest yield in terms of understanding the depth and breadth of African genome variation and its link to our phenotype. Should we be focusing on building a human pan-genome with detailed telomere to telomere African genomes on several thousand people before attempting to scale to millions? To be truly game-changing, an African genome initiative would need far more than just millions of genomes. As we grapple globally, and since the first complete or almost complete human genome in 2003, it is really the function and the link between genetic variation and the phenotype that is so difficult to understand. And what we know now through a lot of research is that for complex diseases, there is not an immutable link between variant and phenotype, because sometimes the same variant can cause higher risk, and sometimes it can be protective depending on context. And we know that this relationship is influenced by the environment and by the genetic background of the individual. It is therefore vital that an African genome initiative at scale include data on demography, behavior, environment, health history, and many phenotypic measurements and other data. So I want to transition now to a view from the ground, and this is about people and families, and it touches on prevention, diagnosis, treatment, and maybe someday accessible and affordable cures for severe genetic diseases. So accurate diagnosis requires clinicians trained and dedicated to identifying individuals and families with monogenic disorders. It requires a laboratory infrastructure or access to laboratories. Often it requires imaging facilities and other special tests. And this requires expertise to interpret the results. So doctors and patients need resources tailored to be effective in their environments to treat patients for the best outcome. Would they have access and can they afford it if these were available? So in the approaches to address monogenic disorders, there's a need to explore prevention strategies. And I had a very interesting conversation earlier today. We know so little about the socio-cultural perceptions and possible acceptability and role that prevention could play in Africa. So in the early 1980s, in a predominantly Catholic population in Sardinia, a prevention strategy was implemented for beta thalassemia. And this reduced the number of affected births and freed up the health allocated purse to support patients with many other conditions. We are getting more insights into prevalence and distribution of sickle cell anemia, with an estimated 6 million affected individuals in Africa. So we need to ask the question, could a prevention strategy that identified at-risk couples and offered them pre-implantation or prenatal diagnosis lower the prevalence and thereby provide more resources to treat affected individuals? I think we need to explore this a little bit more, and I was very happy to see that one of the questionnaires presented by the students was really asking about acceptability of prenatal diagnosis. So I think very relevant research to be done, but very complex in Africa because of cultural issues. So precision medicine and genome editing shows promise for a cure for sickle cell anemia. And who of you were not excited when you heard in mid-February the story from Jimmy Olagere and how his life was changed through genome editing of his blood cells. So this was ex vivo genome editing. And he says of his experience, it's like being born again. But he lives in the US, and because he was amongst the first 7 people on the clinical trial, cost was not an issue. But how much did it cost? And would a child with sickle cell anemia in an African village have any hope of a similar intervention? And I think Ambrose touched on this earlier, that if we really want to think about gene editing in Africa, it would need to be in vivo gene editing, because he mentioned that there were only 2 countries in the continent that actually have the facilities for bone marrow transplantation. So there are several initiatives, for example, the I Hope initiative, which is a partnership between Genetic Alliance and Illumina, which has pledged to dedicate 1/3 of their resources to Africa to support the discovery of mutations from whole genome sequences and to support centers of excellence on the continent while also supporting families, an extraordinary initiative. But then again, I ask myself, are there enough doctors in Africa who can dedicate their efforts to participating? And do they have the time and the resources even to navigate the challenging regulatory frameworks in their countries to make this possible? The hurdles are enormous. So I just want to go on now to what are the potential accelerators for precision medicine in Africa? So late in 2020, Kate Davies, who's a co-chair of our H3Africa Independent Expert Committee, approached me to write a review for the 30th anniversary of the journal Human Molecular Genetics. And she suggested the title, Genomic Medicine Comes of Age in Africa. I think mostly based on what she's seen happening here in our H3Africa consortium. But on reflection, despite the enormous progress that we have made, I could not in all conscience and with conviction make a case that there was a significant clinical translation in the field of genomic medicine in Africa. So the title for the review became a question: has translational genomics come of age in Africa? It makes for sobering reading, this review, which I hope some of you have read and will read. And it highlights the severe paucity of genetic services in Africa and the poor health infrastructure in many regions in Africa to support genomic medicine, because in the absence of that infrastructure and support, you can't implement. And that is really important for all of us. So in the review, we highlight multi-level accelerators for genomic medicine in Africa. And these include, as many of you have said, capacity strengthening, but not only that, also infrastructure development and dedicated funding and support for sustainable implementation of genomic medicine. And this will not be possible without the political will in individual countries. and legislation and policy development to support this kind of implementation. And we also make a case for public education. I think many of you have spoken about that, and creating an awareness which can be done by advocacy groups, patient advocacy groups who can lobby their governments and focus a lens on the extraordinary potential of genomics and precision medicine for the people of Africa. So the H3Africa Consortium has shown the world that exceptional science does come out of Africa, led by African investigators and addressing local problems and health concerns. We have strengthened capacity on many levels, with thousands of trainees being exposed to analysis of genomic data, and some becoming leaders in the field of bioinformatics over a short 10 years. And this bodes well for the future, but is it enough to impact the lives of patients? We need more people like Leon Muteza, like Gita Langori, and many others whose work directly impacts patients with genetic disorders and their families. So Leon has managed, through extraordinary adversity, to develop both a human genetics service in Rwanda and a research program that supports dozens of students. And we need more people like them. So when we think about the future of genomics in Africa, we need to reflect and to take a hard look at translational genomics on our continent. We've seen extraordinary contributions from African researchers to understanding the genomic etiology of infectious pathogens. They have generated large-scale surveillance programs and diagnostic laboratories to monitor outbreaks, and have worked successfully and tirelessly with local governments and health systems to contain these outbreaks. And how extraordinary that we in Africa were the first to sequence and characterize the Omicron variant, despite the fact that it did not arise in Africa. So I think we need to take our hats off to the groups of Christian Happy and Tulio de Oliveira. They've done an extraordinary job, and this is certainly part of precision medicine in Africa. So I think we can really say that thanks to H3Africa, there are now many excellent examples of genomics research in Africa, as evidenced by the presentations that we've seen here at the 19th meeting, but also previous meetings. And we can see a bright future when we look in front of us at the next generation of really talented young researchers. I really enjoyed those presentations. And I think we need to marvel at the energy of the youth and the ideas that they bring. And I certainly look forward to watching their innovative approaches and their entrepreneurship as they carve out a bright future for the translation of genomics in Africa. Thank you.
Michelle, thanks for that. Very insightful, interesting, and showing the word forward and having pointed us a little bit backwards. But please, can we have some questions, comments? Can you take a few? Whilst people are thinking, I haven't quite got my head around the message about Neanderthal gene variants and resistance to COVID as being different between Africans and Europeans. Can you explain that?
Sure, yes, it was such an interesting paper. So when they first found this association, it was association of a very long chromosome sort of segment that they couldn't find the causal variant.
Yeah.
But it was only when they looked at African ancestry populations, those were not even populations from Africa, but they were African Americans. And what they could then show was that the association was not with this whole 75 kilobase region, but was actually with a single variant that was a splice variant. And the paper beautifully outlines how that would affect the biological pathway towards severe disease from COVID. And I think that's what we want to do. We no longer just want to find associations, we actually want to understand how it contributes to the biology. And this paper beautifully did that, but it wouldn't have been possible if it wasn't for these African ancestry populations and for them being able to isolate the African part of the haplotype and pinpoint that one mutation.
Thank you. Any other Yeah.
So this is somewhat related to what you were just saying about precision medicine. And I think if Ambrose were here, he might say something about this, so I'm going to say it for him. So the Science Council of the World Health Organization has come out with recommendations for accelerating the use of genomics for global health. And the recommendations were actually open for public comment for the last 2 weeks and closed yesterday. And so we did send round a link to it, but it was when probably everybody was traveling and might have missed it. And I just want to say that Ambrose got an email that the H3Africa Sustainability Group has been invited to make comments, even though that public comment is closed. So I think we should be working on that fairly rapidly because they are coming out with this in July.
Any other raising of the hands? If not, then Michelle, thank you very much. Really appreciate it. Okay, thank you very much. The next presentation is by Dr. Roger Glass, Director of the John E. Fogerty International Center.
Well, I'm Roger Glass, Director of the Fogerty International Center at the National Institutes of Health. And I'm delighted to be with you, even though I can't be in person. But I wanted you to know what a huge supporter I am of the H3Africa program, and very pleased to see what you as investigators in this program have accomplished over the past decade. Now, Doctors Eric Green and Francis Collins have already spoken about the many successes, so let me provide my own perspective from the Fogerty International Center. I particularly want to recognize the wonderful support I've had by our project officers, Doctors Laura Pavlich and Barbara Sina. The sustainability of the H3Africa program and really the genomics initiative in Africa has been a constant concern for all of us involved with this program from the very beginning. How can we sustain your research over the long run, make the results relevant to all, and build programs to improve health that will really benefit us all? Sustainability for us is building and maturing the research workforce to create the knowledge and ideas, to seek funding, partnerships and support so that these ideas can be implemented and move forward. And finally, linking these results to national priorities to improve health for infectious disease control, chronic diseases, child health, and more. So besides the many discoveries that you've all made over these 10 years, the papers published, the insights gained, how have you matured as researchers, communicated your findings to the public, and translated your research and results into public health practice and public health diagnostics, treatments, and cures that will sustain this endeavor in the long run? How have we done that? Think sustainability. Last November, Fogerty and many of our NIH partners launched a new program, Harnessing Data Science and Innovation in Africa, DSI Africa, a 5-year, $75 million award to link data science and innovation to the major health priorities of your countries. We were surprised, I should say delighted, that of the 19 competitive awards that were granted, 10 went to researchers in the H3Africa community. Recognition that the data science and innovation are not only critical for genomic research, but research on many other diseases. These grants will ensure funding for many of your centers to continue to expand your work for the next 5 or 10 years. And while extending this to topics of cancer, non-communicable diseases, child health, infectious diseases, and more. Let's take the infectious diseases. COVID, for example. The fingerprints of H3 African investigators and NIH awardees are found all over the response of African researchers to this most disruptive, indeed devastating pandemic of the past 100 years. The first strains of COVID were sequenced by your colleague, H3Africa investigator and DSI grantee, Christian Happy, within days of identifying the earliest known patients in Africa. And the capabilities with genomics have been rapidly applied to COVID diagnostics, to tracking the evolution of virus strains around the continent, and involving many of you and other Fogarty trainees in this endeavor. Another major initiative derived from genomics has been this partnership with the Wellcome Trust and NIH on sickle cell disease, also a legacy for the H3Africa program. Your genomic and data skills will be central in research ranging from surveillance of disease, of sickle cell disease, and the diversity of its complications, to mapping the genomes in many countries in the region, and then linking that genomic data with the severity and outcomes and complications of this disease. Of course, the ultimate goal of this research would be to find a cure for sickle cell disease, hopefully through some form of gene-based therapy. So ultimately, understanding of sickle cell disease and its cure and many other single gene disorders will come from the roadmap in human genomics that you will be discovering through this work. Dr. Collins repeatedly states that all the people in the world are Africans, having descended from ancestors who have either remained in Africa or migrated out thousands of years ago. We have so much to learn about our genetic heritage in order to understand human health and disease. As African researchers in genomics, you are demonstrating the critical role you can play in this exciting endeavor, this unfolding story, and how much you have to contribute to this global roadmap for research that will eventually help us all to improve the health of people around the world. Thank you very much, and I'm looking forward to this session and where you will go next in this incredible endeavor. Thank you so much.
Thank you. It's my pleasure to invite Colette Dandere to tell us a little bit about the DBAC and how much has been accomplished.
Okay. Good evening, colleagues. I want first to really congratulate you on the work that you have done, on the milestones that you have achieved, and the journey that you have taken with H3Africa. I think just like you, I'm also in anticipation that I hope that you will continue to find some ways to sustain this momentum that has been created, which has trained so many students. My job today now is to really talk to you about DBAC, the Data and Biospecimen Access Committee. This is your data. We are just there to help you make sure that it is governed and accessed in a way that actually does not that benefits you as well as Africans. So I'm giving this presentation on behalf of the committee and also our chair, Prof. Bayomi. Okay, now in terms of H3Africa data governance, remember when we're talking about governance, we are looking at access and use of data or biospecimens. The structure is as follows. Let me see if the where DBAC sits here, we get our documents or our standard operating procedures and all the information that we use from the steering committee. So the steering committee of the H3Africa decides on what they want, how they want their data to be used and how they want it to be accessed. And we are there to act possibly just like policing that the data is accessed in a proper way. If you see, there are different ways of accessing data. In some systems, data is not available at all, but new systems now require that data be released as fast as possible so that it can be used and it can be beneficial to humankind. However, if you really look at the issue associated with H3Africa, it is with the thinking and the realization that Africa does not have the same capacity to analyze or utilize data as the rest of the world. So the steering committee came up with some ways to manage that process, but at the end of the day, having also a time period when the data will be released anyway. So giving the people who have generated the data, people who have recruited participants and all that, time to exploit that data because of the limited capacity in terms of data analysis. So so our duty is to try to create to balance that need that data must be released. But also African researchers might not be at the same, might not use it at the same pace compared to the rest of the world. And then if you look in terms of our committee, we are set up as a committee of 9. I think we have never operated as 9. I think we've always been missing someone. So we hope we'll get now. Currently, we are also missing one. But what we supposed to have, the expertise we are supposed to have, is as outlined there, and we are supported by ex-officio members, one PI, an independent expert committee member, and a representative of the funders as ex-officio. So here are the current members of DBAC. So we short of one member, so we have Rosalind will be actually conducting the steering committee to find us a member, and that member who we're missing is the one who was the patient advocate. So if you can start thinking about that. We are supported by the Science for Africa, SFA, the new organization, and Rosalind is quite helpful in the background, trying to make sure that DBAC works. So I think I'll leave the issue about why do we want to about data governance. We're all aware of that. I think I want to go into the issues about how do we evaluate or requests when they come. There are several documents that are available for people who want to access data. They are available on the H3Africa website, and I think some documents can be sent when an individual is requesting data. And so the samples that are highlighted, we in red, we really look at them also seriously as DBAC to see whether there is adherence to that. there are areas of reflection that we want to do with the committee, especially on some of the issues that we discussed in the last meeting that was face-to-face. And just to add as an example, just a bit of also communication, because after our last iteration of concerns, the steering committee, I think, went on to finalize the document, which is their role, and uploaded it, but we didn't get the finalized document. And so we were using a document that could have had a bit of variance in some items, not much in terms of the some of the aspects. So there was a bit of a miscommunication on that, because the documents were uploaded in 2020, when the last one we had is the ones that we had worked on in 2019. So, but it's not a major issue. This has not really affected our work. So let me go to the next. So when we do the evaluation, we look at various criteria, and this includes the standing of the PI who is requesting the samples in terms of their skills. We look at whether they also they've got funding, then also ability to do the research that they say they want to do, and one of the issues we try to push the people requesting data is to try to think about how the accessing of data can benefit Africa. So we push on that and try to find out if they can reach out sometimes to researchers in Africa, the benefits for Africa. So when accessing the data, I think we are helped by the Secretariat to really look at the applications when they come, and then they look at if there's anything missing before that comes to us. But at the center of when we look at the requests that come is we're looking at whether there is no harm that is going to be done to the different participants that are being requested on the data. All of the requests require that they have got ethics and also reviews from their institutions. And we also look at whether the science is good science. We're quite a diverse group, so it's quite helpful. So this is the process that a request goes through. Where when a request comes, it goes through the Secretariat here. Sorry, the highlighter is not working well. And then they do all the checks. And when they are satisfied that the request has got all the necessary documents, completed the documents well, then it's released to the committee for members to review, and then it depends whether it's a data request or a specimen request. Sometimes we request some modifications. We send back to say, can you, or clarifications. And then in some issues also, in some instances also, we ask the Secretariat to confirm with the PI if we think that there's a bit of overlap on what already the PI has indicated that they are working on. So the Secretariat does that. And then once we approve, then the Secretariat manages the MTAs and the data access issues. So we have not yet received any biospecimen requests. So you can see all the requests we have received at the moment are actually data requests. And most of the hard work is done on the recruitment and getting of data. We also we look at the plan for how if anyone is to ask for samples, whether they are going to be able to retain whatever they might not have used, or how they are going to destroy such samples. So, like I say, the data requests are managed by the Secretariat, and let me now go on to what we have done. So let me go to the. So we also hold regular meetings as a group online due to the to COVID. Also, particularly when there is a request that most members are disagreeing on, we actually hold a meeting to discuss and then come to a conclusion. We make decisions based on a quorum, and our quorum is 5 out of the 9, but there are certain individuals or expertise that must always have participated in that quorum. If we not meet in quorum, sometimes it might take a bit longer than our turnaround time, so that we accommodate the member, but we've always been able to meet our quorum. I think we have not encountered conflicts yet. So we hope we continue working and also discharging our duty in a manner that is fair, because we really want the story of Africa to be told through your work, but at the same time we also want you to be benefiting from your data. And the H3 BioNet is quite helpful in terms of if there are any issues in terms of there were some issues where some people might not be able to access the data and all that, they are referred to BioNet. There are a list of things that, areas where we think might be areas for conflict of interest, so that as DBAC members, as we are also part of the scientific committee in Africa, we do checks on ourselves when these requests come, or if we are even collaborating with those people who are asking for data. I think one of the things that we would also require a bit of discussion with these committees now, maybe we need to hear what the steering committees stand on commercial companies and all that is in terms of the data access. Because I think at the last meeting we left that we had a discussion, but there was no clarity in terms of exactly how the steering committee feels it should be treated. Let me go there. So these are all the checks that are done by the Secretariat. I'm not going to bore you too much with that. Let me go on to what we have done this year. So you can see in summary, at the end of May, we have received a total of 20 requests. And of these, 9 have been from the US, 4 from South Africa, 2 from UK, and then a single each from the other countries. So yes, requests are starting to come in. And if you look at those 20, we had 1 which we rejected. There's a 1 straight rejection. The 5 had major revision that was requested. Then we've got 10 conditional approvals. That's also, I think we need to possibly communicate, because when we give conditional approval, sometimes there are small things that we want to be clear with the applicant, so that at least once those issues are cleared, then the approval goes. Then there are 2 that were approved without any iterations back to the applicants, and we have got 2 that are now under review. We just received them a few days ago, so we already have started to work on those 2. So if you look at the one that was rejected, that is the one that was collaborating with a commercial entity. So that's why I said we should make that a point of discussion so that there is clarity on that, so that we might start to get those types of requests as more people become aware of the data. And also the request that really made significant changes to the data access agreement, to the point that it became very difficult for us to accept that. When we ask for, let's say, when we give conditional approval, sometimes we'll be wanting to get clarity on issues on the objectives. We want sometimes to look at how they are going to be managing the data, and also benefits for Africa. So we debated. We've got a list of things that we consider. If someone indicates that it is, we consider that is beneficial to Africa, and then we do give also the conditional approvals. So some of the requests that we got one from Uppsala University. They have already successfully downloaded the data. One from University of Constance cited several issues, and I think, Nikki, that was communicated to you, and I think that was sorted, or they indicated that they didn't find the data useful. So, but maybe they were requesting the wrong data. What can we say? The one from Cape Town was successfully downloaded, and we really appreciate where the requesters indicated, the training of students and all that. And then one from the University of Texas also, they have not done much because of COVID. So hopefully they're still in the pipeline and wanting to use the data. So what we have learned is that for now, there are no biospecimens that have been requested, which means your gold here is data, because nobody is going to ask. It appears like people are not going to ask for the biospecimen. So that's your gold. So now I think it's now to look at if the release of the data. I think if there are any issues that you think are being are coming out of the release of the data, we would really want to hear from you so that we can assist. And then we really would like to see Africa to Africa collaborations on this data. And also sometimes we are very, we do ask about African students' involvement, even if someone is, let's say, in the US, and if they are going to be taking an African student there, and all that, we give that positive as positive. But we are not going to, we are not rejecting, I want to make it clear, we're not rejecting requests where there's no African, let's say, clear African benefit without discussion with the applicants. We do enter into discussion. So I think on this one, on commercialization, we really want the steering committee to assist in or come up with a position so that we can, otherwise we would be using our own judgment in terms of giving access to commercial entities should they approach us on that. So that's I want you to know in the back of your mind as you go that that's a sticking point that you need to resolve and assist us so that we can help you better. So thank you very much. And if there are any questions, I would appreciate. Thank you.
Thank you. Right on cue. Thanks very much. Really exciting to see some of the movement in terms of the request. Any questions to comment?
While I'm waiting
Yes,
for questions, can I just acknowledge my colleagues in the audience who are here? Amd, are you here? Can you stand up? We can see you. Where is? Who else is around? Amd? Yes, please come. Stand. and then we also have a third member. I'm forgetting the name. Daima, yes, please. So Robin and them are our new members. Thank you.
Thank you. We've got a question here in the front.
Thank you so much. This is a great initiative and well appreciated. And the fact that we are not getting requests for the samples tells a lot. It tells a lot because some of us have worked in Africa, have struggled so much to collect the samples, but once we have the data, the requests are enormous. So I think as a continent, we need to find a way that enables us to develop, to mine our data in a process that benefits all of us. In a situation where you have so many people asking for the data and very few supporting generation of the data, that's really precocious. And I would prefer or recommend that we have more support to generate the data. We recently had a discussion with the WHO asking to share the data. And my first question was, where does that data come from if we are not supporting generation of the data? So I think more effort should be also focused on generating the data so that we can have something to share. If we don't have the data, then what should we share? My second point on that is commercialization. I think most of us who are working on research in Africa, it has been charity research. Less mining, less discoveries on the data. I would recommend for the committee and most of us working on the continent to come up with a strategy not to be averse to commercializing entity, but rather develop mutual agreements and understanding that enable us to commercialize the data, but at the same time benefit everyone involved in that process. And I think companies like 54 Gene and others are working on that, and we are happy to work with them to come up with a consensus that benefits the researchers and even our communities, because if we keep sitting on the data, it also counterproductive, and we are not helping the communities where we collect the data from.
Thank you so much for that comment. Is that a response to the comment? Sorry. Okay, I can't see Nikki. She's got a huge column in front of her.
Just on the commercialization, so we went through the discussion and we have updated the documents, but I think it's probably another case of it they didn't reach you. So there has been an update on that. And also each data set is mapped to DUO, and the DUO code will say whether commercialization is allowed in the consents.
Thanks so much, Nikki. One at the back there.
Yeah, so I have a question about this group that's actually downloaded the data and then they terminated the agreement. Imagine that I have the data on my computer. So how actually the debug make sure that they are not using?
Yeah, it's a very good question. We did discuss about also in our meeting yesterday about monitoring and evaluation of the use of data. We usually we wait at the end when we see publications that are coming out, and then see whether they've used the H3 Africa. That's the only way we think is possible. Colleagues, fellow committee members, can you help with this? Anyone who wants to input? We have got lawyers in our committee as well, so we
Yeah, I mean, this is one of the points we discussed a lot about how we can monitor what the people requesters are doing with the data after they have downloaded it. And so there is no real way to check for that. What we can do is they give us their feedback, like saying, I successfully downloaded, analyzed, published the data. So this is one way to check it. Otherwise, we have no real way to do it.
Thank you.
So can we have a question at the back there? Oh, you're okay. There are mics going around. You don't have to.
Thanks, Colette.
Thank you, Colette.
So I just have a comment and then 2 questions. So my comment is, I think for me, it's a little bit concerning, and thank you to the DBAC for all their work, but it's a bit concerning for you to say that nobody's ever going to ask for the specimens. If our DBAC doesn't really value our specimens, then it's going to be hard for us to convince other people that they really do have value as well. In terms of my 2 questions, so since most of your requests, at least 50% of your requests have been sent back for requests for further information, I'd just like to understand a little bit better what the scientific review process from the DBAC looks like. Because obviously there has to have been a scientific review process at the receiving institution. And my understanding was primarily the role of DBAC was to make sure that there was no inadvertent or inappropriate use of the data that conflicted with the ethical approval. And then my second question is, since the majority of these requests have been for data rather than specimens, I just would like to understand why it's necessary to have a biobanking specialist on every committee that constitutes a quorum.
Okay, Colette, do you want to respond, or you want Jen to go first before?
Yeah, let's have Jen go first, then we can respond to all the questions.
Go ahead, Jen.
Yeah.
Oh, yeah. So some of this is probably related, but I would say also I wouldn't put too much emphasis yet on it, because really I think most of the requests have been for the Baylor data set, which has been published on and publicized and very popular and was in EGA before anything else was deposited. And so I think there's just not yet an awareness about additional data beyond that about the biospecimens. Not all the biospecimens have been deposited yet. Not all the data has been deposited yet. So I think we won't know until many years out what the community finds most valuable. So I would not cut off biospecimens as a valuable commodity just yet. And then the other thing that I wanted to say, Colette, was I had actually completely forgotten, so thank you for putting it up there, that there were ex officio members, non-voting members, and that some of us could sometimes sit in on these. And I think that might be useful for us to have a couple meetings where there are representatives from the various groups so that we can help to iron out some of those issues as they come up in real life. And go back to the steering committee then with if we need to revise policies or whatever. So I think remembering that is very useful. And then my final thing is actually a question. So sorry, everything else was just comments, but this is actually a question, which is, I guess I was a little confused about since I thought we had specified between data and biospecimens that one was a finite commodity and had to be much more protective than the other, which was an infinite commodity. But it sounds to me like you're applying the same standards to both. Is that correct, or am I wrong about that?
So, let me start by answering Dr. Main, Prof. Main at the back there. I think we are not underplaying the value the biospecimens. We're just indicating what has been requested and trying to think why it is the case. We're not underplaying that at all. But also remember, characterizing biospecimens comes with a lot of expenses as well. That could be a reason. But no, they're quite valuable. So what has been said, that once you start releasing more and more of your data, more and more researchers will be aware of it, and we might actually get to requests. And then coming on to the process of review. In the committee, we are varied in terms of our expertise. We've got some with the legal qualifications and all that. We do almost like oversee like what other ethics committees do, but we mostly based on documents, H3Africa documents and international practice. So yeah, each person actually reviews so when the request comes, each person reviews it independently, and then we share our reviews after we've done your review. Then you can send them, and then we share, and then we can discuss and if there are any issues that are picked. So we use that diversity in skills to try to see whether, especially in terms of the use of data where we think there might be harm. There are international examples where data has been used in a way that possibly harms participants who gave that data. And then your question was, let me go.
The difference between how you treat the data requests versus biospecimen.
In essence, the two of them, data and biospecimen, we treat them the same. What we are on the biospecimen, we are much more stringent in terms of capacity. But on the data, we push people to try to think what could be a benefit to Africa, and sometimes we give them a conditional approval. They can come back. They discuss their issues, but the stringence on the two is a bit different. But most of the rules is just the same. Yeah.
This is very exciting, but we will take one last comment or question from Philip.
Hi.
No.
Oh.
Yeah, Philip, please.
I'm right here. Hello. Hi.
Okay.
So yeah, great to hear those last comments as well. I mean, in my view, like there's always a bit of mission creep that might happen where you don't necessarily want to be taking the place of the ethics board. If somebody's already coming in with ethics, then I think the role should be largely looking at risk, particularly for just data. And I liked what you were just saying before. That's really, and that can be said also for the biologics. And I agree that you want the biologics to be something of a legacy. You can't, it's your way of future-proofing to everyone, really. New technologies that haven't been developed, questions that haven't been asked yet, or you don't even know what those questions are yet. So you want to have that there. And usually if somebody's coming in to use a sample, they had to write a grant, get the money. And so, it's still very, so.
Thank you.
Wonderful. No, I'm going to round it up now. Otherwise, we are never going to get out of here.
Michelle wanted to squeeze a question.
Michelle, no. Yeah, we'll have wine later. So talk over wine. You all have been very. Thank you again. Thank you very much. You all have been very, very patient, honestly, and staying on. Thank you so much. I think the presentation by Nikki would be the last formal presentation we will have for the evening. And Nikki is going to get us to think a little bit more about the long-term plan for data storage. Over to you, Nikki, and thanks again for all you do.
Thanks. I'm going to be super quick here. So this is just to get, so we were going to have a discussion about this, but I don't think anybody's really in the mood for discussing. Maybe have a glass of wine. or two. So I think I'll put some thoughts into your head, but it's something that we do need to think about because, there's all these thoughts, there's all those discussions about, well, this is not the end, this is not the end, but actually it's the end of the funding. So in some cases it is the end. Your project, many of your projects, unless you've got a no cost extension, will be ending soon. So we need to think about what happens next. So I'll just talk about what are H3Africa's assets, what should we sustain, and how to do that. So we have, you saw all the policy documents, we've got guidelines documents, standard CRS, SOPs. Some of these are just sitting on people's computers or on websites that people can't find. Some of them are on the H3Africa website, but who's going to maintain the websites if we have to close the doors and turn off the lights for H3Africa? Somebody's got to maintain those. We have training materials. We manage our training materials and make them all available, but I think the whole consortium has a really valuable, probably a valuable set of training materials. They're probably sitting in many different places. Biospecimens, these are available, but they're also a finite resource. And then we've got the phenotype data, genomic, omic data, and then LC project data. So some of those first things we can do very easily. We can just make sure that they are up on a stable website for as long as possible. We could give them DOIs and put them on Zenodo or Figshare. If they have a DOI, then they're at least citable, and then you get recognition for those. So I think we should do that anyway. Our training materials we make bioschemas compliant and put them on a website so they're completely fair. And we put those on GitHub as well. So we can think about where this sort of set of resources goes. The biospecimens are obviously in the biorepositories, and these guys are going to need long-term support so that the fridges don't turn off. The phenotype and genotype data. So technically this goes to EGA or ENA, but not all the data is going there. So actually the phenotype data that gets submitted is actually a small portion of what you collect. So what happens to the rest? What about when your PhD student who has saved this on their hard drive under their desk moves on to a postdoc somewhere else? That data is going to get lost, and it's extremely valuable data that I think took a lot of effort from a lot of people to collect. So perhaps it's worth thinking about what's going to happen to that. LC project data, the reason this came up was because Alice Matumbo, who had an LC project, she said to me, can we submit our LC data to the archives? I said, well, no, because there's nowhere for us to put it. And she said, but she's got all these data from interviews she's done. It's a lot of data. Her project ended, and actually she'd love to make that available to anybody who has an LC student who wants to have an MSc or PhD data to work on, but she doesn't know where to put it. And we didn't know where to tell her where to put it because doesn't fit in the archive. So I think there might be data sets like that are actually a resource for the consortium or beyond the consortium. We just need to figure out how best to store that. So with the genotype and phenotype data, we know that it goes, this is how it works now. We've got it goes to the archive, which goes to ENA, DBSNP, EGA, and it's all accessible, searchable via the catalog. The phenotype data, some of it has been submitted to ClinVar, but actually that was more genotype data. It was actually from the genome sequence data, but hopefully you're submitting your fantastic results to ClinVar. And then there's the chair resource, which has just been amazing for some of the phenotypes for some of the projects. And then we're trying to put as much of the summary genomic data into AGBD. But what happens in the future is, we've got the archive, somebody's got to, we can actually delete the data in the archive if we disappear, because there's a copy in EGA or ENA. But actually, EGA has come to us several times when the DBAC has agreed to has approved a request, and that person is requesting it from South Africa. They say, just give it to them from the archive, because there's no point in them downloading the data from Europe, from the UK. It's actually sitting in Spain. So we need to think about whether we need to keep the archive for that kind of purpose. The other thing we've been discussing is potentially to have African versions of these. So everything is based in the US or Europe. So there's the NCBI and there's the EBI. And actually we've had discussions, so EGA is trying to have a federated EGA, particularly where data sets cannot cross borders because of national protection laws. So this is happening in Europe. And so Europe's got setting up a number of federated EGAs, and they're really keen to have a federated EGA in Africa, and potentially the archive forms that. We've spoken to the head of the DBGAP, and they said that they're also looking at these trusted partners, and potentially we could be a trusted partner. They've never done it outside the US, but they are open to that discussion. And generally, if you look on African repositories, I think there's one recognized core seal trust, core trust, whatever it is, recognized African repository. So maybe we can think about whether we want to have one of those. But then for the phenotype data, could chair be extended for the other phenotypes that are not going into EGA? So that at least they're kept somewhere and become a valuable resource, even if it's just within the consortium. The results from your genotype to phenotype we can put into HMP, so we'll collect that from the literature. But wouldn't it be great eventually to have an African genotype to phenotype resource? Michelle's had led discussions to think about this before. So these are things we can think about in terms of where to store this. Then the other H3Africa project metadata, the other valuable information about projects. So some of the solutions, just grab the set of files, archive them, and have a README that says what they are, so we don't forget, because everybody names files differently and you don't know what files contains what. For the phenotype data, we could extend the chair REDCap database or have some other kind of database. For the metadata and some additional values or data associated with the projects, we could actually store and have searchable in the catalog. We also have this new, the DSR Africa Open Data Science Platform, and this is based on the Alwaz, it's called Alwaz, it's based on the Terra system, where you have these workspaces, you log in, you find data sets, you extract data sets, and then you run these. And what we could do is actually have a workspace, an H3Africa workspace in there, and then put all the data sets in, so you can actually have a single place to see the data sets, analyze them as well. But just remember that it doesn't mean it's open. So we can have a collection of H3Africa data sets and assets in there, but only people who have access to each data set within there can access that data set. So it doesn't mean it's open. It's as close as it is now. It's just sitting in one place that's findable and much more usable if you want to, particularly if you want to work across projects. So that's another option. So things we need to think about, and not to discuss now, but to think about, is there a will or a need to sustain these other assets other than what goes to EGA and ENA? How do we fund these? What data resources that are not in public data should we keep long term? Are you happy to just keep them in your own project and assume that if you ever need the data 5 years down the line, you'll find it? How and where should be stored and maintained and funded, and who will be the custodian? So if it does go into a more central place and everybody's gone on to other projects, who should sort of discuss the access and manage the access to that? So those are just a few points to think about, so that the gold that we have generated, that you guys have generated in this consortium, has value long term for yourselves, but also for other people. Thanks.
Thank you. Thank you so much, Nikki. And I appreciate the fact that you've given us a little bit of time to have some discussions. So, so it's open for some ideas. Yes, go ahead. Oh, Bernie, yeah, please. Mike and Michelle, you can ask your question. I didn't see you because Michelle was blocking Michelle.
Thank you, Nikki. I have a question for you that also relates to the previous talk about the biospecimens and the data that's associated with it. So as Michelle mentioned yesterday, you can do a lot with a lot of power now to look at P by E analyses, where the E can include a lot of multi-omics, exposomics. There are many, many, many biomarkers of exposures that you can measure in existing samples. But to do that, you need information on how the samples were collected, the timing of them, what temperature they were at, for what time, what tubes they were collected in, and those sorts of things. So I'm wondering if there's any details on that, and if not, if that could be collected and integrated into the existing resources before the opportunity is lost.
No, so there's two sets of data collection. There's the big manifest file, big-ish, which is not so big, manifest file that the biorepositories collect, so they'll know better exactly what individual components are in there. And then a subset of that gets submitted to us, including information from the biobank, like how many aliquots there are and things. So it's actually quite a small set of metadata around the samples that gets collected.
Yeah, and what I would add to that is I think at the moment there is a possibility to go back to the clinical sites and actually have that discussion and collect some of the information in terms of the tubes and the different processes that have been used. I mean, quite a few of the sites are very clear on what they've used to collect it. A lot of it is EDTA tubes and before the processing and how it's stored and how, so it's possible, but right now the minimum data set doesn't have that, but we do have the possibility to go back to investigators and collect it if that's sounds like that would be a useful thing to have for the environmental expansion of this.
You might want to do it before everybody moves on to other projects
Right.
or forgets, I
Yeah.
think, yeah.
Yeah, okay. Yes, Michelle.
Thank you very much. And, you know, I just wanted to again thank Colette and the entire DBAC team for the work that they do, because I think, you know, it's not easy, and the decision making on our behalf so that the data is responsibly used is really very, very helpful. So my question is really sort of a hybrid between these two talks, because when people get the data, they often generate new data. And my question is, what happens to that? And, you know, if it doesn't go into something like the GWAS catalog, but it's actually something that could be useful, you know, we always said it has to come back to H3Africa. So where do we put it? Who looks after it and makes decisions on its behalf?
So for the, in the data biospecimen access release policy, any data that is generated off specimens that are used from the biobanks has to be resubmitted to EGA under the H3Africa umbrella, under the H3Africa project. In terms of new data generated off data, there's no requirement. So I think that will be summary statistics. So we could probably encourage them in the data access agreement to submit that to the GWAS catalog or to a public repository. Actually, I don't know, maybe I can check if there is wording about that. I think we do encourage them to, you know, disseminate their results and data, but I don't think there's a requirement.
So just wanted to mention that about the fund that the coordinating center has to support some of the projects issues with data QC, I think, and getting that data into the H3A Bionet archive, and then perhaps also assist with analysis. So we do have a fund, and we will reach out to those struggling with that aspect of the project.
Yes, I mean, one of the uses of that could be if there's a decision to pull the data sets and, you know, curate them and have them. No, it doesn't have to be in a central place, but at least sort of we know where they are. Then that could be a use of those curators.
I was just going to ask Nikki that, thank you for bringing up all those points. I think it's really a good time to have it. And I wonder maybe it's you know, as the steering committee discusses some of the documents that DEMO has put together in terms of the H3 consortium and moving forward, it's probably not too early to begin to ask some of the questions that you have highlighted at the end of your talk.
Yeah, because then I think you're starting off a new a consortium with a set of assets, right? Because, you know, we don't have money to generate new ones going forward. At least that consortium, the new consortium, will have a set of assets already associated with it, which is valuable for now leveraging to get new funding, I think.
Okay, right. If there are no further questions, all that remains is to thank Nikki very much for really very important conversation. And then I'd like to hand over to Zanil Lombard and to Michelle Skelton to finish off.
Thank you so much. Before we go ahead with acknowledgements, I am just going to give our IEC member, Philip Awadala, just this chance to briefly give us some notes and feedback of his thoughts about the meeting. Thank you.
Thank you very much. So it's with great pleasure on behalf of the IEC that I congratulate H3Africa on another wonderful meeting. And it's just astounding to see where we're at now in the 19th meeting compared to where we were, or what I saw from when I first came to a meeting in and Addis so many years ago now, it's just not even recognizable now. And so many of the thoughts and recommendations that the IEC made along the way, I don't even have that opportunity now to make them again, because they've been so many of them have been addressed. So, what I saw over the last couple of days now has had focuses around training, discovery, translation, and sustainability. And I want to say a few things that are perhaps different than, and I don't think will be contentious, but some things that might be considered helpful, particularly around training as well. So it was great to see such great science. And again, the scholarship is high among the students and the postdocs, and we heard that the energy was high as well. So one of the things I would like to recommend to our young students or PhD students who are in the beginning of their programs or in the midway through their programs, or even postdocs that are here, is to really start taking the bull by the horns, if you like. Get access to the data that's already being made available. It doesn't have to be data generated from just within your own lab. There's data now being deposited in the various data sources. Use that data to think big about the questions. And you don't even have to even think about that in the context of the data that's being deposited in the H3A BioNet repositories. Go out, get data from UK Biobank. Go out and get data from those other sources. There's plenty of great projects that can be done as a PhD student with data that's already out there, right? So think big, ask big questions. Some of the best PhD projects are in projects that aren't necessarily newly collected data. One of the things that I was really struck by as well from hearing some of the talks, as well as just, that the future of genomics in Africa is really bright. And it's great to see that there are 66, almost 70,000 genotypes now available. And so we heard that just a few hours ago. And, a few years ago, that looked like that wasn't necessarily going to be achieved. Now we're at that point. So again, that's there for the students. Go get that data. You can analyze that data. Learn some new techniques as well. I know there have been courses offered in machine learning, and you've had a course earlier on R. If you're armed with those tools, using R, wielding data with a few Python scripts, you can get any job you really want, really. And so once you've got those tools, you can start asking bigger questions as well. So, with even those 70,000 genotypes, a few genomes that are available, that are out there now, you can go get more genomes from other public repositories. The world is yours. With that said, I want to just emphasize that there's still some space, there's some learning here for, we heard about polygenic risk scores, we heard about EQTL mapping and transcriptional and transcription. And again, those machine learning tools will help you think about new ways to translate some of those findings, not just to the clinic, but to your participants as well, right? Some of those tools will be helpful. It's not that far afield, right? You can think of new ways using those tools to potentially use them to translate and be really pushing the personalized medicine forward, in Africa. I just wanted to echo a comment made by Michelle about the importance of good phenotyping. As well, we're already near the end of this project, and we've captured a lot of information from our participants. Clinically assessing participants in terms of outcomes is almost the easy part, but capturing lots of those other variables, those environmental variables we just heard about a moment ago, so some of those socioeconomic variables, they're just as critical to understanding how disease progresses. And with that information, plus a strong biobank, where you bank material, and people may not have yet developed diseases, but might develop diseases in the future, will allow you to go back, use that data, see if you can identify factors that may have led to the disease before, and then develop new tools to capture or prevent those diseases from happening as well. So prevention is just as much a part of personalized medicine as much as curing. So, going forward, start thinking about that as well. And the last piece of advice I wanted to mention, think about a little bit, was around sustainability. So lots of discussion. It's great to be thinking about that now. It would be great, and I think there's a sustainability committee, or a committee that does meet regularly. invite onto that committee decision makers from organizations that you feel are going to be important to you. They may not come with a check right now, but if you get somebody who's at Gates to sit in on some of those meetings, some people might come from industry to sit in on some of those meetings. They can open more doors for you, so they can be part of the committee as well. We've done that for some of the programs that I'm involved with, and that has helped us get in the door before things get launched, particularly at government, particularly with funders like Gates. Bring in NIH, but you can always bring back NIH and the Wellcome Trust to these committees as well. So that's where I wanted to, those are some of the thoughts that I had. It's been a fantastic meeting. The one thing I wanted to highlight was it's incredible to see the number of individuals trained and incredible number of publications as well. And they're not, publications, like counting them this way is obviously important, but where these publications are landing is great as well. We're seeing these are high impact papers. That's going to be critical. That's the currency that's going to help with funding going forward. So, just kudos to all of you. That's just been wonderful to see today. So again, thanks very much. And I will turn this over to Zane.
Thanks so much for this. Okay, let me see if I can figure this out. No? There we go. Right, so I just have a few acknowledgements and then of course the awards for our wonderful fellows. So I want to start off by acknowledging the significant role that the H3Africa Steering Committee has played. This leadership role has been really important in driving the scientific agenda of the consortium forward. And I think each and every one of the people on this slide has really helped us to steer the way that H3Africa has come along these past 10 years. So Duomo, Alashley, Michelle, Enoch, Clement, Nikki, Bohomotsi, Christian, and Ambrose, thank you so much for the contributions that you've made. And I look forward to taking this forward and learning from you in the next year. So thanks. Thank you, everybody. The next group I just want to acknowledge and thank is, of course, all our working groups and the chairs of those. I think the working group has really played such a significant role in amplifying the voice of African researchers that made up which makes up the consortium, and really coming up with not only driving the agenda forward, but coming up with practical solutions and policies and guidelines that really made sense and helped us to implement the research that we really wanted to do as part of the consortium. And of course, also driving the collaborative nature of H3Africa forward by overseeing and really steering forward some of the cross-consortium efforts that everybody was involved in. I also want to thank our independent expert committee. This is really such a privilege to have these Hall of Famers come to the consortium meetings, really give expert advice and have conversations with all of us. I think this has been an excellent opportunity for us to learn and to engage, and I'm just really thankful for all of you who have taken the time to come to the meetings and to engage with the consortium members. And of course, our funders, absolutely essential part of the consortium. And it's gone obviously way beyond just funding us, but really advising us, guiding us, showing us new opportunities and making us think about ways to make the impact of H3Africa Consortium just so much more significant. So thank you to all of you as well. Thank you to our Data and Biospecimen Access Committee who was represented here. I just want to acknowledge all of them. Colette gave a great thought-provoking presentation now, and I think is going to lead to much more thinking about how we share our valuable data and biospecimens, how we engage with our bank managers of the biobanks, and how we really make sure that this invaluable resource is just going to grow in value as we go forward and we engage with people across the globe. So let's move over then to the Travel Fellowship Awards. And here there are so many names, as Philip pointed out. There are so many wonderful trainees that have formed part of the consortium and that the coordinating center has been able to support to come to these meetings to not only share with you the wonderful findings that they're making, but also to learn from these and to network. So there's 2 slides worth of names here. As you can see, this has been a wonderful effort, and I'm ending with the final one of the 19th meeting where the coordinating center was able to support 40 fellows, and we really enjoyed all your talks. Thank you so much. And these are the award winners from our wonderful presentations that we heard. Excellent speed presentations. Our runner-ups on Tui Ya Achia, Sheikh Kise, Natui Vincent, Modibo Goita, and in first place, congratulations to Ruth Nanjala for a wonderful presentation. If you are still here, we ask those fellows to come up to the front to receive your prizes. I know many of the fellows have had to leave already, but I'm hoping some of you are still in the audience. Okay. Maybe they're taking a coffee break after all the hard work that they were doing. Thank you so much. Well done. Excellent work. Okay. We'll do a picture hopefully afterwards. Oh yes, one more. Congratulations. That's it, right? Okay, this is a really, really big thank you and acknowledgement to our coordinating center team, to Tanyan, Rolanda, Francis, Confidence, Shumi, Michelle. Huge round of applause. Thank you so much for all the effort that you put into bringing these meetings together. I'm just amazed at every meeting I come to that everything is just so perfectly organized and such a smooth experience. And I really don't know how you do it. So thank you once again. It's been a wonderful meeting, as all the other 18 of them have been. Thank you so much. Yes. And then last, but certainly not least, what would the H3Africa Consortium be without the members? And at this meeting, we've had wonderful engagements, wonderful presentations. I thank each and every one of you for the engagements, for the discussions. It's been so wonderful to see everybody face to face again and to have opportunities to catch up with our family. This is a wonderful consortium of people who are ready to do great science, and I really look forward to seeing what else we're going to achieve over the next 10 years. So congratulations, everybody. Thank you for your commitment and for your leadership, and thank you for this meeting. Thanks.
We have some gifts for our host. Lashley, please come and join us. Madame. Woo.
Thank you. That's very nice.
Sophia, please join us. Are you still around? Don't hide away. Yeah. Thank you so much for all your support. You're amazing. Oniki Pefoladen, are you here? Thank you so much. Thank you, everyone, and I hope you have a safe journey home. Thank you.